Hello, hello,
Hello, hello, hello, hello, hello.
ವರ್ಷಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇಲ್ಶಾಲೆಯ ಚಿಕ್ಕ ಹೊಟ್ಟೆಯಲ್ಲಿ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949ರಂದು ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಮಹಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯು ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶವಿದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿರುವುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ H. ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಹಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾರೆ. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ. ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೊಂಡರು. ಮಾನಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈ ಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡಗಳು ವಿಸ್ತರಿಸಿದವು. ಹೀಗೆ MGM ಕಾಲೇಜು ಹಂತ ಹಂತವಾಗಿ ಯಶಸ್ಸಿನ ಪಥದಲ್ಲಿ ಮುಂದೆ ಸಾಗಿತು. ಉಡುಪಿಯಲ್ಲಿ MGM ಕಾಲೇಜನ್ನು ತೆರೆದ ಬಳಿಕ ಡಾಕ್ಟರ್ ಮಾಧವ ಪೈಗಳು ಜಿಲ್ಲೆಯ ಬೇರೆ ಬೇರೆ ಕಡೆಗಳಲ್ಲಿ ವಿವಿಧ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭಿಸಿದರು. ಇಂದು ವಿಶ್ವದ ಪ್ರಮುಖ ವಿದ್ಯಾ ಕೇಂದ್ರವಾಗಿರುವ ಮಣಿಪಾಲದ ಕಾಲೇಜುಗಳು ಹಾಗೂ ನೆರೆಯ ಜಿಲ್ಲೆಗಳಲ್ಲೂ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭ ಮಾಡಲು ಮೂಲ ಪ್ರೇರಣೆ MGM ಕಾಲೇಜಿನ ಯಶಸ್ಸು. ಈ ಎಲ್ಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳಿಗೆ ಮುಕ್ಕುಟಮಣಿಯಾಗಿ ಇಂದು MGM ಕಾಲೇಜು ರಾರಾಜಿಸುತ್ತಿದೆ. ಶೈಕ್ಷಣಿಕವಾಗಿ ಮಹತ್ತರವಾದ ಸಾಧನೆಯನ್ನು ಮಾಡಿರುವ MGM ಕಾಲೇಜು ಉಡುಪಿಯ ಸಾಂಸ್ಕೃತಿಕ ಚಟುವಟಿಕೆಗಳ ಕೇಂದ್ರವಾಗಿ ಗುರುತಿಸಿಕೊಂಡಿದೆ. ಕಾಲೇಜಿನ ದ್ವಿತೀಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೂಶಿ ಹರಿದಾಸ ಭಟ್ಟರು ಆ ಹುದ್ದೆಯ ಘನತೆ ಗೌರವಗಳನ್ನು ತಮ್ಮ ಪ್ರತಿಭೆ, ಪಾಂಡಿತ್ಯ ಮತ್ತು ಪರಿಶ್ರಮಗಳಿಂದ ಹೆಚ್ಚಿಸಿದರು. 2010ರಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಮೌಲ್ಯಾಂಕನದ 4ನೇ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ TA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ನ ನೂತನ ರಿಜಿಸ್ಟ್ರಾರ್ ಡಾಕ್ಟರ್ ರಂಜನ್ R ಪೈಗಳು, ಕಾಲೇಜಿನ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾದ ಶ್ರೀ T ಸತೀಶ್ U ಪೈಗಳು ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಸದಸ್ಯರಾದ ಡಾಕ್ಟರ್ HS ಬಳ್ಳಾಲ್ ಮತ್ತು ಶ್ರೀ ವರದರಾಯ ಪೈಗಳ ಸೂಕ್ತ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕಾಲೇಜು ಹೊಸ ಸಂವೇದನೆಯತ್ತ ಸಾಗುತ್ತಿದೆ. ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ H ಸುಂದರ್ ರಾಯರಿಂದ ಇಂದಿನವರೆಗೆ ಹಲವಾರು ದಕ್ಷ ಪ್ರಾಮಾಣಿಕ ಪ್ರಾಂಶುಪಾಲರಿಂದ ಸಾಮಾಜಿಕವಾಗಿ ಸ್ಪಂದಿಸುತ್ತಾ ಸಾಧನೆಯ ಪಥದತ್ತ ಸಾಗಿ ಬಂದಿದೆ. ಪ್ರಸ್ತುತ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ K ಲಕ್ಷ್ಮೀನಾರಾಯಣ ಕಾರಂತ ಅವರ ನೇತೃತ್ವದಲ್ಲಿ ಇಂದಿನ ಸಾಮಾಜಿಕ ಅಗತ್ಯಕ್ಕೆ ತಕ್ಕಂತೆ ಅನೇಕ ಅಭಿವೃದ್ಧಿ ಕಾರ್ಯಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡು ಸಾಧನೆಯತ್ತ ಆಶಾಭಾವನೆಯನ್ನು ಹೊಂದಿ ನಿಂತಿದೆ. ಅಮೃತ ಮಹೋತ್ಸವದ ಸ್ಮರಣಾರ್ಥ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಪ್ಲಾಟಿನಂ ಜುಬಿಲಿ ಕಟ್ಟಡ ನಿರ್ಮಾಣ, ಮುದ್ದಣ ಮಂಟಪವನ್ನು ಮೇಲ್ದರ್ಜೆಗೇರಿಸುವ ಮೂಲಕ ನವೀಕರಣ, ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಕೌಶಲ್ಯಾಭಿವೃದ್ಧಿ ಕೇಂದ್ರದ ಸ್ಥಾಪನೆ, 75 ವರ್ಷಗಳ ಸವಿನೆನಪಿಗಾಗಿ 75 ಅರ್ಥಪೂರ್ಣ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಆಯೋಜಿಸಲು ಸಂಕಲ್ಪಿಸಲಾಗಿದೆ. 1949-50ರಿಂದ 2023ರ ತನಕ ಕಾಲೇಜಿನ ಇತಿಹಾಸ ಶೈಕ್ಷಣಿಕ ವಿಷಯದಲ್ಲಿ ಅತಿ ಮಹತ್ವದ್ದಾಗಿದೆ. ಒಬ್ಬ ಧೀಮಂತ ವ್ಯಕ್ತಿಯ ಕನಸು ಈಗ ಲಕ್ಷಾಂತರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿದ್ಯಾರ್ಜನೆ ಮಾಡುತ್ತಾ ಒಂದು ಸಣ್ಣ ಶಾಲೆಯಲ್ಲಿ 89 ವಿದ್ಯಾರ್ಥಿಗಳಿಂದ ಆರಂಭಗೊಂಡ ಸಂಸ್ಥೆ ಇದೀಗ ವರ್ಷಕ್ಕೆ 3,000ಕ್ಕೂ ಅಧಿಕ ವಿದ್ಯಾರ್ಥಿಗಳು MGMನ ಅಂಗಳದಲ್ಲಿ ಶಿಕ್ಷಣ ಪಡೆಯುತ್ತಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ಸಾಧನೆಗೆ ಕಾರಣರಾದ ಪ್ರತಿಯೋರ್ವ ಆಡಳಿತ ಮಂಡಳಿಯವರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ಪೋಷಕ ವೃಂದ ಮತ್ತು ಎಲ್ಲ 75 ವರ್ಷಗಳ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಧನ್ಯವಾದಗಳು. ಇಂದಿನಿಂದ MGMನಲ್ಲಿ 75ರ ಅಮೃತ ಮಹೋತ್ಸವದ ಹಬ್ಬ ಶುರುವಾಗ್ತಿದೆ. ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲಿ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949ರಂದು ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ ಎಚ್ ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು.
I request our beloved principal, Professor Lakshminarayan Karanth, to escort Dr. Krishna Prasad, retired HOD, Zoology Department, MGM College, Udupi, to the stage. Thank you, sir. I now request our principal to florally honor Dr. Krishna Prasad. Thank you, sir. I request our principal and Dr. Krishna Prasad to light the lamp and begin the day, the second day of BIOSEM 2024. Thank you, everyone. I now request Dr. Krishna Prasad, retired HOD, Zoology Department, MGM College, Udupi, to kindly share with us his views about this seminar.
Distinguished gathering here, my friends, esteemed scholars, experienced people, students of MGM, I am most honored to be here today for this function. I have to thank Professor Lakshminarayan Karanth for the good events he has arranged in the campus. I am honored to extend you a warm welcome to all the esteemed scholars, teachers, my good friend Dr. Rajagopal, to share his experiences in this eventful function. I had worked here in MGM College for nearly 2 decades. Even more than that, I am proud to be the part of this event and wish you all a good memory of this function. I extend you a warm welcome to this event. Thank you very much.
Thank you, sir, for the kind words. It is indeed a great honor to have you with us. We are now done with the inauguration session for the second day. We'll start with the technical sessions shortly. I now request Pallavi of 2nd BSc to take over.
In the intricate dance of nature, each species plays a vital role. Conservation is our pledge to protect this choreography of life. A very good morning to one and all present here. I, Pallavi Rao of 2nd BSc, stand before you as the Master of Ceremony for the 3rd technical session of the 2-day National Seminar on Biodiversity Conservation. I would like to take this moment to heartily welcome everyone gathered here. Today, we have with us Dr. S. Basavarajappa as a resource person. I request Ishwar sir to escort the resource person to the stage. Hailing from Davangere district, Basavarajappa sir completed his bachelor's degree from DRM Science College, Davangere, and his master's degree and PhD from Karnataka University, Bangalore. Sir also has a PGDEE and PGDEM degree from Indian Institute of Ecology and Environment, New Delhi, and National Institute of Environment, Lucknow. He has been working in the educational field since 1989, teaching in various colleges and universities like University of Mysore, Kuvempu University, Karnataka University, and many more. Besides these, Sir also has a vast research experience. He is associated with Indian Institute of Science in VGNL project from 2018 to till date, and many more projects. Dr. S. Basavarajappa's contribution to the field of science are truly commendable. He has contributed in collecting the nucleotide sequences of geographically distinct species like stingless bee and Apis dorsata population. Sir's dedication to fostering a culture of lifelong learning and intellectual curiosity has inspired countless individuals. He has guided 14 PhD students, out of which 7 students have been awarded with PhD. 7 are working presently. He has guided 5 MPhil pursuing students and 80 MSc students. Sir has published 120 scientific research papers in foreign and national journals of international repute, 185 popular science articles in various daily, weekly and monthly magazines. He has also written 3 mini-books on animals-related aspects in Kannada. He is also associated with Zoology Encyclopedia published by Prasaranga University of Mysore. A distinguished individual, Dr. Espasarajappa is recipient of many awards such as the Outstanding Scientist Award 2023 at INSO Pondicherry, Nalvadi Krishnadevaraya Award 2023 at Mysore, Best Senior Scientist Award at World Biodiversity Congress 2013 in Chiang Mai city of Thailand. Sir specializes in the areas of entomology, epidology, silkworm pathology and conservation biology. It is really a great honor and privilege to have Dr. Espasarajappa sir here with us. Please join me in welcoming sir and expressing our gratitude for his valuable presence. A hearty welcome to you, sir. I request Yogesh Kumar sir, Department of Botany, to florally welcome Dr. Espasarajappa. Now, let us engage with Sir's presentation and let's benefit from his insights and expertise. Over to you, sir.
Hello, I am audible. Hello. Hello, am I audible? Hello, yeah. ಇಲ್ಲೇ ಇಟ್ಬಿಡಿ ಮೇಡಮ್. ಎಸ್ ಓಕೆ. Hello. Thank you, Miss, for your brief introduction about my journey. Good morning, everyone. Respected dignitaries of the dais, student friends, my friend Professor Ishwar Bhatt, all the students. It gives me a great pleasure to stand before you and part of this event. I am very much proud to be here to share my few ideas, I can say, or work what I have done during my journey, especially in University of Mysore and Kuvempu University. So I am going to speak on exclusively on anthropogenic interferences on wildlife and biodiversity. So I will be, next slide please, next. Okay, so my talk is centering around 4 issues. One is on honeybees, second one is butterflies, then third one is insect pollinators other than butterflies and honeybees, then fourth one is aquatic insects. Don't get fear. Is it possible to cover all these 4 aspects within stipulated time? Of course, I will try my level best to cover all these aspects. In previous days, so many speakers have focused on biodiversity problem, conservation aspects and extinction of different species. But now I am focusing on only the insect fauna. So they are living in and around us. What is going to happen or what is happening to those species? Many of the time, we are speaking about what is going on outside other than our surrounding environment. But I am focusing on what is going on around us, because we are living with so many insect creatures, insect species, which are partly part of our day-to-day life, and directly or indirectly they are connected with us. So they are part of our welfare also. So With this brief background, again I am not going to, already Dr. Arvind yesterday talk and even keynote speaker in the morning spoken about various aspects of the reasons for biodiversity decline and wildlife decline. So, but just to prelude few slides I am just showing. See, the development as human becomes civilized, the problem started from that particular point of time. So, Anthropocene during this particular period, 8,000 years ago, so the man started developing farming activity and also started moving to different parts of the continent. As a result of that, agricultural activity, animal husbandry practices are gradually expanded at different parts of the world. So, and at the same time it is also coincide with the industrial revolution, and as a result directly indirectly various anthropogenic activities affected biodiversity and wildlife. Next. So, at the same time man has also started inventing so many things to make his life convenient and comfortable. As a result of that, he started exploiting, overfishing so much of life on this planet Earth. So, till date we have 7.6 billion people, representing only 0.01%. But we are, after the dawn of civilization, we are losing or lost already 83% of the mammals, already Dr. Arvind made a mention very clearly. So, then in 1970 to 2014, you see wildlife loss in vertebrate species including mammals, fishes, birds and amphibians as well as reptiles. 60% we have already lost. Just we are representing 0.01%. Next. Next. So, as per the data, so 16,700 populations belongs to 4,000 species. So, these species we have already cleared, lost. Next. So, next, same thing. So, we are in the, I think today's mass extinction, that is currently Earth is undergoing 6th mass extinction, and about as per the red list IUCN, so most of the species, both wild flora as well as fauna, facing high risk of extinction. Next. So, you see here the natural, the extinction is a biological phenomenon, spontaneously it occurs. But when man is intervening with the developmental activity, with the biodiversity, so the literally the process of species extinction started from 1000 to 10,000 times more than the natural phenomenon, and every day we are losing dozens of species. Next. This is what the percent of endangeredness, all these species and all these factors facing in the nature. So, Earth surface damaging and lost, affected more than 50%. Red color indicates endangerment. Oceans this much, 70%. Like that, the kelp forests, so live coral covers 50% almost. Like that, wild mammals, endangered species like plants and threatened species almost 10%. So, wetlands, so more than 90% are facing lot of problem. So, this particular image shows very clearly where we are, how much intervened with the different natural ecosystem. Next. Of course, this is also associated with the climate change, unsustainable agricultural intensification, then trafficking and eating of wild animals, as well as urbanization, deforestation and industrial activity. These are all circling around different activities connected to human welfare, and as a result we are creating lot of threat on the existing wildlife and biodiversity. Next. So, as a result of that, so we are just making habitat fragmentation. It is called habitat fragmentation. Next. And as a result of that, wildlife declining, 37% we have exploited already, 7% due to climate change, it is a natural one. So where it is man-made activity and habitat degradation, 31% of the pristine, pure, natural habitat got degraded, and 13% we have lost habitat. Next. And as a result of that, 31,000 species are threatened with extinction. So this many hectares of forest already destroyed, then these many hectares of land of earth, it is supposed to be used for agricultural activity, farming activity and other purposes, is degraded. You cannot bring back to the original state. So such a loss we have already incurred in the nature. Next. So as a result, so the mammals are facing lot of problem, livestock is also facing lot of problem. Next. And the major cause is threat is due to habitat loss. That is, these are the three important elements, key factors for the habitat degradation. As a result, our natural ecosystem, even wild animals are facing a kind of threat and extinction. So habitat degradation, habitat destruction and habitat fragmentation. One more important activity is going on in the urban landscape and agricultural landscape, that is habitat modification. Habitat modification. Other than these three, habitat modification is also playing a very important role and creating lot of threat on existing, locally existing, especially insect species, that I am going to show you later. Next. So as a result of biodiversity is under crisis problem. So habitat loss, invasive species introduction, over exploitation. Next. And we are in Homogenocene, that is current epoch, where you see the biodiversity is diminishing, biogeography and ecosystems around the globe are almost looking alike. It is because of introduction of invasive species, because we are moving here and there, now the whole world is a small village. So moving here and there, as a result we are carrying lot of invasive species and inadvertently we are spreading. Next, these are the invasive species spreading, Lantana camera, Eupatorium, Parthenium, like this. Next. And even animals also, these are all I think yesterday made a mention. So Gaines snail and Clarias and Tilapia and Pterygoplectis fish species. So these are all invasive species and affecting our local indigenous species, their survivability under problem. Next. So next. So this is what with this brief introduction I wanted to focus on my topic, that is honeybees, butterflies, pollinating insects. So India is tropical country, so where we have highly diversified ecosystems and the ecosystems are varied and vegetation types are different. You cannot get a similar type of habitat in different landscapes. It is highly diversified. So I am focusing only on honeybees here, that too wild honeybees. So we have very good published data, lot of research work has been carried out on domesticated species, honeybees, beekeeping activity, apiculture. But very few reports are available about wild honeybees, because wild honeybees like next. Next. Why I am talking about wild honeybees? Because the wild honeybees are major pollinators and they are consistent foragers and they are feeding on plants, nectar and pollen. Of course, even domesticated honeybees also feeding on nectar and pollen of different flora. But thing is, they are not harming our environment. At the same time, they are contributing lion's share to the beehive products, particularly honey and beeswax and other products. So next, Government of Karnataka recently, after realizing the importance of honeybees, declared honeybee as one of the state insect. So then social aspects, I am not going to tell more about social aspects. It is quite interesting also, because honeybees have very good impressive traits and they lead a social mode of life. They are truly social insects and living in an advanced society. It is as good as like human beings society, and they provide continuously, consistently hive products. Next. So these are all, we have very good diversity with respect to honeybee species. So in Asia, about 7 to 9 honeybee species are distributed, and these are all one on the other way contributing a lot to the biodiversity and local wildlife. Next. So these are the species, they are diverged into 3 lineages, cavity nesters, and these are all the cavity nesters, and among these there are 2 types of honeybees, dwarf bee and giant bee. These are wild species. Next. So these are the species I am focusing. So wild honeybees. See, this is this is Apis dorsata, Apis cerana, Apis florea, and this is stingless bee, and this is Apis cerana. This is domesticated species. Okay. Of course, these 4 species are local species, indigenous to Karnataka, even India also, throughout India. Whereas we have got one more species, that is Apis mellifera. I think some of you are knowing about apiculture, where Apis mellifera is used as it is an exotic species, Italian species. It is used for beekeeping activity because of it is a very good pollen gatherer, produce more quantity of honey. Next. So these 2 are open nesters. You cannot use them for beekeeping activity. You cannot rear them under human captive condition. They prefer to survive in the wild. Whereas Apis mellifera, Apis cerana can be domesticated, or you can use them under human captive condition in the name of beekeeping activity, apiculture. So of course, all the species have very good caste system, that is queen, only one queen will be there, many drones will be there, that is male, and thousands of workers will be there. So of course, very well established caste system is there, and they lead social life. Next. That is not my part here. So this is how they prefer arboreal condition. They establish big sized nests with these are the 3 important members in a colony, that is queen, drone and worker. Okay. And of course, they show nest fidelity. I mean to say, a kind of faithfulness, love and affection towards one particular type of nest. So this nesting place is very important for the establishment of big sized colonies, particularly by wild bees. And what I shown in previous slide, that is Apis dorsata, Apis florea and stingless honeybees. So they are not sharing a common platform, especially for nest building activity. Next. So this is what especially the Apis species construct hexagonal shaped cells, where developing all these will be there, and they used part of the nest for depositing, storing pollen, and also part of the nest is used for honey storage. Part of the nest. Remember, I underline part of the nest, not complete nest. We presume that the honeybee colony means everything is filled with honey. No, it is wrong. Part of the nest, they store pollen and honey. Why I am telling, I will tell you later. Next. So this is what the typical schematic representation of the honeybee colony. You can see here the peripheral portion of the comb is with queen cell. It is dome shaped. Only 1 or 2 will be there. During breeding season, more number of queen cells will be formed. Okay. Whereas next about it, brood chamber, pollen and honey is stored on the top portion. Okay. Next. So this is how the Apis dorsata prefers big sized, tall, limbed trees, ramified branches of a tree, big tree like ficus tree, like that, and it constructs like this. This is the typical establishment, typical structure of the comb, U-shaped structure on the surface of the flat limb. Okay, this is typical. You can see here, and you can see the white portion is filled with honey. White portion is filled with, only this much part is filled with honey. Okay, we are thinking that it is completely filled with honey. As a result of that, so it is greed, human this one. So we are simply destroying the whole comb, whereby we are removing so many, we are killing so many individuals of honeybees. Next. Of course, nowadays these are all the host plant species, and even nowadays especially the Apis dorsata now modified its nesting behavior. Instead of nesting on the trees, big trees, it is preferring now on human built structures. It is because of so many reasons. Again, anthropocentric activity. So it is modified its habitat. Next. So even during different seasons on the tree limb as well as human built structures, where especially in arid, semi-arid and malnourished region we have recorded, and there is no much variation with respect to this region as far as distribution of colony is concerned. Next, this is what you can see here. So the light blue colored one is trees, and this one is human built structures, especially in arid region, semi-arid region, arid region, where Apis dorsata prefers more human built structures. In semi-arid region and in malnourished region it prefers trees, because malnourished region is known for good forestation. Next, density and abundance also varies with respect to the nesting activity. Why I am showing all these little bit later, I will come to know. Next, and one more thing, you see here the association. So about 78 families of plants which belongs to 78 families, and so many 240 species of plants are pollinated by honeybees, remember. Okay, and these plant species provide a very good pollen and nectar for honeybees. Next, using that we have prepared a pollen calendar for the wild honeybees. So do the honeybees get throughout the year pollen and nectar consistently? So this is what the graph shows, the fluctuation, variation during different seasons at different region. Okay, that means honeybees never get consistently, wild honeybees particularly never get consistently the foraging source. As a result of that, they show migratory behavior. While doing migration, they are come in contact with so many human interferences. As a result, so many colonies are declining. Next, and even we have classified like that because climbers, herbs, shrubs, trees like that, economically important, ornamental, medicinal, nectar, pollen, like that, different classification we have made. This is all to know about the existence of honeybee population, number one, and what are the different plant species we are established in and around us, that is nearby garden, college, institutional gardens, nearby forest, agricultural land, this and that, how they are going to influence the honeybee population, and in turn these species are going to get benefited. Pollination leads to propagation, then multiplication of the species or individual plant species would occur. So this we have recorded. Next. So these are all the preferred, wild honeybees preferred nest host plants. So nesting site like us, how we do prefer very good shelter, very good residential house. Same way, if nesting site is not good, honeybees never construct, never establish colonies. In the wild, I am not talking about beekeeping activity under human captive condition, in the wild, because these species provide lion's share to the honey production and hive products, but we are least bothering about wild honeybee species in the nature. We are uprooting all these are all the plants grown in different agricultural ecosystem on the roadside also, even in the modified landscapes. These are all the plants we are destroying, uprooting all these. So if these plant species are not available, definitely the wild honeybee population is going to be declined. Next. So this is how bees, you can see in a single tree colony aggregates of wild bees you can see. So they establish, if means that particular plant is providing ideal surface, there is no predatory interference, there is no human interference oftenly. So as a result they live safely. Next, like this tall trees, eucalyptus tree, you can see the aggregates colony. So now we are losing this kind of trees in many of the places. Next, and see in the coconut garden like this different rain tree. Next, even in human built structure also. Next, and you see here on human built structure on the ave of building. So such a giant colony is established, and these are all the man-made surfaces. And even in these places also, I said it, honeybees modified their nest building activity and preferring to these particular human built structures where man is oftenly intervening with their nest building activity. As a result, we are creating lot of disturbance for their nesting activity. Next. So these are all the percent occurrence, abundance, that all we have calculated. Next. This is another species, Apis florea. Whatever I said so far, it is Apis dorsata. This is also wild species. It is a small, beautifully stylish bodied creature, Apis florea. Its nest is like this, like a pot. And interesting thing is, it is not preferring big, tall, wide tree limb to establish nest. It prefers like this small sized limb, where the honeypot comb is completely encircled. So totally this species prefers tall branches with thin size, diameter in size. Okay. So this kind of comb it constructs, where honey is stored here and remaining part is brood and other things. Next. So this is how you can see on the small tree with the different height, you can see the colonies. Next. And you see here Apis florea nest at the peripheral portion of the tree limbs, not on the main branch, peripheral region of the tree. See like this, you can see here. See bamboo plant like this. Next. You can see like this small thin branches should be there to protect, so that any birds, any human interference directly should not go and attack. So masking, it is masking around the colony like that. Next. And another interesting thing is, Apis florea never construct comb on the human built structures. Remember, never construct. So far no one has recorded, but now the Apis florea is also modified its nesting behavior because of non-availability of suitable plants. So it is constructing on the temples where there is little human interference, religious places. Next. Like this you can see here. This is Apis florea, not Apis dorsata. It is a small bee. So it is also now switching towards, opting human built structures. Next. So this Apis florea prefers, these are the plant species, potential plant species for establishment of the colony, like a shrubby or herby vegetation somewhat. Okay, next. So this is, and another one is Apis cerana. Apis cerana indica, it is commonly used in apiculture, that is beekeeping activity. And now it is the time, that is especially spring season, you can, if you go to forest, so much of small swarms you can get, the Apis cerana indica swarms. And it is unusual one. And this Apis cerana swarm and small nest, you can see on the, it is not a open nester. Remember, it is a cryptic nester. Never construct open nest on the big tall buildings. Wherever crevices are there, so where it nests. So these are all survival strategies of the honeybee species for their safe survival in the ecosystem. Next. So another one more interesting bee is stingless bee. We have, so far I said stinging bee, Apis species. Now stingless bees, they don't have stinging apparatus. It is a defensing organ to protect themselves from any predators or enemies. Okay, next. These stingless bees are commonly called dammer bees in India, and they are called musre jenu in Kannada, in Karnataka, musre jenu. And of course they have their own this one, and as far as biodiversity is concerned. Next. So why we need to study about stingless bees? Because they don't sting. Do not abandon their hives because these are all establishing perennial nests. They are not migratory species. Perennial nest means throughout the year in one particular place the nest means they will be remain forever, even 5-6 years they will be remain there if undisturbed condition happens. Then easy to extract their hive products and less vulnerable to diseases and ability to pollinate small sized flowers. Apis species pollinate big sized flowers, whereas we have small sized flowers where big honeybees like Apis dorsata, Apis florea cannot get in and collect nectar and pollen. So under such circumstances where stingless bees do that job because their body is very small. I will show you next. So as far as their biodiversity is concerned, next. So they are belongs to again Hymenoptera and they are distributed in almost all America, Africa, Asia, Indo-Malayan and Australian regions. Biodiversity is so vast. Next. So this is what you see: neotropical, Indo-Malayan, Australian and Afrotropical. These are the different genera. Each genera is with so many species. As far as stingless bee biodiversity is concerned, lot of research work has been carried out in Brazil, other parts of the world other than India. Next, they play a very important role for pollination and propagation. Next, so see 461 species have been identified in the neotropical region, Afrotropical where 50, then in Indo-Malayan 90 species, in Australia 11 species. Next, and in India, next, so we have 24 stingless bee species so far described. Okay, 10 species in China and 3 species in Sri Lanka. Next, if you come to Karnataka where we have almost next 6 stingless bee species have been described. So in our area we have identified 3 species, especially next in southern part of Karnataka. Okay, that is Tetragonella pegdeni, Tetragonella vicrami, Tetragonella iridipenis. Next, so very quite interesting thing is stingless bees never construct hexagonal shaped cells. What I have previously shown, Apis species, usually Apis species construct hexagonal shaped cell. It is a what I can say space saving strategy. Whenever they establish arboreal nests where weight is one of the major constraint, because of gravitational pull the comb will fall. So to avoid that, wherever they establish hexagonal shaped cells so that they can make use effectively the space where they establish nest and they can store good number of amount of pollen, good amount of nectar as well as good number of brood. That is the strategy. Whereas these species never construct arboreal nest, they construct terrestrial nests on the land surface. And the species wise very interesting thing, so they have different shaped brood cells. So arrangement of brood, based on that we can identify different species. So take for example vertical brood comb, here this is the species, like that irregular shape, circular shape, like this. Okay, next, this is what, go back, this is what you cannot see openly. It is a cryptic nest, very difficult to locate. Next, and this is what on the exterior part of the crevices where they establish the nest opening like a tube or irregular shaped like this or small tube like this. You can see these are all the main gates, entrance tubes through which the few bees will come and going out from the colony. Okay, next. So based on the go back, based on the shape and size of the opening we can identify different species. Next, so these are all, again you see here the honey pot, dome shaped, this is honey and this is all pollen and this is how they construct cells. Next, this is what the pollen and brood chamber. It is totally different from Apis species. It is just like, resembles like a grape bunch. Next, so these are all the places where the stingless honeybees nest their colonies. Muddy wall, rock, stones, cement made items like this. Okay. So this is how you can see. So means to say, nesting structure, nesting habitat is totally different compared to epispecies and non-epispecies, that is stingless honeybees. Next. Next. So of course, to our study we have selected different regions. Go back. We have covered Chamarajanagar, Kodagu, Mysore, Hassan, Bengaluru rural, Chikkaballapura, Kolar district in southern part of Karnataka to do this particular task. Next. Next. This was the study area. Next. Of course, we have used so many multi-pronged approach, different methods, wherever whatever the type of ecosystem is there. So accordingly, we have to modify our methodology to collect what is exactly going on happening with all these natural colonies of stingless bees. We have collected lot of information. Next. So I am focusing only on human interference. Not I am here mentioning about other factors. Of course, so many other problems are there. When you are living in the nature means certainly so many problems will be there we have to face. So this is what we have just created. Next. This is what the common nesting niche, the wild honeybees, and you can see here tall trees, even tall buildings. These are all black colored ones are honeybee colonies, Apis dorsata, and these are all colonies. This is what we call Apis florea prefers the moderate height elevation, then stingless bees prefers lower surface. Means to say, next, if you go to this particular thing, it is an ecosystem where the different trophic levels are there. In each trophic level, different species are surviving without any competition. Okay. So next. So this is what they prefer, nesting elevation minimum 20 feet to 70 feet height. Okay. And Apis florea 2 to 35 feet, whereas Trigona 5.5 to 25 feet. So it is very clearly reveals that nesting habitat is totally unique, species specific. It is not same. Next. So then another interesting thing is, you see here Apis dorsata always prefers higher elevation to establish its nest. Next. And sometimes Apis dorsata, not Apis florea, prefers to establish more number of colonies. It is called colony aggregates. So like this, 2 colonies, 3 colonies in a 1 square meter area we have recorded. You can see the number of colonies. So this kind of trees we referred as bee trees, where they provide ideal habitat for the establishment of good number of colonies, number 1, and nearby that particular tree, whatever resources, particularly floral source and water source, easily available, abundantly available during different seasons. So such trees are designated as bee trees. Now what is happening with these bee trees? Do we conserve such type of trees? Should any protective measures are required to protect such type of, because honeybees, as you know, many of the plants, particularly 14% of the plant species are dependent on honeybee pollination, and our majority of more than 60% of the agricultural crops are depending on honeybees pollination. So therefore they are very important, vital to the ecosystem. Okay. So therefore their presence, their protection, their survival is very important for our human welfare. We have to protect them. But what is happening in the nature? Next. So because of these anthropogenic activities, wild honeybee colonies are declining. Here I am not telling about individual, colony as a whole I am talking. Okay. Next. See, this is what we are doing. Next. These are all once upon a time we have recorded during our earlier days time. They provided very good branch and very good number of honeybee colonies. Now after 5 years we had been there. So this is what is happening on the roadside, road widening. Next. See, go back. Yeah. Next. So, as a result, see 2006 to 2007 and 2007 to 2008, gradual decline of Apis dorsata, Apis florea, Trigona species. Next. The major reason is pesticide poisoning, uprooting of trees, this much %, trimming of tree limbs, cultural practices, conventional unhunting and other unidentified reasons. So, about 7.3% on an average decline has been recorded with respect to Apis dorsata population. Next. And this is what the graph shows. Next. So, normal colony hunting, normal colony burning, people are also burned the honeybee colonies because of fear, because honeybees, Apis species are stinging one, highly ferocious, venomous insects. If you go and disturb them, definitely they go and they will sting. Next. So, this is what is happening, man-made activities. Next. And you see the road widening, especially in urban area, Mysore. We have taken Mysore as one of the model city for studying the effect of human interference on wild honeybee like Apis dorsata population. You see, earlier it was on the footpath, but now the road has expanded as the population increases. So, as a result, all these trees, once upon a time, they provided very good surface for the honeybee colony establishment. Because of this, all these trees are uprooted. As a result, we are not getting good number of honeybee colonies. Next. So, these are all the pesticides we are using in agricultural ecosystem. You see, particularly organochlorine, organophosphate, herbicides and pyrethroids. Of course, we need to use pesticides. When we have to use, where we have to use, how we have to use. This is very important thing to make a note, particularly our agricultural friends, farmers particularly. So, especially bees are very active during morning hours. They are very busy with collecting pollen and nectar. During that particular time, if you spray pesticides, definitely flowers are getting contaminated. So, when they go and collect the nectar, contaminated pesticide residues, small droplet of pesticide residues, they are going to carry to their hives and deposit, and biomagnification, a little thing accumulating, and one fine day the whole colony become envenoming, poisoned. Sometimes worker bees, while collecting the nectar and pollen, there itself they get poisoned and they will die before coming to the colony. That is what bee poisoning. In front of the colony, so many worker bees are dying and dead bees you can record. Even in case of domesticated apicultural activity, beekeeping activity, there also beekeepers have come to know because of pesticide poisoning. In front of the bee box, so many hundreds of worker bees dying while carrying the pollen load. They carry pollen and deposit temporarily on the hind limbs of the colony, body. So, with carrying pollen load, they die. This is what is happening. This is what pesticide poisoning. Next. And we are burning colonies while harvesting. So, this is another one. Next. And this is what unscientific harvesting of honey, hunting. See, what the people usually do in the urban area, I said it. Now, honeybees have modified their nesting habitat and switched over to the human built structure. They are offering human built structure, where people by afraid, they are allow these particular people, these are all Bihari people, migrated from Bihar to Karnataka, and many people are staying in Mysore. They will be allowed to remove the colony, complete colony. See, in the name of complete colony, thinking that complete colony is filled with honey, complete colony is removed. As a result, developing brood, pollen, even the eggs, all the colony we are going to remove. As a result, so many honeybees are dying, so many colonies are, so colony number is declining in the human habitat buildings. Next. This is how you see the big sized colony, how big it is. So, more than 1.5, 2.5 feet to 3 feet big. Next. Next. And this is how month wise all the 3 species colony number is declining. It is because of human activity, human interference. Next. So next. And for stingless bees, burning, hunting, pesticide spray on the entrance of the nest, pouring chemicals into the nest, and mud or sealing of the nest entrance, mud or cow dung, cement, plastic cover, plastic bag, gunny bag. These are all the things man is doing against the wishes of stingless honeybees. Next. These are all the places. You see here the mud plastering, see plastic blocking. So like this cow dung pasting and demolishing the colony like this. These are all, and sticks inserting, because these are all creating a kind of nuisance to the people. So, but they are not stinging. We are removing all these colonies. They are not harmless. Next. And see demolishing, see the swarm, honeybee swarm. Next. As a result of that, very good number of colony in different districts like by Bangalore, Chikkaballapur, Kolar, we are next. So these are all the human activities: destruction, burning of nest, pesticide application, hunting, sealing, and all. Next. See the demolishing of residential houses and percent of colonies destroyed in different districts. Next. So we have suggested few suggestions to the people who are doing all these activities. Next. Coming to, this is one part, honeybees. Next is butterflies. So you know butterflies, they are beautifully colored, and of course they are pollinators. Of course all butterflies are not doing pollination service, but the larval stages, immature stage of butterflies is very important. They become prey to so many animals in the food chain, and they become a part of so many animals' food web. Next. And so far around the world 19,238 butterfly species identified, of which in India we have 1,504 butterfly species. Next. The butterflies belongs to these 5 groups, families. So like Nymphalidae, Lycanidae, Hesperidae, Pyridae, and Papilionidae, and these are all the species identified butterfly species. Next. And they have been enlisted because some are endangered, some are endemic in India under Wildlife Protection Act. Why I am showing all this? These are all the butterfly species living around us, one on the other day, one on the other season. They are living with us. Next. So they act as good indicators of environmental quality, because immediately if the temperature rises or humidity comes down, they are sensitive to recording or showing their behavior, attitude in the changing environment. Next. So we have selected Mysore district as a whole, and so many agricultural ecosystem and landscapes we have identified and recorded. Next. So and even in Chamarajanagar district also, this is forest area, green colored one, this is agricultural landscape we have selected to record what is happening with the butterfly fauna. Do they face any kind of human intervention? I am only talking about human intervention, no other factors I am not bringing here. Next. So these are all the species, see how beautiful they are. Hesperidae family members. Next. And Lycanidae. Next. Then Nymphalidae. Next. Then Papilionidae. Next. Then Pyridae. So beautifully colored butterflies we have around us. Next. So we have recorded in different agri-agricultural ecosystem and their density, abundance, and what is the status, common, very common, rare, endemic, like that we have recorded. Next. And these are all the protected species in Chamarajanagar district we have recorded, good number of species. Next. And even the migratory species also, because they do migration, undertake migration from one area to another area. In a huge swarm they migrate. So these are the species we have recorded in this area in Chamarajanagar and Mysore district. Next. This is what migrant butterflies: double banded crow, dark blue tiger, and common crow. Migrate to one place to another place in search of maybe finding suitable habitat, in search of finding suitable nest host plants. Remember, butterflies prefer flora for 2 to 3 important reasons. One is, some of the species do pollinate. While pollination, they collect nectar, they never collect pollen. Adult's life stage is very short. So, nectar, whereas larval stage, the life cycle 4-stage holometabolous insects. So, larval stage, they prefer food plants. They feed on plants, leaf and other pods, shoot and all. Okay, then that is second reason. Third reason is they prefer good host plants for depositing eggs, oviposition, egg laying, because soon after egg laying, egg has to hatch into larva. The larva, newly hatched larva, should get good, sufficient, nutritious food for its survival. No parents are going to look after newly hatched larva. It has to take on its own care about its survival. So, and another thing is, fourth reason is, butterflies prefer ideal breeding site, breeding ground for mating activity and reproductive activity. If all these things are not properly happen, if they are not getting suitable places or habitat, either in the natural ecosystem or in the man-made ecosystem or modified landscapes like parks, gardens, agricultural ecosystems, where butterflies never undergo for any kind of breeding activity, egg laying capacity is not going to happen, normal population you won't expect because of all these reasons. So therefore, here the flora is very important. Okay, next. And these are all endemic species: Papilio buddha, Arpala alea, Throsus estimata, Tridominus, like this. These are all the endemic species. Nowhere in the part of the world they are available, exclusively in particular region, endemic species. Next. These are all again one. Next. Then what are the anthropogenic activity problem? Next. Even there also, see, mowing, burning, vegetation removal, road kills. While migration, they will be come in touch with vehicles and got killed. That is what road kills, and considerable number of butterflies are dying while migration. Pesticide application, then all these activities at different habitats like aquatic habitat, park, residential area, institutional campuses, road verge, nursery, cultivated land, vegetation, weedy vegetation. These are all the places you might have seen. You are come in touch with all these places in different ecosystem. Even in Udupi also all these places are there, but we least bother about all these areas, what is happening. So, in and around us, so many different habitats are there, unique habitats are there, where so many things are going on, so many good species are available. So, what are they surviving properly, normally? Are they facing any kind of human interference? So, that all we are least bothering. So, this is what in these areas butterflies are facing lot of disturbance. Next. So, different insect species, butterfly species belongs to different families are facing lot of problem. Next. So, next, this is what butterflies, they are also not safe. So, coming to other pollinators other than butterflies and honeybees. So, other insects pollinators which belongs to so many orders. Next, Scolioptera, beetles and weevils, dipterans, flies, hemipterans, bugs commonly, then hymenopterans other than butterflies, Formicidae members, wasps, Vespidae, then Lepidopterans and Odonids. These are all we have recorded during our observation. Next, by taking Kodagu as one of the model district, because Kodagu comes under Western Ghat, and where all these family members we have recorded. Next. Of course, their relative abundance, percent occurrence, pollinators density, that all we have recorded. These are all in the terms of data, but don't worry, I will show you images next. See, the hemipterans like the insect, then Odonata, then Lepidopteran, particularly Eraceous species. These are all, their population is decreasing. Next. See, you see the moths, pollination, nocturnal pollination. Next. During night time, very good number of plant species are pollinated by butterflies, moths. Next. This is how. Next. See, larval pollination. Even larvae is also contributing considerable amount of pollination service. Next. And small bees, not apis or not stingless bees, other bees. Next. Beetles, they are doing pollination service, but we have not estimated properly. We have recorded in Kodagu district. Next. And these are all the coleopteran species going to pollinate all these plant species, their common name, scientific name. Next. And even dipterans, flies commonly. Next. Bugs, hemipterans, heteropterans, like that. Next. Of course, this I have already made a mention. Next. Wasps. Next. Bumblebees. Next. Ants. These are all consistent foragers. They collect pollen as well as nectar. Next. See here, they collect good amount of pollen and do pollination service. Their presence is very important for particularly this particular species for their survival. Next. See, why we need to know about pollinators in general? Because if the crop is pollinated by pollinators like insects, you can get uniform seed set, and seed size would be good. Then shape of the fruit is good. Then yield is 1.5 times enhanced more. There are so many reports. Agricultural university people in different agricultural universities around the India and other parts of the world also, they have recorded. Then the yield is, you see, particularly oil yielding plants, 27% of the oil yield is increased from the normal pollination done by man-made activity or any other way. So as a result, take for example our own onion crop and garlic, where 50 to 70% higher yield has been recorded due to because of bees and pollinators pollination. Next. So their diversity is like this, abundance year by year it is decreasing, 2020 to 21, 21 to 22, 23 to, it is because of human interference. What are those human interference? Next. We have recorded the number of flora in a different habitats. The number is decreasing, and accordingly their availability and percent occurrence we have designated as very good like that. Next. And accordingly we have recorded nectar and pollen plants. Their percent occurrence also revealed considerable variation. Next. Very quite interestingly, surprisingly, in 20 to 21 started we working, 21 to 22 no much decline recorded, but 22 to 23 and onwards 2.8% of flora and as well as insect species, pollinators declining has been reported in Kodagu district. Kodagu is supposed to be a Western Ghat, but we have not recorded touched forest ecosystem, we have recorded in agricultural ecosystem. So even Kodagu like area also where pollinators are declining. Yesterday Dr. Arvind made a mention about that is renting of honeybee colonies for doing pollination service in Western countries like United States of America. Now not only America, around Western countries, around the globe, people are crying about declining of pollinators. It is because of mainly human activity. Of course, climate change, global warming, I am not touching. Those are all the other aspects, but human intervention is maximally impacting on the pollinators' decline. This is what pollinators. Next. Now, another interesting thing, aquatic insects. Of course, next. So far whatever I just dealt, it is on exclusively on terrestrial ecosystem. Now coming to aquatic ecosystem, even in aquatic ecosystem also, where man is interfering a lot and he is creating lot of disturbance. So aquatic insects are very important, playing very important role in the ecology of freshwater ecosystem preferably, and they are vital for processing organic matter, transporting energy along stream channels, lateral to the flood plains, food webs, even vertically down into the stream bed. So they are playing a very important role, and whatever the leaf litter and other things are falling in the forest ecosystem, our majority of the rivers are originated from the forest ecosystem, that too from Western Ghats. So many rivers are originated from Western Ghats, where a lot of forest is there, and forest litter, that all ultimately going to during rainfall through rain water flow getting into the river courses. So all these leaf litter, decaying litter gets converted into nutrient rich energy, carbon and other things to the river bank. That is taken up by some other detrivorous feeders, some other organisms which are living in the water. So this is how it is unimaginable what a type of the contribution the aquatic insects do in the river ecosystem. So hence in that sense the aquatic insects are very important for converting litter waste, leaf waste, forest waste into energy rich carbon containing molecules to the various group of animals. Next. So this is what we have taken our study area, Sakleshpur. It is almost in the midst of Western Ghats. Beautiful place, Sakleshpur. It is 38 to 40 kilometers away from Sakleshpura. So it is nearby Kadumane. I think some of you might be knowing about Kadumane. It is untouched area, and very difficult to go to this particular place by without taking permission. You will not be allowed to go there. Of course, we are working with Vishweshwaraya Jala Nigama Limited, Indian Institute of Science undertaken project. So we have been allowed to go to this particular place and do sampling activity. Next. And these are all the different places we have selected. Of course, latitude and other things are there. Next. And this is what the area, project area. Ettinavale, I think project, you might have heard about Ettinavale project. It is still going on. And here this Ettinavale is one of the river, and so many tributaries are Kempuvale, Karivale, like that. So many river basins, joining to this Ettinavale, and ultimately this Ettinavale water is reaching to the Netravati river ultimately. So this is the area, and these are all the sampling sites, river basin we have collected. Next. See, this is how check dams have been constructed to the different tributaries of Ettinavale river. The purpose is simply, I am just telling, I am not going in detail. So purpose is to collect water, because during rainy season all these river basins are overflow. Ultimately they go to ocean, sea. So to store all those water and collect and lift to different areas like drought prone areas like Kolar, Chikballapur, Tumkur and Haveri district, where water is one of the scarce commodity. So to provide drinking water supply, this particular project is initiated. It is still going on. Okay. So this is how check dams have been collected. So to establish this type of check dams, lot of forest has been degraded. Next. So you see here, these are all the places, and you see the river basin where we have collected aquatic insects particularly, because aquatic insects indicate the quality of the river, number one, and quality of the, amount of degradation happened in the river basin of all these rivers. Okay, next. So, insect facing threat due to man-made activities. Next. See, the aquatic ecosystems are preferred for so many reasons, preferred breeding grounds for so many insects. Take for example, some of the lepidopterans are living inside the water. Some of the dipterans prefers benthic region, river bed for breeding activity. Some of the aquatic, insects like odonates, mayflies, epimeropterans, dragonflies, damselflies prefers submerged vegetation for conducting breeding activity. Their immature stage, naiad, what we call, not larva, naiad stage is living in the submerged water. They are predaceous in nature. They feed on planktons. Take for example beetles, coleopterans, their larval stage prefers benthic region. So, whatever deteriorates, degrading food and other materials are there, they go and eat. In this way, they are playing a major role for controlling the what we call pest population and also some of the harmful insect population in the aquatic ecosystem. So, then the host plants undertaken for metamorphosis, pebbled river is required for dipterans and coleopterans like that. Next. So, these are all the insect group we have recorded in during dry season and wet season. Next. And a lot of differences we have recorded. Next. This is what the difference, population size difference during different season. Next. So, the percent occurrence of all these indicates there is a group specificity with respect to the distribution of different group of insects in different area, river bed. Take a river as a whole, water column what we call upper surface, middle surface, benthic region bottom. In all the surface area where different group of insects are distributed. Next. So, usually the check dams have been constructed in the upstream. You see the population size, density, abundance of aquatic insects, it is more, whereas in downstream it is affected. You see here drastic decline. So, means to say establishment of check dam altered the population size of aquatic insects, belongs to different groups. Next. So, like this we have calculated the adult and subadults juvenile proportion ratio. Does the check dam activity, human man-made activity interfering with the life cycle breeding activity of the aquatic insects? So, this is what the proportion we have recorded, adults and subadults in upper stream, downstream, like that. Next. And these are all the different species of epimeropterans facing threat, unable to metamorphosis into adult. In the larval stage itself, so many number of all these species are dying because of, I will show the images. Next. Damselflies, larval stage, naiads, these are all predaceous insects. They feed on mosquito larvae, control the mosquito population, harmful mosquitoes like Anopheles, Aedes, so on and so forth. Next. See dragonflies, these are all one. Next. And coleopterans, larval form, immature stage of larva. Next. See, this is what diving beetle going bottom and collect detrivorous, detritiform food and feed and make the river bed clean like that. Next. And hemipterans, bugs, water skates, water pond, like that. Geridae family members, back swimmer, water scorpion, water striders. These are all predaceous insects. Maintain the quality of the water. Their presence indicates the quality of the water. Water is not polluted. So many aquatic insects we consider as indicators of quality of water. Next. Placophterans, they prefers to survive in the pebbled water where water is cool and fresh, there is no pollution. Next. And dipterans, trichopterans recorded. Unfortunate thing is most of the study site area, because of check dam construction, lot of human interference was recorded. As a result, pollution occurred. So, we have recorded Chironomus larva chironomids. Chironomous larva presence is indicating the pollution, water is polluted. So, the Ethinole river is flowing in the midst of Western Ghats where no human interference. But now, most of the study area on the river bed, Chironomous larval population is increasing. It is because of human activity, it is because of pollution. Next. So, this is what environmental degradation, you can see. I have just brought only few images because some of the images I should not show here. That is what. Next, because the project is going on, I should not reveal all the images. Some. This is what the base of the river, normal, pebbled bed, riverine bed or bottom like this. You can see the vegetation. Next. See what has happened. Completely changed, water course is completely changed. See the construction, see the digging of basin of the river basin. Next, see sand bed totally modified, completely removed. See big machines are used for construction of check dams. Next, see because of this siltation would occur because of landsliding. On either side of the check dam, big forest area is covered, cleared and established big check dams. As a result, erosion of soil would occur. As a result, siltation occurs. So, larval stages, so many insect larval forms are still, they are dying due to because of this particular activity. Next, see deposition of sand on the submerged vegetation. So many insects unable to complete their life cycle. Next, this is what is happening. Next, see the forest cover. See how they are layer by layer they are removing. To install around 10 to 12 feet diameter sized pipes, 10 to 12 meter diameter sized pipes, big pipes installation in the midst of forest to collect the water and pump to longer area, that is drought prone areas. Next. Next. So, next. So, these are all epimaplies, Placopterans stone flies and Trichopterans caddis flies commonly called. These are all indicators of fresh water quality, river pollution. So, their presence is declining nowadays, their population size is declining. That indicates the Ethinole river is getting polluted. Next. Of course, all these groups, odonates, hemipterans, coleopterans, getting adversely affected by human activity. Next. Next. Same thing. Next. So, go back. So, we have suggested site specific mitigation measures, what to take and what not to take. Of course, this is another part of our work. So, how to overcome this particular problem, but it is not one by telling this or by undertaking 1 year work, you cannot repair the damage what we have caused due to because of this water lifting project activity. It is one example case study in Karnataka. Likewise, throughout India, so many rivers are getting like this. Next. Next. So, aquatic insects, their feeding propensities, growth rate, life cycle, reproductive potential, these are all correlated with so many prevailed biotic and abiotic factors. So, therefore, understanding about all these is very important nowadays, because we need development, we need water. Of course, everywhere now people are crying for water, especially drought prone areas where they are getting once in a week water, very difficult. Of course, this type of activities are required, but at what cost? Question mark, we have to think. Next. So ultimately, unpleasant human man-made activities or man-made activities interfering with the nature, interfering with the so many species of insects, terrestrial, aquatic, amphibious insects. So we are responsible for bringing down their population size. It is because of our welfare. So therefore, on this line we have to think and we have to act accordingly what best we can do. So my intention is to show all these problems, issues, particularly for young friends like graduates, because in future course they are going to study for MSc and they may get into research field. So where this kind of burning issues, if they attend, so this kind of seminars purpose is served. Next. So go back. Coleopterans are major, dipterans are scavengers in the benthic region, predators, plecopterans and trichopterans are indicators of water quality. Next, this is what biodiversity means to say. You know very well, everything is inclusive here, not only mammals, not only vertebrates, invertebrates also. Next. So the laws for biodiversity, it is there in website, you can get it. Okay, it is not just by saying one few measures you cannot control the biodiversity loss. A lot of things should be done. Next. So ultimately, biodiversity or wildlife to be valued, we should respect. That's what in the wildlife studies there is a separate course, wildlife values, aesthetic value, cultural value, economical value, ecological value, like that. We should know about the value of wildlife and biodiversity. Conserved, restored, wisely used, maintaining ecosystem service, sustaining a healthy planet, delivering benefits essential for all the. So we should not bother about only our generation. We should think about future generation. What best we can give to next generation by saving environment, by saving or conserving biodiversity and wildlife. So an environmentally sustainable society satisfies the basic needs of its people without depleting or degrading its natural resources. This is what we are supposed to do. Society, sustainable society, and its natural resource, and thereby preventing current and future generations of human from meeting their basic needs. Next. Thank you. Next. I am very much thankful to the organizers, preferably my friend Professor Ishwar Bhatt, principal and organizers of this conference, this institute, for the opportunity to speak a few aspects about what we have done during 15 to 18 years of research work. And I am also thankful to my university. All the time university is encouraging me, and my research students. It is not alone myself done. So many research scholars, we have contributed their work, preferably all these, and even MSc students project. So thank you very much for your patience hearing. If you have any questions, please.
So congratulations for a very useful, thought-provoking preparation and PowerPoint presentation. Here, as I represent the Faculty of Agriculture,
Yes, sir.
I have got one important question. You see, particularly during flowering season in the arcanut or paddy, food crops or the commercial crops, if you do not spray chlorpyrifos or the pesticides or insecticides, there will be a flower dropping. It is an all India phenomenon. If we spray, it will affect the bee population.
Yes.
So we are much concerned. In the last month in ICR, that is Indian Council for Agricultural Research, there was a meeting where they said 60% now we are the food security is endangered in India.
Yes.
If the food security is not there, the country will plunge into international food politics. But this is how best we can prevent this. Is there any time frame where we can avoid the mortality of bees, spraying is inevitable.
Yeah, of course.
Because food security, we know organic farming, everything is there, but we have crossed all the limits. Food security is very important. But particularly in Malnad region, Western Ghat region, during the flowering season, the flower falling, the inflorescence dieback, we call it. But if you spray, the bees will be annihilated. So what best method we can, like mosquito repellent be there, mosquito be there, but it should not affect us. Do you have any method? It will be helpful, and I request you to bring an extension material for the benefit of farmers. This is very good, because particularly awareness of bee contribution or butterfly contribution is almost nil among the farming community.
Really, you are right.
That it is very much useful. Thank you for this.
Thank you.
Thank you.
Sir, that's what I am addressing this kind of issues. See, there are so many alternative methods, sir. We can go for plant-based pesticides, phytochemicals. We can use one option. Second option, we should study about the biology, behavior of, that is foraging behavior of honeybees, preferably honeybees. So usually they are very active during morning hours. That is around 7 as the sun rises, 7 to 10:30 up to 11 o'clock. They are very active, hyperactivity you can see. So during that particular period, instead of using sprays, you can go for solid crystals like or powderable this one. So instead of spraying, you should apply to the bottom of the plant, flowering plant, with that particular smell, so many insects can avoid to visit, this one, the falling of that particular thing. Through the root zone, you can provide some like fertilizers, so you can provide like that. So means to say, like that alternative measures we should find out, sir. So where research, lot of research has to be carried out. So I am sorry, I don't know much about as per my knowledge goes. So in that field we are lagging behind about developing new techniques, how to avoid contamination during spray. That's what. So best way is to avoid during foraging activity. So we can spray the pesticide, whatever pesticides and other things to avoid the flower fall during afternoon hours, evening hours, night hours we can do. So that's what we can. Thank you, sir.
Hello, sir. Myself Muskan Kumari from St. Joseph Vaz College. Sir, our college is planning to take up mangrove honey project as it is closer to the mangrove hub. So, can you tell me the management and maintenance of the honey bees over there?
In mangrove vegetation?
Yeah.
Mangrove vegetation. Yeah, you can go for beekeeping activity, Apis species, that is Sirana and Apis mellifera you can use. First of all, you should study about the mangrove area where you wanted to establish apiculture activity, floral source, flowering season, nectar and pollen availability. That's what I have shown. Floral calendar, pollen calendar. You can collect it from in consultation with the Department of Apiculture, Government of Karnataka. They will guide you what to do and what not to do and how to initiate the beekeeping activity in such areas, new areas. You can get subsidy also. Okay, you can get this one instructions and advice and technical support also. So thereby you can start doing it. And of course, once you install the bee boxes, then naturally you will going to learn, and the people from Apiculture Department, Government of Karnataka, they will guide you.
Thank you, sir.
Okay.
Sir, good morning. I like the point about a river and the developmental activities and the connection of losing the insects. Now, as you have said, sir, development is required, and for development, environmental degradation is necessary. But the way the loss of insects and other fauna, especially which is not taken note of by activists, there is a lot of activism to save environment, but this point, especially about losing the insects, is not raised. And I feel the loss of insects is one of the great point that is to be considered, because that can lead to loss of humanity itself if we don't give enough attention to it. So what I want to ask, can this be a part of policy formation for government during the developmental projects? And now that the national elections are also coming up, development is going to be surely the plank for elections. So when they form their manifestos, can the insects make some entry into their manifestos?
See, whatever developmental projects are initiated are going to be undertaken. The concerned state and central governments are going to suggest, after completion of the project, what is the duration you require, how much you are going to deteriorate the local habitat, then how are you going to restore the lost biodiversity or wildlife or insect in general. That all they will suggest. That's what I said, site specific mitigation measures one has to undertake and one has to follow. Okay. So by doing so we can restore the insect population in that particular area. Okay. So lost insects can be rejuvenated by re-establishing the original habitat as was there earlier before starting the activity. So that is how one can bring back the original thing. I think, am I correct? Your question?
Yes, sir. The study that has been made, now these type of studies have to go to the people, these type of studies have to reach
That's what I wanted to share, I wanted to present here in this type of ambience, because youngsters are there, you people like youngsters, definitely you can mind what is happening, what is going on. So there are a lot of protest was happened before initiation of the Ethinole project, but despite all those protests, it is going on. It is almost 70% work is over, remaining 30% work is there, but no one can stop. Okay.
Yes, sir. That's why I'm saying the developmental activities will not stop. But if insects make the entry into their, when the projects are formed, how to rejuvenate, there is no provision made for rejuvenation of what is being lost.
No. It is there. Usually, that's what I said. Any type of this type of mega projects initiated, government is strictly insisting. Department of Forest and Wildlife strictly insisting. That's what we are on the almost fag end of the project, and even site specific mitigations measures during submission of the project report, they are asking strictly. What all the mitigation measures you are suggesting to bring back the lost fauna, flora going on. But it is a general trend in India, whatever we undertake major projects, we only worrying about our welfare. Remaining thing I don't want to tell here, everybody knows we are in India.
Yes, sir.
What is happening with the many projects. See, road widening is happening, but after damper or establishment, whatever on either side of the road, simply they forget. Lot of waste they simply they dump. Clear? No one is going to take care. This is what is happening with every project. So we just only worrying about completion of our work. That's all. So remaining repairing things we least bother.
Thank you for the insights, sir. It was a wonderful sharing from you.
Thank you. Thanks.
So one more question.
Yeah, madam.
Nice informative talk, sir.
Thank you.
Sir, you have mentioned everything like anthropogenic causes and all. From like past 1 year, I'm working on varroa mites, which infects the honeybee.
Yes, madam. See,
so whether it infects the wild
colonies.
Colonies also. So what is the measures
Madam,
you are taking?
varroa mite problem is not much there in wild honeybees, particularly in Karnataka. We have not recorded varroa mite, but one more mite is there, porotic mite. This is porotic mite we have recorded very first on Apis dorsata. That mite is now deposited in the accession number so and so in Columbia University. I have sent that insect very first we have reported from our laboratory. Porotic mite, it is a flower mite. That flower mite is infesting to Apis dorsata and interfering with the Apis dorsata colony, and the colony becomes so aggressive. Honeybees become so aggressive because of infestation of that porotic mite. But varroa mite problem is not much there with wild honeybees. We have not come across. Thank you.
Sir, hello sir. Sir.
Yeah.
Okay sir, different types of honeycomb ಬಗ್ಗೆ ಮತ್ತೆ ಹನಿ ಬಗ್ಗೆ ಹೇಳಿದ್ರಿ. ಸೊ ಅದರದ್ದು ಲೈಫ್ ಸ್ಪ್ಯಾನ್ ಆಗಿರ್ಬೋದು, ಅದರ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್ ಸೇಮ್ ಇದೆಯಾ, ಸ್ವಲ್ಪ ಡಿಫ್ರೆಂಟ್ ಇದೆಯಾ, ಮತ್ತು ಅದೊಂದು ಹನಿಕೋಮ್ ಇದ್ರೆ ಎಷ್ಟು ವರ್ಷ ಬದುಕ್ಬೋದು, ಡಿಫ್ರೆಂಟ್ ಹೇಳಿದ್ರಲ್ವಾ?
Apis dorsata, wild honeybee, commonly called rock bee, then giant honeybee, if it is undisturbed in the natural condition, the colony can survive years together. Of course, it is a migratory species. It migrates. When floral source is declining, so it migrates from one place to another place. Apis dorsata, wild honeybee. Similarly, Apis florea, also migratory species, but it is not a good flyer. It is small bee. It can migrate to longer, smaller distance. Okay, it can survive for about 6 months to 9 months, maximum 10 months. Whereas stingless bees, I said, stingless bees can survive 4 to 5 years in a same area, same colony, unless until if you abandon, it won't die. Colony will be keep on establishing and becoming more and more bigger in size, because queen is flightless in stingless bee. When she lost after mating behavior, she will come down and she starts establishing the colony, and she will be inside the colony. She cannot come out because of fatty abdomen. That is what we call a physiological modification, physogastricity, enlarged abnormal enlargement of the abdomen. As a result, stingless bees, only small entrance will be there, coming out, only few bees coming and getting in. So the queen unable to come out, she will be remain. So people are just blocking because of problem, human interference blocking. So there itself colony will die. So it is a perennial colony. 4 to 5 years will be surviving. So queen will survive 4 to 5 years. After queen, when she loses her egg laying capacity, new queen will be emerged. That's how different species going on. Of course, Apis cerana and Apis mellifera, it is different thing. You may be knowing colony division, multiplication, people are doing it. So colony multiplication means more number of colonies. More number of colonies means more quantity of honey can be produced. This is what is apiculture people are doing. That is different one. So I am focusing only on the wild species where very little research work is going on, and most of the people are very much worried about domesticated honeybees. A lot of research work has been carried out.
Good morning, sir. A very wonderful session it was. And my question is also on stingless bee. So I have a doubt whether they can survive in the disturbed area, most disturbed area.
Stingless bees?
Yes.
More disturbed area, no madam, cannot.
Because I have a practical problem.
See, I am talking about wild colonies. Okay. Meliponiculture is different. Meliponiculture, stingless honeybees rearing is called meliponiculture. So many Keralites, Kerala people, even in so many Western Ghats area, even in plain area also, people are doing meliponiculture. That is captive stingless bee keeping activity.
Okay.
But you should not disturb.
Okay.
Under human captive condition, stingless honeybees you can rear.
But I am not talking about captive breeding. There is one colony in the car, in my car, in between the door, and it was almost from last 6 to 8 months. Even though, as you said, blocking of the entrance, anything, it will be remain as it is.
Sometimes so, if they get blocking, all these interference is there, if they get good, what we call less predatory interference, like some of the wasps will come and feed on the stingless honeybees, pollen carrying honeybees. They hunt, they predate. And sometimes if flora is good around the colony, water availability is good, and propolis, that is plant resin exudate available, they will not go even if there is a disturbance.
Okay, thank you, sir.
Good morning, sir. I have a question regarding bees. As you said, the extraction of honey is carelessly handled. The whole hive is extracted. Are there people who know how where actually the honey is located and only extract that part, and how people can know about this?
Yes, yes. See, you just go to YouTubes. There are so many YouTubes where how honeybee, wild honeybee colonies are hunting, how honey is extracted, that all you can get. Of course, in Karnataka, especially the government of Karnataka, Department of Apiculture, taking lot of care. Even in agricultural university also, some of the apicultural departments are there. They are giving a training to how to harvest honey from the wild colonies. They give training. Okay, and there are so many new methods are there. So only, even in beekeeping activity also, this particular technique or practices are followed. So only you should know about the bee biology before honey hunting. Okay, where the colony usually on the top portion, it is bulged. It is coming like this. It is a vertical sheet, bilayered vertical sheet with hexagonal shaped cells. So top of the comb on one side, it is bulged. That portion itself is, you can consider it as a honey stored part. No other part is filled with honey, remember, only top portion, because top portion is attached to this substratum. So it can bear the weight. Then gradually it is coming thickness down. Okay, so that portion only where you can use certain devices, so smokers, to smoke, spray smoke on that particular area and keep away the honeybees, and see even honey hunting is again scientifically you should know. Two types of honey you can see, matured honey, immatured honey. So matured honey means, see, when nectar is collected by the honeybees, the nectar contains 80% of moisture, only 20% of sugar solution, fructose and other things. So during the process of honey making, when it is coming into the honeybee colony, while carrying, honeybees will carry the nectar and deposit in the chamber, honey storing pots. So there are some bees, scout bees what we call, in hive bees what we call, they will do fanning, wings beating activity, thereby increasing the microclimate of the colony. It is cryptic activity, one cannot see openly. As a result, continuously they do, moisture content is evaporated and comes down to 80% to 20%, 18 to 20%. So during that time, what they do, honeybees completely fill the hexagonal shape, then cap. So if the open hexagonal shaped cell is capped, that honey is matured honey. If you go for harvesting such type of comb, honeybees will not show aggressivity. They will not become aggressive. They allow you to touch. If it is not sealed, they become aggressive if you go and touch. So even use smokers, so aggressiveness will be there. So by seeing the biology, by seeing the size of the comb, by seeing the capping of the colony, that is cell, you can go for honey harvest. These are all the things during training people will guide you. So thereby only any part you can remove, remaining rest of the part you can retain. So queen will be there somewhere. So you can restore the whole individuals. That's how scientifically one can harvest.
Thank you, sir. That was indeed a thought-provoking session. As we conclude the third technical session, it is my honor to extend my heartfelt gratitude to Dr. S. Basavarajappa and all of you, our wonderful audience, for your active participation. I request Dr. M. Vishwanath Pai sir, HOD of Computer Science, to present a memento to our guest speaker as a token of gratitude and appreciation. Thank you, sir. Here we conclude the session. Thank you, one and all. Please note, the next session will begin within 10 minutes after the tree break.
ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
The skeleton which you see, the skeleton of Balaenoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales. It is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balaenoptera. It has a special reference to the enormous big feeding device called as baggins. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these baggins help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಿಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see, the skeleton of Balaenoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balaenoptera. It has a special reference to the enormous big feeding device called as bags. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these balance help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿತಿಮಿಂಗಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balaenoptera, called as the giant of the sea. This was located on the entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balaenoptera. It has a special reference to the enormous big feeding device called as bags. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these balance help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balanoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balanoptera. It has a special reference to the enormous big feeding device called as balance. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these balance help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಿಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ನಮಸ್ಕಾರ, ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balanoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be
Dr. Vignesh, a highly accomplished individual in their field of expertise with an impressive track record and numerous accolades to their name, Dr. M.S. Vignesh has made significant contribution to sociology. Shri Manchale Suryanarayan Rao Vignesh was born in the year 1954 on 2nd of March. He did his graduation from Lal Bahadur College, Sagara, majoring the subject Economics, Sociology and Philosophy. He completed his post-graduation majoring Sociology at Manasa Gangotri, Mysuru. He obtained his doctorate from Shivaji University, Kolhapur, Maharashtra. Our beloved guest, Dr. M.S. Vignesh, has served in the field of education for more than 37 years, beginning from the year 1979 to 1981 when he was first appointed as lecturer in Vijaya College, Mulki. He was a lecturer and department head of sociology at Christ College, Bengaluru. He was associate professor and head of the Department of Agricultural Extension and Rural Sociology at University of Agricultural Sciences, Shimoga, and later became professor and head of the Department of Agricultural Extension and Rural Sociology till the year 2016. Besides teaching, Dr. M.S. Vignesh have had years of administrative experience. He had been appointed as a Dean of Student Welfare at Keladi Shivappanayaka University of Agricultural and Horticultural Sciences, KSUAHS, Shimoga. He had served as member of Board of Studies and Academic Council for more than a decade in the University of Agricultural Sciences, Bengaluru and KSUAHS, Shimoga. Furthermore, he published more than 10 books and 50 research papers in referee journals. One of the most popular books is Counseling Students, published by Westville Publishers, Delhi. He is one of the founders of Arakanat Research Center, KSUAHS. The awards and credentials designated to Dr. M.S. Vignesh include FAC, Fellow of the Academic Council, Institute of Documentation and European Studies, International Center for Academic Research, Belgium in 1985. He has been recognized as source person by Virginia Tech University, Fellow of the Family Planning Association of India, Bombay in 1988. He received award of Distinguished Counselor by Himachal Pradesh Agricultural University, Palampur. He has been the member of International Peace and Collaborative Organization, USA in 2009. He received Dr. APJ Abdul Kalam National Award in the year 2019. Today, we are fortunate to have Dr. M.S. Vignesh grace us with their presence. Their profound knowledge in institutions and traditions in maintaining biodiversity and expertise in sociology have garnered admiration, respect from peers and professionals alike. I take this moment to heartily welcome you, sir. Their insights and perspective will undoubtedly enrich our mastery. Now, I request Ishwar Bhatt, sir, to florally welcome Dr. M.S. Vignesh, sir. We are all eagerly waiting for your knowledgeable words. Over to you, sir.
The honorable delegates, I sincerely thank you for having given me this opportunity. During our childhood days as a students, many of us used to dream of getting admission to MGM college. At that time, during 1970s and 80s, every disciplined student always thinking of getting admitted to MGM college for his PUC. And I tried, but I failed. During 1974, having completed my 10th standard, at that time my maternal uncle was working as a speech pathologist in Manipal Hospital, he asked me, come let us, I will be admitting you to MGM college. At that time, Professor Kushi Haridas Bhatt, a well-known scholar in Karnataka at that time, was the principal. And I tried for 4 days, but I could not get. Now I feel happy to be on this dais. Then later I joined DVS college and pursued education. So such a prestigious college it was. Such is. And then particularly for the distinguished scholars, it is my pleasure to give you a brief introduction about one of the well-known agriculture scientists of India, Professor Nayak, Dr. M.K. Nayak. He is with us. I am really happy to introduce him. He is one of the renowned scientists working towards the prosperity of farming community, and he became the Vice Chancellor, Honorable Vice Chancellor of Keladai Shivopanayaka University of Agriculture and Horticulture Sciences, Shivamogga, a newly established university, an offshoot of GKVK, US Bangalore. And the second Vice Chancellor, he started developing at the Iruvakki campus. A beautiful campus came into existence there in the Malnad region, stretching over more than 700 years. So I requested him, if the opportunity comes, let's just briefly address the delegates here. The major students are here. Particularly an integration between traditional university and agriculture university would be better. So with this, again thanking you for giving this opportunity. Let me begin my study. My topic is Indian institutions and traditions supporting biodiversity. See, biodiversity is the very essence of Indian culture and civilization. Now let me briefly explain. Most of the thinkers have developed so many systems of administration in the world, like communism, socialism, democracy, monarchy, so many systems, capitalism, so many systems, like Karl Marx, Max Weber, Lenin, Mao, so many. All of them defined what is a country. Country is a defined geographical area wherein human beings exist. They never imagined the coexistence of animals and other charachara jivigalu, we call it. They never imagined. And they became the stalwarts of the Manu Kendra Chintane, the human centric thoughts, theories. Everything is for the welfare of human beings. And that philosophy, ideological values, created all such problems nowadays we are experiencing. But Indians long back defined what is country. A country is a definite geographical area. They call Aryavarta, Khanda, where apart from human beings, all other creatures have a right to live. That is called animism. Nowadays, what we call the so much stalwarts, now Karl Marx is venerated, Mao is venerated, Lenin is venerated, Max Weber, Lasky, so many thinkers, but they never gone beyond the realm of human beings, human centric. They never idealized the nature. Nature is for human welfare. So socialism, it may be communism, they never recognized. What we are experiencing now, the wildlife is entering into the cities, entering into the human habitat. It is nothing but a sort of protest against the exploitation. So that is why I decided how best we can revive the Indian traditions. Now we have got sacred groves called Pavitravana. In the Asia we have got largest Pavitravanas. During 2002, the Forest Act was amended to give a legal status to those forests, protecting the animals there, the species. So here now, in course of time, the great Indians have developed functionally related practices in harmony with nature, flora and fauna. In fact, the edifice of Hindu rituals center around nature, mountains and rivers. It is basically animism in nature. Animism means worshipping the nature, objects of nature, protecting them, treating them as equally important to that of human beings. Next. Animism is the belief that objects of nature, places and creatures all possesses a distinct spiritual essence. Animism perceives all things animals, plant, rocks, rivers, weather, systems, human handiwork and in some cases words as being animated. So this is next. Generally, 3 levels of biodiversity is recognized. Each of them is functionally interrelated. They are diversity of species, number of different species represented in a given community or society. Genetic diversity, diversity among plant and animal population. It is the variation in genes and genotypes within the species. It is one of the driving forces of adaptation to the changing situations. Next. Now, the important ecological diversity deals with the variations in ecosystem within a given geographical system. Ecological diversity is the largest extent of biodiversity. Now that is endangered. Within each ecosystem, there is a great deal of both species and genetic diversity. Next. Now, India enjoys large number of sacred groves, and that is in every state. In fact, Maharashtra is the first. It has got more than 6,000 sacred groves, followed by Kerala. In spite of population pressure, the sacred groves are protected there. Both animals and plants are protected, well protected. And the third, for Karnataka state, we have got a sacred groves. So here, extent of forest which are preserved and regenerated. A sacred grove, also called sacred forest company, it is practically nowadays it is called Pavitravana, or we have got Devarakadu, like that it is designated, and it is unique to the Asian countries. And some of the African countries have it, not to this extent. It is deeply historically penetrated. Whether from a few trees to several acres, the spaces of these sacred groves are protected by local communities because of their religious beliefs and traditional rituals that run through several generations. During 2002, a Forest Act was amended to include that as a protected forest, as a deemed forest or protected forest. Next. Sacred groves help in the protection of a number of rare, endangered and endemic species. Sacred groves are important repositories of flora and fauna diversity that have been conserved by local communities in a sustainable manner. Let me give an example. You know mango pickles, main midi anta heltaralla, that is very popular. That is usually a very rare. In Malnad region, particularly in Sagar, Swarab, Tirthalya region, once we used to enjoy a large number of trees of mango pickles bearing trees. Now it is endangered. If at all we see that, only in the sacred groves we see now. Very delicious traditional trees, that is variety, mango, appe midi anta karitave, kelirbodu neevu, appe midi, and that is very popular. That is. Now it is on the verge of extinction. But in the sacred groves, that you can see in plenty. That is. Then, sacred groves are important repositories of flora and fauna diversity that have been conserved by local communities in a sustainable manner. Next. Now, importance of sacred groves. ಈ ಪವಿತ್ರ ವನದ ಪ್ರಾಮುಖ್ಯತೆ ಏನು? The vegetation cover of the sacred groves improves the soil stability of the area and also prevents soil erosion. The sacred groves are often associated with ponds, streams or springs which support the water requirements of the local people. Thus, the vegetative cover of sacred groves helps in recharging the groundwater. That is, rainwater harvesting is also very well managed there. Next. Now, almost all states enjoy such traditional sacred groves, also called Pavitravana. Most of them is well protected with its versatile biodiversity. It is estimated that there are more than 3,000 such sacred groves in India. Highest number of such groves are protected in Maharashtra, 1,600, followed by Kerala, 1,500. In Kerala, that is wonder. In spite of that population pressure, where the encroachment of land is going, but these sacred groves are protected. And Karnataka, 1,476. There are more than 8,080 such sacred groves in India. Almost all states, starting from Himachal Pradesh, Assam, Meghalaya, northeastern hill states, almost all states enjoy these sacred groves. This is a very important inbuilt institutional mechanism to protect biodiversity. It is an inbuilt mechanism, and that is why it is called basically animism. Next. Legal status of sacred groves in India. People believe that any kind of disturbance will offend the local deity, causing diseases, natural calamities or failure of crops. Now, I fully agree with our previous professor Basuraj for a very enlightened talk on apiculture. But nowadays, these sacred groves are protecting these beehives throughout India. If at all beehives, we have that one because of these sacred groves, where people do not destroy them. Even the wild animals are also protected out of fear of that deity. Now, lot of honeycombs you can see, honey beehives you can see in these sacred groves. Particularly if you happen to see in Himalayan region, Himachal Pradesh, plenty of beehives you can see there in the forest. All those are protected forests. Now, the institutions of sacred groves have been legally protected under community reserves in the Wildlife Protection Amendment Act 2002. Why I am specifically stressing this point? 2002, in the IAS preliminary examination, this question was asked year before last year, and one of my students asked me, then I referred that one. It is 2002 Amendment Act. There is on different topics in preliminary examination they ask, and this was, and majority didn't answer. That is why I specifically included this one. Next. Now, threats to these sacred groves. Nowadays, there is a threat. So, with a sense of extension orientation, I have included this one. How best you can help. The students are here, the professors are here, working in this field. A very good topic of biodiversity. You have a national seminar on this. So, an applied aspect. The disappearance of the traditional belief systems as are now considered mere superstitions, but were fundamental to the concept of sacred forests. Then rapid urbanization and developmental activities such as roads, railways, tracks, dams, including commercial forestry, have been destroying various sacred forests. Then encroachment has led to the shrinkage of some of the large forests. The transformation of the primitive forms of nature worship into formal temple worship. This is a first primitive worship pattern was there. Let me give an example. All of you heard of Sigundareshwari temple. First it was a very small deity under a beautiful tree, peepal tree, in a sacred grove on the bank of that Hole Bagilu called. And now suddenly there was an upsurge in and the beautiful temple was erected, almost 10 to 15 acres of land was deforested. And from a small cottage type structure to a big temple it has come, and it has become an all India pattern. So, the temple culture resulted in the destruction of ecological balance in the area and a lot of garbages, and it has become more of a tourist spot rather than of a divine spot. There are in the temples the difference is tourist spot and divine spot. Divine spot is always revered and respected, whereas a tourist, that is altogether a different attitude. That is, now invasion by exotic weeds is a serious threat to some groves. Pressure due to increasing livestock and fuel wood collection, that is another thing. Fuel wood collection and grazing of livestock. Nowadays, for example, in the Western Ghat region, traditionally, so gomalas were there. Have you heard of gomalas? The community cow lands. That is exclusively, but because of population pressure, and that is being systematically encroached. And nowadays we cannot see gomalas. It is very difficult. And similarly, Soppinabetta system. Traditionally, we have Soppinabetta system. Professor Naik knows very well. He also from the coastal region, and it was well protected. The philosophy of Soppinabetta system was community ownership and individual maintenance. Community ownership. There is no legal ownership for any individual. One cannot sell it out. One cannot destroy that one. He can get the leaves and litters from that trees. Well maintained. But nowadays that is subjected to plantation crops. That is also encroached. So, the loss of Soppinabetta system and gomalas, the major threats in the Western Ghat region regarding the biodiversity, where a lot of awareness program need to be created among the farming communities. Now, next. Since the time immemorial, spelling mistake, that should be time immemorial, the Indians have developed a time-tested organic farming system both as tradition and institution. That is, nowadays it has become an all India character. Many clique organizations have started highlighting the importance of organic farming. But another issue is, through organic farming, we can provide food security to the nation. That is another issue. But a lot of research is going on regarding the organic farming. Particularly agriculture universities have taken up. For example, our own university have Organic Farming Research Center. It must have started during his regime, I think, our MK Naik's regime. It is make a very good contribution. Iruvaki, yes, thank you.
By default, it is organic.
Yes, organic farming. Actually, hats off to Professor Bhatt. Nice organization, nice seminar. It is really a very good seminar. But few progressive farmers should have been invited here. Its picture would have been different. They could have asked a lot of questions to our Professor Basavaraj regarding beacon. And our university has a very major project on apiculture. That is ASCRP project. And we have got a Brahmavara Agriculture Research Station. In fact, in the entire South Kannada region, people stopped paddy cultivation because of labor problem, cost of labor. And this Brahmavara Agriculture Research Station started developing a mechanized paddy cultivation system. They popularized small machines to transplantation, harvesting, and now it has become very popular here. That is. So here, why I have developed a time-tested organic farming system, both as tradition and institution. Now, let me tell you one thing. Recently, Professor Palekar started highlighting on organic farming, like Jivamrita. And in Shumagga in APMC yard, his lecture was there. There we asked one question. Whatever, sir, you are telling, it is not new. As early as 1970s, ICAR has published 11 volumes, that is cultivation practices during Vedic period. It is an excellent contribution. There all the system, Jivamrita, Panchamrita and all these things are there, biodynamic farming, everything is meticulously reported there, which research result also. Agnihotra farming was there. 11 volumes. ICR has published. And the summary of that one he started propagating. So don't be under the impression that the agriculture scientists are against the organic farming. They support that, but integrated farming system, because food security is the priority. Now here, Vrukshayurveda gives a detailed account of organic farming. Vrukshayurveda by Parashara, sage Parashara, and that is one of the oldest analogy of organic farming in the world. Where several countries even never dreamt of cultivation practices, and during that time, Vrukshayurveda, a very concrete, it is my suggestion all of you should read that book. Then a very pertinent command over this biodiversity, ecodiversity, we can get it. How that is prospered, very practical analysis he made. And then good reference material, that is farming system, and even today this great work serves as a good reference material. It is based on the firm principle of Panchamahabhuta and Naisargika Krishi or natural farming. Next. Now coming to the topic, anthropogenic influence on biodiversity, two types, micro influence and macro influence. Now let me give the micro influence, how it affects. In every field you can see gutka packets. It has become a menace. ನೀವು ಅಚ್ಚ ಕನ್ನಡದಲ್ಲಿ ಹೇಳಿದರೆ ತೋಟದಲ್ಲಿ ಎಂಥ ಪ್ರಾಬ್ಲಮ್ ಆಗಿದೆ ಅಂದರೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಅರ್ಕಾನಟ್ ಹಾರ್ವೆಸ್ಟ್ 25 to 30 ಲೇಬರ್ ಕಮ್, and each one minimum 5 gutka packets he consumes. Just imagine. You see, please note it, the heap of sand on the seashore never problems us, but a small speck of sand in our shoe, and it causes, like that it has micro level. ಇವತ್ತು ನೀವು ತೋಟದಲ್ಲಿ 1 ಇಂಚ್ ಭೂಮಿನೂ ಪ್ಲಾಸ್ಟಿಕ್ ಇಲ್ಲದೇ ನೋಡಲಿಕ್ಕಾಗೋದಿಲ್ಲ. Nothing, yes, and then they throw it out there. ಏನಾಗ್ತದೆ, ಪ್ರಾಬ್ಲಮ್ ಏನಾಗ್ತದೆ, ನಾನು ಹೇಳ್ತೇನೆ ದಟ್ ಈಗ ಮೊದಲೇ ಲೇಬರ್ ಸಿಗೋದು, ಇಟ್ ಈಸ್ ವೆರಿ ಪ್ರಾಬ್ಲಮ್ಯಾಟಿಕ್ ಟು ಗೆಟ್ ಅಗ್ರಿಕಲ್ಚರ್ ಲೇಬರ್ ಈಸ್ ವೆರಿ ಪ್ರಾಬ್ಲಮ್. ಆ ಪ್ಲಾಸ್ಟಿಕ್ಸನ್ನು ತೆಗೆಸ್ಲಿಕ್ಕೆ ನಾವು ಮತ್ತೆ ಲೇಬರ್ ಹಾಕಬೇಕು, ಮತ್ತು ಅವ್ರು ಗುಟ್ಗ ತಿನ್ಕೊಂಡು ಅಲ್ಲೇ ಹಾಕಿರ್ತಾರೆ. ದಟ್ ಈಸ್ ವೈ ಐ ರಿಕ್ವೆಸ್ಟ್, ಮೈ ಸಿನ್ಸಿಯರ್ ರಿಕ್ವೆಸ್ಟ್ ಟು ಫಾರ್ಮಿಂಗ್ ಕಮ್ಯುನಿಟಿ, this type of organizations, if farmers come, they learn a lot of things, and it becomes applicable to them practically. And then ಇದೇ ಪ್ರಕಾರ ಅವರು ಊಟಕ್ಕೆ ಈ ಪಾಲಿಥಿನ್ ಬ್ಯಾಗಲ್ಲಿ ಫುಡ್ ತರ್ತಾರೆ, ಅಲ್ಲೇ ಎಸಿತಾರೆ. ದೆನ್ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, ಎಷ್ಟು ನೀವು ನ್ಯೂಟ್ರಿಷನ್ ಗೊಬ್ಬರ ಏನೇ ಹಾಕಿದರೂ ಕೂಡ ಬೇರೆ ತಗೊಳ್ಳಿ, ಯಾಕೆಂದರೆ ಅದ್ರ ಬೇರೆ ಮೇಲೆ ಕೂತ್ಕೊಂಡ್ಬಿಟ್ಟಿರ್ತದೆ ಪ್ಲಾಸ್ಟಿಕ್ಕು. It the nutrition uptake will not be there. So this is the major problem, that the micro level. Can you see an inch of land in India, a road in India, without plastic, without empty bottles, without sachets? So what to do? Only creating awareness, vast extension program, organizing training programs in the safe disposal of plastics. That is the only alternative, because plastic we cannot completely eliminate. It has become a part of our nature. Only thing, how best we can, that is micro level, micro influence. It has become a very big menace. And then macro level, worst hit sector is farming. Nowadays farms have become dustbin of plastics. Insecticides and pesticides. ಇನ್ನೂ ಹೇಳಬೇಕು ಅಂದರೆ, ವಿ ಪರ್ಚೇಸ್ ಮೆನ್ಯೂರ್, ಕೌಡಂಗ್ ಮೆನ್ಯೂರ್. ಆ ಗೊಬ್ಬರ ಹಳ್ಳಿಗಳಲ್ಲಿ ಗೊಬ್ಬರ ತಯಾರು ಮಾಡ್ತಾರಲ್ಲ, ಇನ್ ದಿ ವಿಲೇಜಸ್ ದೇ ಪ್ರಿಪೇರ್ ಕೌಡಂಗ್ ಮೆನ್ಯೂರ್. ಅಲ್ಲಿ ವೇಸ್ಟ್ ಪ್ಲಾಸ್ಟಿಕ್ಸ್ ಎಲ್ಲ ಅದರಲ್ಲಿ ಹಾಕಿರ್ತಾರೆ. ಆ್ಯಂಡ್ ವೆನ್ ವಿ ಬೈ ಇಟ್ ಅಲಾಂಗ್ ವಿತ್ ಪ್ಲಾಸ್ಟಿಕ್ಸ್ ವಿ ಗೆಟ್ ಇಟ್, ಆ್ಯಂಡ್ ದಟ್ ಈಸ್ ಡಂಪ್ಡ್ ಇನ್ ಟು ಅವರ್ ಎಸ್ಟೇಟ್ಸ್. ವಾಟ್ ವಿಲ್ ಹ್ಯಾಪನ್? Water will not be percolated, and it affects the root, and it attracts pesticides and insects and lot of diseases. So these practices we should, insects and pesticides, many of the pests and insects have become immune to such pesticides and insecticides. Weedicide is posing a potential threat to land productivity. Weedicide, ಕಳೆನಾಶಕಗಳು ಹೇಳ್ತಾರಲ್ಲ, it is posing a very big threat to land productivity. Next, macro effect, global warming. And nowadays it is said that the net falling in the Arka is because of warming, global warming, temperature. Then global threat to farm productivity, potential threat to food security, depletion of global water resources, adverse effect on human health. The relationship between culture and ecology was the integral part of ancient Indian society. Interactions with diverse biological entities and concern for their conservation remained important components in the structure of ancient civilization of India. Next. The relationship between culture and ecology was the integral part of ancient Indian society. interaction with diverse biological entities and concern of their conservation remained important components in the structure of ancient civilizations of India. Action plan. This type of forum should develop an action plan, making a resolution. You see, there are two types of personalities. One is part of the problem, another is part of the solution. Should our personality become part of the solution or part of the problem? Part of the solution. Now we have to struggle. We have to make a recommendations how best we can solve this problem. And re-establishing biodiversity is not a very big issue. As you know, all of us have 5 senses: touch, taste, listen, hearing, like that, panchendriyagalu. Few people have 6th sense. That is nothing but common sense. And abundance of common sense is called wisdom. This is not a very big problem. Only thing is it needs common sense. Common sense, not to throw plastics in the agricultural land. That is common sense. Not to throw empty bottles and sachets on the road. That is common sense. So purely our recommendations doesn't pertain for a, please, as the students are there, it is our experience that tells you, for a successful life and career, only 20% intelligence is sufficient and 80% common sense. That is why here some resolutions, it is not a big revolution, doesn't need a huge money or like that, huge capital, infrastructure, those things. Creating awareness about the importance of biodiversity conservation, that is at this elementary level. At the childhood level, that should be at the primary level, awareness on that is very important. Gidavagi baggadu maravagi baggite anta. At the childhood level, when we create impression, that lasts forever. Mind can be managed, maintained, nurtured. Uncultivated mind is a liability, as uncultivated land is a liability. Whatever may be the level of fertility, it is of no use, like that. So we have to give proper importance in the socialization process of childhood. That is, here I see the school going children opening the chocolate and throwing that on the road. So that becomes a part of their personality. You see, practice makes man perfect, but imperfect practice makes man imperfect. Simple logic. Then inclusion of practical modules in every extension programs by the academic institutions. Really this is a very wonderful program. But academic institutions and in line departments of agriculture, horticulture, industry and among others. Why I am giving much importance to these academicians? Now this eco-diversity is in the most dangerous position. We in the rural area, we understand the direct impact of this one. And this is only the academicians can help saving this country from natural drifting. Whenever this country was on the stage of collapse, this is only the academicians who helped this country to rise to the higher level. When the entire more than 60% of population was suffering from hunger, acute shortage of food, it was the professor assistant professor in Madras University, Swaminathan, an academician, saved this country through Green Revolution. Later, when the massive unemployment program throughout India, the youngsters were struggling hard to earn their livelihood. At that time came an economics professor of Punjab University, Manmohan Singh. Liberalization, globalization and privatization. Nowadays you cannot see a single youngster, even with 10th standard, 7th standard, he can get a one or the other job. So 60% of this vast youngsters, that is again the academicians protected this country from drifting. So we must feel proud that this is the academicians, as Plato told, academicians alone can rule the nation, because we are capable of thinking beyond ourselves. So with this, the massive program should be created. Particularly, you can involve NSS activities, can become one of the important NSS activities, where you work in the NSS camps in the villages, and there you can create awareness. That is Department of Agriculture, Horticulture, Industry and among others. Thank you all. If you have any question, you can ask. And this is not about intelligence, it is purely common sense approach, and that is needed. Thank you.
Any questions? Any questions? Okay. That was indeed a knowledgeable session. We extend our sincere gratitude to our presenter, Dr. M.S. Vignesh, for sharing the expertise and insights with us. We have another resource person with us. I request Vaishnavi Pai to introduce Mr. Dr. M.K. Nayak. Before we conclude this session, I would like to invite Mr. Shailaja H., IQAC Coordinator and Head of the Department Physics, to present a memento as a token of appreciation to our guest speaker. Where is she? Ishwar Bhatt will give her. हाँ ಈಗ ಯಾರಿಗೆ present Okay. So I request MK. Thank you, sir. Here we can conclude the session. I thank each and everyone for your patient listening. Thank you.
Good afternoon, everyone. We shall disperse for lunch shortly. But before that, there are a couple of announcements. Participants of the seminar, kindly note, there's a feedback form included in the files given to you at the time of registration. Please fill those forms and drop them into the boxes kept just outside the hall after the next technical session. At this moment, I would like to take the opportunity to thank the Department of Journalism of MGM College. We are immensely thankful for their support on both days, including photo and video coverage of the entire seminar for documentation and live streaming of the events of both days on YouTube and Facebook Live. I thank Mr. Suchit Kotian, Head of the Journalism Department, Mr. Manoj Kumar, Assistant Professor, Journalism Department, and all their students for helping us out in our time of need. Thank you, everyone. We shall break for lunch now.
Sometimes there's no one else who loves me like you used to. Sometimes
ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲೇ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949 ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲುವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ H. ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. ಸಾವಿರದ 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡದಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಮೌಲ್ಯಾಂಕನದ 4ನೇ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ PA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ನ ನೂತನ ರಿಜಿಸ್ಟ್ರಾರ್ರಾದ ಡಾಕ್ಟರ್ ರಂಜನ್ R ಪೈಗಳು, ಕಾಲೇಜಿನ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾದ ಶ್ರೀ T ಸತೀಶ್ U ಪೈಗಳು ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಸದಸ್ಯರಾದ ಡಾಕ್ಟರ್ HS ಬಳ್ಳಾಲ್ ಮತ್ತು ಶ್ರೀ ವರದರಾಯ ಪೈಗಳ ಸೂಕ್ತ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕಾಲೇಜು ಹೊಸ ಸಂವೇದನೆಯತ್ತ ಸಾಗುತ್ತಿದೆ. ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ H ಸುಂದರ್ ರಾಯರಿಂದ ಇಂದಿನವರೆಗೆ ಹಲವಾರು ದಕ್ಷ ಪ್ರಾಮಾಣಿಕ ಪ್ರಾಂಶುಪಾಲರಿಂದ ಸಾಮಾಜಿಕವಾಗಿ ಸ್ಪಂದಿಸುತ್ತಾ ಸಾಧನೆಯ ಪಥದತ್ತ ಸಾಗಿ ಬಂದಿದೆ. ಪ್ರಸ್ತುತ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ K ಲಕ್ಷ್ಮೀನಾರಾಯಣ ಕಾರಂತ ಅವರ ನೇತೃತ್ವದಲ್ಲಿ ಇಂದಿನ ಸಾಮಾಜಿಕ ಅಗತ್ಯಕ್ಕೆ ತಕ್ಕಂತೆ ಅನೇಕ ಅಭಿವೃದ್ಧಿ ಕಾರ್ಯಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡು ಸಾಧನೆಯತ್ತ ಆಶಾಭಾವನೆಯನ್ನು ಹೊಂದಿ ನಿಂತಿದೆ. ಅಮೃತ ಮಹೋತ್ಸವದ ಸ್ಮರಣಾರ್ಥ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಪ್ಲಾಟಿನಂ ಜುಬಿಲಿ ಕಟ್ಟಡ ನಿರ್ಮಾಣ, ಮುದ್ದಣ ಮಂಟಪವನ್ನು ಮೇಲ್ದರ್ಜೆಗೇರಿಸುವ ಮೂಲಕ ನವೀಕರಣ, ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಕೌಶಲ್ಯಾಭಿವೃದ್ಧಿ ಕೇಂದ್ರದ ಸ್ಥಾಪನೆ, 75 ವರ್ಷಗಳ ಸವಿನೆನಪಿಗಾಗಿ 75 ಅರ್ಥಪೂರ್ಣ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಆಯೋಜಿಸಲು ಸಂಕಲ್ಪಿಸಲಾಗಿದೆ. 1949-50ರಿಂದ 2023ರ ತನಕ ಕಾಲೇಜಿನ ಇತಿಹಾಸ ಶೈಕ್ಷಣಿಕ ವಿಷಯದಲ್ಲಿ ಅತಿ ಮಹತ್ವದ್ದಾಗಿದೆ. ಒಬ್ಬ ಧೀಮಂತ ವ್ಯಕ್ತಿಯ ಕನಸು ಈಗ ಲಕ್ಷಾಂತರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿದ್ಯಾರ್ಜನೆ ಮಾಡುತ್ತಾ ಒಂದು ಸಣ್ಣ ಶಾಲೆಯಲ್ಲಿ 89 ವಿದ್ಯಾರ್ಥಿಗಳಿಂದ ಆರಂಭಗೊಂಡ ಸಂಸ್ಥೆ ಇದೀಗ ವರ್ಷಕ್ಕೆ 3,000ಕ್ಕೂ ಅಧಿಕ ವಿದ್ಯಾರ್ಥಿಗಳು MGMನ ಅಂಗಳದಲ್ಲಿ ಶಿಕ್ಷಣ ಪಡೆಯುತ್ತಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ಸಾಧನೆಗೆ ಕಾರಣರಾದ ಪ್ರತಿಯೋರ್ವ ಆಡಳಿತ ಮಂಡಳಿಯವರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ಪೋಷಕ ವೃಂದ ಮತ್ತು ಎಲ್ಲ 75 ವರ್ಷಗಳ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಧನ್ಯವಾದಗಳು. ಇಂದಿನಿಂದ MGMನಲ್ಲಿ 75ರ ಅಮೃತ ಮಹೋತ್ಸವದ ಹಬ್ಬ ಶುರುವಾಗ್ತಿದೆ.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balanoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1,600 men and 25 elephants. The zoological nomenclature of this organism is Balanoptera. It has a special reference to the enormous big feeding device called as baggins. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these baggins help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಿಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balanoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales. It is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balanoptera. It has a special reference to the enormous big feeding device called as baggins. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these baggins help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು.
ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗ ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲಿ ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949 ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ ಎಚ್ ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. ಸಾವಿರದ 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡಗಳು ವಿಸ್ತರಿಸಿದವು. ಹೀಗೆ MGM ಕಾಲೇಜು ಹಂತ ಹಂತವಾಗಿ ಯಶಸ್ಸಿನ ಪಥದಲ್ಲಿ ಮುಂದೆ ಸಾಗಿತು. ಉಡುಪಿಯಲ್ಲಿ MGM ಕಾಲೇಜನ್ನು ತೆರೆದ ಬಳಿಕ ಡಾಕ್ಟರ್ ಮಾಧವ ಪೈಗಳು ಜಿಲ್ಲೆಯ ಬೇರೆ ಬೇರೆ ಕಡೆಗಳಲ್ಲಿ ವಿವಿಧ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭಿಸಿದರು. ಇಂದು ವಿಶ್ವದ ಪ್ರಮುಖ ವಿದ್ಯಾ ಕೇಂದ್ರವಾಗಿರುವ ಮಣಿಪಾಲದ ಕಾಲೇಜುಗಳು ಹಾಗೂ ನೆರೆಯ ಜಿಲ್ಲೆಗಳಲ್ಲೂ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭ ಮಾಡಲು ಮೂಲ ಪ್ರೇರಣೆ MGM ಕಾಲೇಜಿನ ಯಶಸ್ಸು. ಈ ಎಲ್ಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳಿಗೆ ಮುಕ್ಕುಟಮಣಿಯಾಗಿ ಇಂದು MGM ಕಾಲೇಜು ರಾರಾಜಿಸುತ್ತಿದೆ. ಶೈಕ್ಷಣಿಕವಾಗಿ ಮಹತ್ತರವಾದ ಸಾಧನೆಯನ್ನು ಮಾಡಿರುವ MGM ಕಾಲೇಜು ಉಡುಪಿಯ ಸಾಂಸ್ಕೃತಿಕ ಚಟುವಟಿಕೆಗಳ ಕೇಂದ್ರವಾಗಿ ಗುರುತಿಸಿಕೊಂಡಿದೆ. ಕಾಲೇಜಿನ ದ್ವಿತೀಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೂಶಿ ಹರಿದಾಸ ಭಟ್ಟರು ಆ ಹುದ್ದೆಯ ಘನತೆ ಗೌರವಗಳನ್ನು ತಮ್ಮ ಪ್ರತಿಭೆ, ಪಾಂಡಿತ್ಯ ಮತ್ತು ಪರಿಶ್ರಮಗಳಿಂದ ಹೆಚ್ಚಿಸಿದರು. 2010ರಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ PA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ನ ನೂತನ ರಿಜಿಸ್ಟ್ರಾರ್ರಾದ ಡಾಕ್ಟರ್ ರಂಜನ್ R ಪೈಗಳು, ಕಾಲೇಜಿನ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾದ ಶ್ರೀ T ಸತೀಶ್ U ಪೈಗಳು ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಸದಸ್ಯರಾದ ಡಾಕ್ಟರ್ HS ಬಳ್ಳಾಲ್ ಮತ್ತು ಶ್ರೀ ವರದರಾಯ ಪೈಗಳ ಸೂಕ್ತ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕಾಲೇಜು ಹೊಸ ಸಂವೇದನೆಯತ್ತ ಸಾಗುತ್ತಿದೆ. ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಎಚ್ ಸುಂದರರಾಯರಿಂದ ಇಂದಿನವರೆಗೆ ಹಲವಾರು ದಕ್ಷ ಪ್ರಾಮಾಣಿಕ ಪ್ರಾಂಶುಪಾಲರಿಂದ ಸಾಮಾಜಿಕವಾಗಿ ಸ್ಪಂದಿಸುತ್ತಾ ಸಾಧನೆಯ ಪಥದತ್ತ ಸಾಗಿ ಬಂದಿದೆ. ಪ್ರಸ್ತುತ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೆ ಲಕ್ಷ್ಮೀನಾರಾಯಣ ಕಾರಂತ ಅವರ ನೇತೃತ್ವದಲ್ಲಿ ಇಂದಿನ ಸಾಮಾಜಿಕ ಅಗತ್ಯಕ್ಕೆ ತಕ್ಕಂತೆ ಅನೇಕ ಅಭಿವೃದ್ಧಿ ಕಾರ್ಯಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡು ಸಾಧನೆಯತ್ತ ಆಶಾಭಾವನೆಯನ್ನು ಹೊಂದಿ ನಿಂತಿದೆ. ಅಮೃತ ಮಹೋತ್ಸವದ ಸ್ಮರಣಾರ್ಥ ಶ್ರೀ ಟಿ ಮೋಹನ್ ದಾಸ್ ಪೈ ಪ್ಲಾಟಿನಂ ಜುಬಿಲಿ ಕಟ್ಟಡ ನಿರ್ಮಾಣ, ಮುದ್ದಣ ಮಂಟಪವನ್ನು ಮೇಲ್ದರ್ಜೆಗೇರಿಸುವ ಮೂಲಕ ನವೀಕರಣ, ಶ್ರೀ ಟಿ ಮೋಹನ್ ದಾಸ್ ಪೈ ಕೌಶಲ್ಯಾಭಿವೃದ್ಧಿ ಕೇಂದ್ರದ ಸ್ಥಾಪನೆ, 75 ವರ್ಷಗಳ ಸವಿನೆನಪಿಗಾಗಿ 75 ಅರ್ಥಪೂರ್ಣ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಆಯೋಜಿಸಲು ಸಂಕಲ್ಪಿಸಲಾಗಿದೆ. 1949-50ರಿಂದ 2023 ತನಕ ಕಾಲೇಜಿನ ಇತಿಹಾಸ ಶೈಕ್ಷಣಿಕ ವಿಷಯದಲ್ಲಿ ಅತಿ ಮಹತ್ವದ್ದಾಗಿದೆ. ಒಬ್ಬ ಧೀಮಂತ ವ್ಯಕ್ತಿಯ ಕನಸು ಈಗ ಲಕ್ಷಾಂತರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿದ್ಯಾರ್ಜನೆ ಮಾಡುತ್ತಾ ಒಂದು ಸಣ್ಣ ಶಾಲೆಯಲ್ಲಿ 89 ವಿದ್ಯಾರ್ಥಿಗಳಿಂದ ಆರಂಭಗೊಂಡ ಸಂಸ್ಥೆ ಇದೀಗ ವರ್ಷಕ್ಕೆ 3,000ಕ್ಕೂ ಅಧಿಕ ವಿದ್ಯಾರ್ಥಿಗಳು MGMನ ಅಂಗಳದಲ್ಲಿ ಶಿಕ್ಷಣ ಪಡೆಯುತ್ತಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ಸಾಧನೆಗೆ ಕಾರಣರಾದ ಪ್ರತಿಯೋರ್ವ ಆಡಳಿತ ಮಂಡಳಿಯವರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ಪೋಷಕ ವೃಂದ ಮತ್ತು ಎಲ್ಲ 75 ವರ್ಷಗಳ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಧನ್ಯವಾದಗಳು. ಇಂದಿನಿಂದ MGMನಲ್ಲಿ 75ರ ಅಮೃತ ಮಹೋತ್ಸವದ ಹಬ್ಬ ಶುರುವಾಗ್ತಿದೆ. ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲಿ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949ರಂದು ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ, ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ ಎಚ್ ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. ಸಾವಿರದ 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡಗಳು ವಿಸ್ತರಿಸಿದವು. ಹೀಗೆ MGM ಕಾಲೇಜು ಹಂತ ಹಂತವಾಗಿ ಯಶಸ್ಸಿನ ಪಥದಲ್ಲಿ ಮುಂದೆ ಸಾಗಿತು. ಉಡುಪಿಯಲ್ಲಿ MGM ಕಾಲೇಜನ್ನು ತೆರೆದ ಬಳಿಕ ಡಾಕ್ಟರ್ ಮಾಧವ ಪೈಗಳು ಜಿಲ್ಲೆಯ ಬೇರೆ ಬೇರೆ ಕಡೆಗಳಲ್ಲಿ ವಿವಿಧ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭಿಸಿದರು. ಇಂದು ವಿಶ್ವದ ಪ್ರಮುಖ ವಿದ್ಯಾ ಕೇಂದ್ರವಾಗಿರುವ ಮಣಿಪಾಲದ ಕಾಲೇಜುಗಳು ಹಾಗೂ ನೆರೆಯ ಜಿಲ್ಲೆಗಳಲ್ಲೂ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭ ಮಾಡಲು ಮೂಲ ಪ್ರೇರಣೆ MGM ಕಾಲೇಜಿನ ಯಶಸ್ಸು. ಈ ಎಲ್ಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳಿಗೆ ಮುಕ್ಕುಟಮಣಿಯಾಗಿ ಇಂದು MGM ಕಾಲೇಜು ರಾರಾಜಿಸುತ್ತಿದೆ. ಶೈಕ್ಷಣಿಕವಾಗಿ ಮಹತ್ತರವಾದ ಸಾಧನೆಯನ್ನು ಮಾಡಿರುವ MGM ಕಾಲೇಜು ಉಡುಪಿಯ ಸಾಂಸ್ಕೃತಿಕ ಚಟುವಟಿಕೆಗಳ ಕೇಂದ್ರವಾಗಿ ಗುರುತಿಸಿಕೊಂಡಿದೆ. ಕಾಲೇಜಿನ ದ್ವಿತೀಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೂಶಿ ಹರಿದಾಸ ಭಟ್ಟರು ಆ ಹುದ್ದೆಯ ಘನತೆ ಗೌರವಗಳನ್ನು ತಮ್ಮ ಪ್ರತಿಭೆ, ಪಾಂಡಿತ್ಯ ಮತ್ತು ಪರಿಶ್ರಮಗಳಿಂದ ಹೆಚ್ಚಿಸಿದರು. 2010ರಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಮೌಲ್ಯಾಂಕನದ 4ನೇ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ TA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ನ ನೂತನ ರಿಜಿಸ್ಟ್ರಾರ್ ಆದ ಡಾಕ್ಟರ್ ರಂಜನ್ R ಪೈಗಳು, ಕಾಲೇಜಿನ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾದ ಶ್ರೀ T ಸತೀಶ್ U ಪೈಗಳು ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಸದಸ್ಯರಾದ ಡಾಕ್ಟರ್ HS ಬಳ್ಳಾಲ್ ಮತ್ತು ಶ್ರೀ ವರದರಾಯ ಪೈಗಳ ಸೂಕ್ತ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕಾಲೇಜು ಹೊಸ ಸಂವೇದನೆಯತ್ತ ಸಾಗುತ್ತಿದೆ. ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ H ಸುಂದರ್ ರಾಯರಿಂದ ಇಂದಿನವರೆಗೆ ಹಲವಾರು ದಕ್ಷ ಪ್ರಾಮಾಣಿಕ ಪ್ರಾಂಶುಪಾಲರಿಂದ ಸಾಮಾಜಿಕವಾಗಿ ಸ್ಪಂದಿಸುತ್ತಾ ಸಾಧನೆಯ ಪಥದತ್ತ ಸಾಗಿ ಬಂದಿದೆ. ಪ್ರಸ್ತುತ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೆ ಲಕ್ಷ್ಮೀನಾರಾಯಣ ಕಾರಂತ ಅವರ ನೇತೃತ್ವದಲ್ಲಿ ಇಂದಿನ ಸಾಮಾಜಿಕ ಅಗತ್ಯಕ್ಕೆ ತಕ್ಕಂತೆ ಅನೇಕ ಅಭಿವೃದ್ಧಿ ಕಾರ್ಯಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡು ಸಾಧನೆಯತ್ತ ಆಶಾಭಾವನೆಯನ್ನು ಹೊಂದಿ ನಿಂತಿದೆ. ಅಮೃತ ಮಹೋತ್ಸವದ ಸ್ಮರಣಾರ್ಥ ಶ್ರೀ ಟಿ ಮೋಹನ್ ದಾಸ್ ಪೈ ಪ್ಲಾಟಿನಂ ಜುಬಿಲಿ ಕಟ್ಟಡ ನಿರ್ಮಾಣ, ಮುದ್ದಣ ಮಂಟಪವನ್ನು ಮೇಲ್ದರ್ಜೆಗೇರಿಸುವ ಮೂಲಕ ನವೀಕರಣ, ಶ್ರೀ ಟಿ ಮೋಹನ್ ದಾಸ್ ಪೈ ಕೌಶಲ್ಯಾಭಿವೃದ್ಧಿ ಕೇಂದ್ರದ ಸ್ಥಾಪನೆ, 75 ವರ್ಷಗಳ ಸವಿನೆನಪಿಗಾಗಿ 75 ಅರ್ಥಪೂರ್ಣ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಆಯೋಜಿಸಲು ಸಂಕಲ್ಪಿಸಲಾಗಿದೆ. 1949-50ರಿಂದ 2023 ತನಕ ಕಾಲೇಜಿನ ಇತಿಹಾಸ ಶೈಕ್ಷಣಿಕ ವಿಷಯದಲ್ಲಿ ಅತಿ ಮಹತ್ವದ್ದಾಗಿದೆ. ಒಬ್ಬ ಧೀಮಂತ ವ್ಯಕ್ತಿಯ ಕನಸು ಈಗ ಲಕ್ಷಾಂತರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿದ್ಯಾರ್ಜನೆ ಮಾಡುತ್ತಾ ಒಂದು ಸಣ್ಣ ಶಾಲೆಯಲ್ಲಿ 89 ವಿದ್ಯಾರ್ಥಿಗಳಿಂದ ಆರಂಭಗೊಂಡ ಸಂಸ್ಥೆ ಇದೀಗ ವರ್ಷಕ್ಕೆ 3,000ಕ್ಕೂ ಅಧಿಕ ವಿದ್ಯಾರ್ಥಿಗಳು MGMನ ಅಂಗಳದಲ್ಲಿ ಶಿಕ್ಷಣ ಪಡೆಯುತ್ತಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ಸಾಧನೆಗೆ ಕಾರಣರಾದ ಪ್ರತಿಯೋರ್ವ ಆಡಳಿತ ಮಂಡಳಿಯವರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ಪೋಷಕ ವೃಂದ ಮತ್ತು ಎಲ್ಲ 75 ವರ್ಷಗಳ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಧನ್ಯವಾದಗಳು. ಇಂದಿನಿಂದ MGMನಲ್ಲಿ 75ರ ಅಮೃತ ಮಹೋತ್ಸವದ ಹಬ್ಬ ಶುರುವಾಗ್ತಿದೆ. ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿ ಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲಿನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರಿದೆ
and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales. It is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1,600 men and 25 elephants. The zoological nomenclature of this organism is Balanoptera. It has a special reference to the enormous big feeding device called as baggins. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these baggins help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಿಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿ ಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲೀನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balaenoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balaenoptera. It has a special reference to the enormous big feeding device called as baggins. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them large amount of debris and planktons come, and these baggins help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿ ತಿಮಿಂಗಿಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು. ನಮಸ್ಕಾರ, ನಾನು ಸ್ನೇಹ ವರ್ಗೀಸ್. ನಾವಿಂದು MGM ಕಾಲೇಜಿನ ಝೂವಾಲಜಿ ವಿಭಾಗದ ಮುಂದಿರುವಂತಹ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಅಸ್ಥಿಪಂಜರದ ಬಗ್ಗೆ ಮಾಹಿತಿಯನ್ನು ನೀಡಲು ಬಂದಿದ್ದೇವೆ. ನಿಮಗೆಲ್ಲ ನೀಲಿ ತಿಮಿಂಗಿಲದ ಬಗ್ಗೆ ಎಷ್ಟು ಗೊತ್ತು? ಬನ್ನಿ, ನಾವಿಂದು ತಿಳಿಸಿಕೊಡುತ್ತೇವೆ. ತಿಮಿಂಗಿಲಗಳು ಸಾಗರ ಸಸ್ತನಿಗಳು. ಇದು ಬಲೀನ್ ತಿಮಿಂಗಿಲ ಎಂಬ ಜಾತಿಗೆ ಸೇರುತ್ತದೆ. ಇವುಗಳು 30 ಮೀಟರ್ ಉದ್ದ ಹಾಗೂ 180 ಟನ್ ತೂಕ ಇರುತ್ತದೆ. ಇವು 20ನೇ ಶತಮಾನದಲ್ಲಿ ಭೂಮಿಯ ಎಲ್ಲ ಸಾಗರಗಳಲ್ಲಿ ಕಾಣಸಿಗುತ್ತಿದ್ದವು. ಇಲ್ಲಿಯವರೆಗೆ 3 ರೀತಿಯ ನೀಲಿ ತಿಮಿಂಗಿಲಗಳ ಜಾತಿಯು ಇಂಡಿಯನ್ ಮತ್ತು ಪೆಸಿಫಿಕ್ ಸಮುದ್ರದಲ್ಲಿ ಕಾಣಿಸಿಕೊಂಡಿದೆ. ಇವುಗಳು ನೀರಲ್ಲಿ ವಾಸಿಸುವ ಮೀನನ್ನು ಹಾಗೂ ನೀರಿನಲ್ಲಿ ವಾಸಿಸುವಂತಹ ಪ್ರಾಣಿಯನ್ನು ಹಿಡಿದು ತಿನ್ನುತ್ತದೆ. ಬನ್ನಿ, ಇನ್ನಷ್ಟು ಮಾಹಿತಿಯನ್ನು ಇದೇ ವಿಭಾಗದ ವಿದ್ಯಾರ್ಥಿನಿಯಿಂದ ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳೋಣ.
This skeleton which you see is the skeleton of Balaenoptera, called as the giant of the sea. This was located on Malpe and was considered to be washed ashore on October 1977. As it was selected in a state that could not be presented, it needed construction and amendments. And finally, on 16th August 1978, it was installed in this corridor. The skeleton of this organism weighs about 7.5 tons, and the skull alone weighs about 2.5 tons. This is one of the species of the whales which is considered to be one of the largest and the heaviest species of the whales in the world. If you see the vertebral column of this skeleton, it is very long. It measures about 13 meters in length. The entire weight of this organism is 130 tons, which is a weight which can be only supported by the sea. If you compare the weight of this organism to the other organism, it weighs about 1600 men and 25 elephants. The zoological nomenclature of this organism is Balaenoptera. It has a special reference to the enormous big feeding device called as bags. These are like plates which help in filtering the water. Whenever the whale is trying to feed, it takes up large amount of gallons of water inside, and along with them, large amount of debris and planktons come, and these balance help in filtering those. These organisms always move in herds, and they come near the ocean regions only during the time of breeding or in the search of food. According to the International Union of Conservation and Naturalists, this is one of the species among the 6 species which are considered to be near extinction. So there's a lot of awareness in saving this species of the whales.
ನೋಡಿದ್ರಲ್ಲ ವೀಕ್ಷಕರೇ, ಇದಿಷ್ಟು ನೀಲಿತಿಮಿಂಗಲದ ಮಾಹಿತಿ. ಇನ್ನಷ್ಟು ವಿಡಿಯೋದೊಂದಿಗೆ ಮತ್ತೆ ಸಿಗೋಣ. ಧನ್ಯವಾದಗಳು.
ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲೇ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949 ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ H. ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. ಸಾವಿರದ 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡಗಳು ವಿಸ್ತರಿಸಿದವು. ಹೀಗೆ MGM ಕಾಲೇಜು ಹಂತ ಹಂತವಾಗಿ ಯಶಸ್ಸಿನ ಪಥದಲ್ಲಿ ಮುಂದೆ ಸಾಗಿತು. ಉಡುಪಿಯಲ್ಲಿ MGM ಕಾಲೇಜನ್ನು ತೆರೆದ ಬಳಿಕ ಡಾಕ್ಟರ್ ಮಾಧವ ಪೈಗಳು ಜಿಲ್ಲೆಯ ಬೇರೆ ಬೇರೆ ಕಡೆಗಳಲ್ಲಿ ವಿವಿಧ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭಿಸಿದರು. ಇಂದು ವಿಶ್ವದ ಪ್ರಮುಖ ವಿದ್ಯಾ ಕೇಂದ್ರವಾಗಿರುವ ಮಣಿಪಾಲದ ಕಾಲೇಜುಗಳು ಹಾಗೂ ನೆರೆಯ ಜಿಲ್ಲೆಗಳಲ್ಲೂ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭ ಮಾಡಲು ಮೂಲ ಪ್ರೇರಣೆ MGM ಕಾಲೇಜಿನ ಯಶಸ್ಸು. ಈ ಎಲ್ಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳಿಗೆ ಮುಕ್ಕುಟಮಣಿಯಾಗಿ ಇಂದು MGM ಕಾಲೇಜು ರಾರಾಜಿಸುತ್ತಿದೆ. ಶೈಕ್ಷಣಿಕವಾಗಿ ಮಹತ್ತರವಾದ ಸಾಧನೆಯನ್ನು ಮಾಡಿರುವ MGM ಕಾಲೇಜು ಉಡುಪಿಯ ಸಾಂಸ್ಕೃತಿಕ ಚಟುವಟಿಕೆಗಳ ಕೇಂದ್ರವಾಗಿ ಗುರುತಿಸಿಕೊಂಡಿದೆ. ಕಾಲೇಜಿನ ದ್ವಿತೀಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೂಶಿ ಹರಿದಾಸ ಭಟ್ಟರು ಆ ಹುದ್ದೆಯ ಘನತೆ ಗೌರವಗಳನ್ನು ತಮ್ಮ ಪ್ರತಿಭೆ, ಪಾಂಡಿತ್ಯ ಮತ್ತು ಪರಿಶ್ರಮಗಳಿಂದ ಹೆಚ್ಚಿಸಿದರು. 2010ರಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಮೌಲ್ಯಾಂಕನದ 4ನೇ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ TA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಶನ್ ನೂತನ
I request Dr. Ishwar Bhatt, Professor of Zoology, to escort Dr. Rajgopal sir to the dais. I am delighted to introduce Dr. P.K. Rajgopal sir, the retired professor and HOD of Botany, MGM College, Udupi. Dr. P.K. Rajgopal sir pursued his BSc in our MGM college, MSc in Manasa Gangotri University of Mysore, and obtained his PhD degree from Mangalore University. His doctoral thesis was on fern and fern allies of Karnataka state, a taxonomical study, under the guidance of Dr. K. Gopal Krishna Bhatt. Dr. Rajgopal sir has taught botany for 37 years at MGM College. He has published a book titled Pteridophytes of Karnataka State, jointly with the Dr. K. Gopal Krishna Bhatt. He has also contributed in the Pteridophyte section Flora of Karnataka, recently published by Karnataka Biodiversity Board. He has co-authored the book Karnataka Rajya Kosha Sasya Samputa, published by Hampi University. He has also authored on the number of books and written articles in Kannada on topics of botany of general interest. He has published a few research papers in journals which are Indian Fern Journal, Taxon and Journal of Medicinal Plants. He has even discovered a new species from Udupi which is named as Isotus udupiensis. He has also presented paper in symposia at Pune in Maharashtra, Itanagar in Arunachal Pradesh and Burdwan in West Bengal. Dr. Rajgopal sir is the recipient of Lifetime Achievement Award given by the Indian Fern Society, Chandigarh, in the year of 2017. He was also the president of Indian Fern Society for the tenure of 2 years during the year 2017 and 2018. I would request Mrs. Shruti Nayak ma'am, HOD of Botany Department, to florally welcome our guest. Thank you, ma'am. Now I would like to hand over the stage to Dr. P.K. Rajgopal sir. Over to you, sir.
Of course, a session after a sumptuous food. All dignitaries of the dais, ladies and gentlemen. At the very outset, I thank Shruti Nayak and her colleagues for inviting me to give a presentation today. And to talk here in my alma mater, where I learnt my botany and taught botany for nearly 40 years, student and staff. And I also heartily eminent scholars. The papers were very scholarly and I enjoyed them very much. My presentation is little having a little deviation from this flow. It is on a basic topic of collection and identification of a neglected group of plants called pteridophytes. I think most of the people, except who have studied botany, were not aware of what is meant by pteridophytes. They're actually a neglected group of plants, I can say, and most people consider them as weeds. Under the guidance of Dr. Gopal Krishna Bhatt, we jumped into that topic to find out the distribution of this group in our state, Karnataka state, because it was almost untouched except for few papers here and there. So we did extensive field work and made the herbaria sheets of those collected plants identified them and written the exhaustive description, and subsequently we could manage to publish a book on that topic also. Our field work was nearly spread over 4 to 5 years in the mainly with the Western Ghats, more than 100 collection trips, and followed by the literature collection, visiting to various herbaria, identifying them, and making herbaria of cultivated plants. So all these went off, and it took nearly 5 years. So that will be the basis of my presentation today here. And with that scarce knowledge of pteridophytes, I have made a small PPT about what are pteridophytes, their biodiversity, of course, within our limit, and what danger they are facing and how to overcome that. First slide. First of all, what are pteridophytes? As I already told you, it is a small group of plants, to say in a way, totally neglected. And if I say pteridophytes, many people may not understand. If I say ferns, people may get some knowledge about them. They are called generally coming under ferns, and their relative, their related plants are called fernelites. So ferns and fernelites together become pteridophytes. Now, when we say, so it's a group of plants. When we say plant, generally a layman will have a picture of it should have a root, stem, leaf, flower, fruit and seed. But students of botany know that there can be a plant without any one of these. For example, algae, no stem, no root, no leaf, no flower, no seed, but still they are plants. So in that evolutionary history of plants, these pteridophytes come somewhere in the midway. They are exactly in the middle place of the plant kingdom. So above them we are having gymnosperms and flowering plants, angiosperms, and below them there will be bryophytes and algae. So such a very, what can I say, very important place they are occupying in the plant kingdom. And as it is clear, here you can see that this is a good, as they are in the midway, see they do not have root, stem, leaf. So they have root, stem, leaf, but they do not have flower, fruits and seeds. So that's why I am telling them they are occupying a midway in the plant kingdom. Now here their entire property is only leaves. What you can see, nothing but leaves. So they are full of leaves. Their roots, rhizome, etc. are under the soil, and they are very, they do not grow into trees. They are small plants, herbaceous plants, and their reproduction, their photosynthesis, both are done by leaves. In angiosperms, you can see the reproduction is separated into flowers, fruits and seeds, but here leaves are doing the photosynthesis as well as they are doing the reproduction. So they do not have seeds, they have spores. The spores are found under the leaf, mostly under the leaves. And spores are produced in box-like structures called sporangia, and the sporangia are grouped into sori under the leaves. So that is their identification. We are doing the identification mainly based on that, because for not having any other structures like flowers, petals, stamens, we have to depend on their distribution of sporangia into sori on the underside of leaves. And they are generally found in the moist, shady places. So they require, because they are the first land plants, till that time actually algae, they are all aquatic plants. Very first group of plants, algae, they are aquatic plants. Gradually the bryophytes came, which require water. They are in the muddy soil with full of moisture. They are growing, but for the first time, the pteridophytes invaded the land. So we have to say that, we have to tell them as first terrestrial plants. And that's why we can see them in shaded conditions, and because they are just entered into the land, that's why they still require water for their reproduction and all for their survival. And they also prefer shade. So tropical rainforests are their best home. Where are they found? As I already told you, they are found mostly in the forests, thick forests, rainforests. And when you enter into a forest, definitely they will attract our eyes. We can immediately see them everywhere. on the ground, on the tops of trees as epiphytes. So it will catch our eyes when you enter any thick forest. So as you know, the Western Ghats are the best place to find them. Apart from that, natural forests, they are also occurring in our surroundings. We can see them in the crevices of the compound walls, on the inner wall of the wells, on the roadsides, in earth cuttings, on the railway track on either sides. When you move in the bus on the two sides of the road, and sewage along the sewage water, like that various places you can see them. So their identification is very simple. They will never produce any flowers or fruits or seeds, so you can simply identify them by seeing their leaves, and turn the leaf backside, you can see the distribution of their sporangial clusters in a variety of ways. Another interesting thing about the this group is they have got two morphological conditions. Dimorphic, you can say. The plant exists in two ways. One is called sporophyte and is called gametophyte, which are independent plants. They exist, then only the life cycle is completed. So both are independent, both are photosynthetic, both are reproducing, but one is reproducing by asexual method, by spores, whereas the other one is reproducing by sexual method. The main plant is a sporophyte. It is asexual plant. It disperses the spores, and by that way it is reproducing. The spores which are falling in the ground will develop into a miniature plant, as good as say microscopic, underneath the soil, which we cannot locate easily. So 1 or 2 millimeter size, heart shaped, but photosynthetic, without root stem leaf. Even they don't have root stem leaf. Real root stem leaf are not there, but rhizoids are there, and such a gametophyte is there. So it has got sex organs. So that will be reproducing sexually and forming zygote. That zygote will develop into a sporophyte. So that is one of the most interesting aspect of pteridophytes, having two generations alternating each other to complete the life cycle. Next. Now these pteridophytes, once upon a time, millions of years ago, existed in large numbers. Not only large numbers, they are also grew to the size of giant trees, 30-40 meters height. Such thick forests existed in the past, say 400 million years ago, and they ruled this earth for 60 million years. 60 million years they were dominating on this earth, without the chirping of birds, roaming of vertebrates, or without, let us say, no humans at all possible. So in such a condition, they were ruling the forefathers of present day pteridophytes were like that. And next slide. So that is the view. The forerunners of pteridophytes were like this. They are forming thick forests, and which totally disappeared, not by human intervention, not by pollution, it is by natural calamities and catastrophes. This completely, they vanished from this earth and totally replaced by the same group pteridophyte with the herbaceous small plants having a vast distribution, but lesser in number. Their number is very much limited when you compare to other groups of plants. Next. So these two are that old forest, which still serve us in the form of coal and petroleum. They continuously serve our human beings even today, which we unearth in the form of coal and petroleum products. So which have such a condition of pteridophytes is not existing today. Next slide. Now the present day pteridophytes, as already told you, is mainly, most of them are made up of herbaceous plants. Next slide. So this is the scene today. That was the scene millions of years ago during Carboniferous era. Now when you enter a forest, this may be some Agumbe forest, when you just enter, the pteridophytes growing on the land, growing on the trees, hanging from the trees will attract you, will catch your eyes, and inside the forest, along the streams, they are found in large numbers, and that is the present day condition of next. This is another invasive pteridophyte which occupies the tops of hills of the forest in cleared areas. You can again see a full growth monoculturing type of natural growth, which have invaded the cleared land in the forest. So you can see that what the changes happened in the life of pteridophytes. Next. Now, as I already told you, they grow in the damp situations in the forest as undergrowth under the canopy of evergreen trees. They are also they are called terrestrial pteridophytes, and most of the pteridophytes are terrestrial. Next comes, in majority point of view, if you go to the second level, the epiphytes. That is another group of pteridophytes, depending on the habitat, which are growing on trees, hanging from the branches of trees, and some of them are extremely delicate and intricate plants. They can be sighted inhabiting. Apart from that, as I already told you, in large cities also you can locate them in the crevices of buildings, on the roadsides, along the sewage water, and such other places. However, we have to say that the greatest diversity is in the tropical forests. So naturally, we have to come down to Western Ghats and Northeast Himalayas. Northeast Himalayas and Western Ghats are the great repositories of our pteridophytes. Next. I already commented on this. Very curious aspect is there occurring of two different stages, totally independent but circulating to complete the life cycle. So it becomes very delicate in their protection. This point comes on their preservation. We have to see both sides. One requiring water for reproduction, for carrying sperms. Another, I said, adult plant. So both must be compensated. So that has the problem in their conservation. So that is the most curious aspect of pteridophyte life. Next. So this is the life cycle of pteridophyte. I can say it's a fern. Ferns are advanced than their allies. So you can see on two sides, this is the adult plant or the sporophyte. So that is the alternating gametophytes. See size difference, functional difference, appearance difference, functional difference is there. Still they have to circulate so that life history is completed. So this is the reproducing by spores produced in sporangium. Sporangium is in groups called Scier, Sorus, Scier plural, and the spore produced will develop into a gametophyte, a small thalloid heart-shaped miniature structure having sex organs, Archegonia antheridia, fertilization, zygote, and again you get a new plant. Next. Now, pteridophytes in India, if you come to the Indian point of view, as I just commented, Northeast Himalayas and Western Ghats are the rich repositories of the pteridophytes in India. The Western Ghats, as you know, which spreads over 6 states, starting from Gujarat, traversing Goa, Maharashtra, Karnataka, Kerala, Tamil Nadu. These 6 states, this long strip of Western Ghats spreads a length of 1,600 kilometers long, and the major 60 part of that is in our state, Karnataka, and the remaining 40% of Western Ghats in other states, other 5 states. So naturally we expect more pteridophytes in our place, but it's not like that. The Kerala and Tamil Nadu having more pteridophytes than our state. The reason is very simple: more rainfall there and higher elevated mountains. So it requires cool weather. They require cool weather and more rainfall. That's why our state stands next to Tamil Nadu, and even though we are having a long area of Western Ghats in our state. Next. Just to see the comparison of flowering plants and pteridophytes, the total angiosperms in the world is 3,54,000. Rough, we cannot make a 100% correct number. It is impossible in the plant and animal kingdom. It is ever changing. However, we can say that it is around 3.5 lakh, but the pteridophytes are 13,000 in the world. Nearly 30 times more angiosperms. So 30 times less the pteridophytes. Coming to our country, we have got 13,000 angiosperms and 1,100 pteridophytes, 10 times less. So that is the magnitude of pteridophytes. Next, what are their habitats? They are going in a variety of habitats. The places where they grow are terrestrials, as I told you, most of them are terrestrials. Many of them are epiphytes. Epiphytes are pteridophytes which are attached to other trees in the crevices of other trees and hanging from there. Of course, they are not sucking food. They are just for attachment. They are there, and they are hanging from the trees of the forest. They are called epiphytes. Next place comes to them. They are lithophytes, which are attached to rocks in the running. Naturally, we are having so many streams inside the forest. There will be rocks. In the crevices of rocks, you can see a variety of pteridophytes. They are called lithophytes. So that will be the main group. Then we have got, of course, climbers. Few pteridophytes are showing climbing habit also, and very few are hydrophytes, which are growing in water. No pteridophyte grows in the ocean. Marine pteridophyte is not there, but they are found in the lakes and rivers. So they can be called as hydrophytes. Some are hygrophytes in the marshy places. Also, you can see them, and very few are halophytes. Halophytes means not in marine water, but in the backwaters. So there is one very interesting pteridophytes there, which are plenty in our coast, in the backwaters of our sea coast. And there are some pteridophytes which are very filmy. They are called filmy ferns, Hymenophyllum, Trichomonas. There are two groups are there, two genera are there, and they are again, conservation is a problem about them because of their very delicate nature of leaves, and also tree ferns, just to see their relation with the old fern, pteridophyte history. We have got a few hundred, maybe 100, 200 species of tree ferns, which are, of course, they are not that tall, some 30, 40 feet at the maximum. So these are the different variety of habitats they are seen. Next. I have just given some common pteridophytes in our place based on our work of the Western Ghats. So I have given a long list. You can just move on. Lygodium, Hymenophytes, Ophioglossum, Selaginella, Ediantum, Etherium, Cristella, Lindsaya, Teres, Viteria, Isoetis, Lycopodiella, like that. Like this, we have got 181 species were reported by us. So I have given some important members I have listed here. Tree fern. So these two ferns are very interesting in our vegetation. Cyathea gigantea and Cyathea nilagariensis. I think both of them are endemic. You find only in India. Especially nilagariensis is endemic species. So it is very interesting to see these two tree ferns. Next. So what I listed earlier were the common pteridophytes in the coastal belt. If you enter to the Ghat forest, higher elevations, it will go on changing. Distribution, the members, their density, everything will change as you go up and up in the higher elevation. So you get a different list of pteridophytes in the Ghat section. Next. So many of them are epiphytes, some of them are terrestrials, and so on. Next. So next, some few slides I will show you just to show the beauty of pteridophytes, how attractive and how eye-catching these plants are, which are, even though they are neglected. So once you enter into that field, you will find excitement in their collection and in their study. So this is a fernelli, not fernelli, but pteridophyte. It is called Lycopodiella ceranua. Next. This is a huge climbing fern. It is called Stenoclina palustris, plenty in the Yana forest. Next. This is Lygodium flexuosum, very common in the seacoast around our residential areas. You can see this again climbing. Next. This is another extensive growing fern everywhere, especially on the earth cuttings. So those who have traveled in the railway line must have certainly seen this as huge thickets on either sides and also on the roadsides. It's a very plastic type leaves, very ideal for decoration during any function. They will last for a number of days, they will not wither. So such a highly dramatic plant. Next. This is Ophioglossum reticulatum, a primitive pteridophyte. You can see a long cones here, so called they do not have the reproductive part is separated from the leaf, is a separate part. Yes, next. This is Isoetes udupiensis, first reported from Udupi and named after the place Udupi. So Isoetes udupiensis reported in the year 2005 and it was published in the taxon, the journal taxon. Next. This is again a common plant, Selaginella delicatula. You commonly see them on the shade in the arecanut gardens. Next. Walking fern, again common on the around our houses on the compound walls. It is called walking fern, Adiantum. Next. This another species of that, Adiantum latifolium. Next. This also very common locally. Need not go to the forest. You can find in the coastal belt. Cheilanthus. Now it is called Cheilosoria. So every time you can see the. So when we say pteridophyte, you have to see immediately you have to see the backside to see the arrangement of sori. So that is their trademark. So wherever I show the photographs, I always show the backside of these ferns because as they do not have any flowers or fruits. So next, Lindsia, another extremely common. Next, terrestrial, Tiris, a number of Tiris species are available locally, and see the sori on the margins of the lobes of the leaf. Tiris. Next, see how beautifully the sori in large numbers on either side. Tiris vitata. This is a very good plant for bioremediation of cleaning the wastewater, and it is generally found growing on the sewage water, alongside sewage water. Next, Blechnum, another common plant on the river sides. Next, Tiris, Pterogramma. This is silver fern. Backside you can see powdery white mass along with the sori. Pterogramma calomelanus. Next, already seen. Next, see how beautifully the sori are arranged on the backside of the leaf in Angiopteris. It is a huge coming close to the tree fern, but not tree fern. It is having very huge leaves, and on the underside of the leaflets you can see such a beautiful arrangement of sori in 2 rows along the narrow branches. Next, another very common epiphyte, Drainaria quercifolia, bird nest fern it is called. See the nesting of the leaves to collect the water and debris so that it can survive in the top of trees, because it has no connection with the soil. It has to make arrangements to collect debris and water so that its roots can absorb those things and it can survive. See the sori distribution on the, so variety of distribution of sori you can see if you go on seeing these different ferns. Next, this is another epiphyte, Viteria, very grass-like leaves, appears like a grass, may be mistaken for a grass until it produces the sori. Next, see Pyrosia, see the arrangement of sori, how beautifully artistically it is arranged. Next, Cyclosaurus, another type of sori arrangement. Next, Hemeritis. This also, if you go to Hebride in the foots of hills, in the foothills, we can find this, the very webbed arrangement of sori. Next, this is found in water, Ceratopteris electroides, highly branched leaves. Next, this is purely aquatic, Marsilia, used as food, and this also very difficult to believe it is a fern, but here the sori are on separate small seed-like structure, so it is called Marsilia, found in the paddy fields and other water bodies. Next, another aquatic fern, Salvinia, which is an invasive plant which invades, actually if you introduce into a place, in 1 or 2 years it will occupy the entire pond or river, pond or lake. So Salvinia. Next, another aquatic fern, I told you very few, so 3 we are having locally, all the 3 which I am showing now, they are available here. So this Azolla is also very common, very small fern with small leaves and economically very important. It is having a association of a bacteria, cyanobacteria in the leaves, which helps in nitrogen fixation. So it is purposefully added to the paddy fields to get the crop, better crop. So it does have a ecological importance. Next. This is what I was telling you, the Acrosticum, the only fern that we find in the brackish water, huge fern, and see the backwaters along which it is growing so profusely. So it is called Acrosticum. Next. Osmunda, another fern which is growing near streams. Next. Adiantum, another species. Next. So that is found only in the higher elevations of forests. Cyclosaurus, this is also restricted to deep forests. Next. Macrothelopteris, again a forest pteridophyte. Next. Next. Continue. Nephrolepis, a decorative fern. It is found in the forests also, so they have introduced into the gardening. And in many gardens, in pots, you must, very common in the pots in most of the gardens, and all of you must have seen this, but you may not be knowing that it is a fern. Next. Tectaria, a terrestrial fern, sometimes occupying the entire forest floor if you go to the deeper forests. Next. Another species of Tectaria, this plenty of sori on the underside. Just imagine how many number of spores they can produce and how many gametophytes will come, but all may not survive, of course. Next. This Asplenium, we have got many species of this, most of them are epiphytes. Next. Ampelopteris, another plant which is used as food by the people in the who are living in the countrysides, an edible pteridophyte. Next. Sphenomeris, again growing luxuriantly in the forest. Next. A tree fern called Sciatia gigantea, which I was referring earlier. Next. Next. Can move faster. Himata. Next. Pteridium. So this already I showed this scene. So its backside is here. We can see the sori arrangement. Yes. Next. See how large the sori are and completely occupy the undersurface, as you see in the Rheopteris cochleata. Again restricted to thick forests. Next. Bulbitis, along the streams of rivers inside the forest. See the sori arrangement on the leaves on the underside. Next. Next. Continue. This is the interesting fern, the filmy ferns. They have got highly specific. Their specificity is so great that some species they can grow only in one tree, one particular tree they can grow. So such a specificity is there. So especially such ferns, it is very difficult to conserve, because if that tree is lost, this fern is also lost. And they are very sensitive to the climatic conditions. They have got a microclimate. Apart from the general climate, they have got their own microclimate. So if there is any disturbance there, they cannot continue their survival. Next. Hymenophyllum, this same thing. Next. So this is a fernelli called Plagimiururus philanthum. Formerly it was called by another name. So see the reproductive structures separately at the tips of the branches. Next. Epiphyte. Next. Anthropyrum plantigenium, very beautiful fern, an epiphyte. Next. Next. See, Oleandra, another extremely beautiful epiphytic fern in our forests. So, but becoming rare and rare. When I visited some 20 years ago and my recent visit, I can see a lot of changes in the distribution. So, telling that they are in the verge of extinction. Next. Another one, beautiful fern, Asplenium nidus. See the sore. Next. Already focused. Microsorum embryonaceum, thin leaves, the sore on the other side. Epiphyte. Next. Next. Again epiphytes. All these are epiphytes. See how beautifully the sore are arranged. Next. Loxograme. See, just like Nama, Devara Nama, you can see so many lines on the other side. I don't know what made them to have such beautiful markings on their leaves. Purpose is same, production of spores, but occurring in a number of ways. Next. Next. Another interesting plant, Equisetum, a fernelli, not fern, fernelli. Again, having a very interesting botany students, they take the study of this, very interesting anatomy and reproduction. Days together, my teacher, Professor Ishwaran, was teaching, and 10 hours he took to complete the study of this. 10 hours he was describing the hymenophyllum in the class. Just imagine. And also the practicals also, to take sections of that, see under a microscope, its anatomy is a very interesting study. Silotum, believed to be very primitive pteridophyte originally, but now they say it is advanced. It is a interesting pteridophyte, and thing is that they do not have roots, they got rhizoids, no leaves, only stem. Stem is everything. Stem is green, and and sporangia, three trilobed sporangia you can see. Just a green stem does photosynthesis. So root-like structures are there, but they don't have vasculature. That's why they are called rhizoids. So no leaves at all. Small innations we get. Anyhow, it is one of the very interesting plant. And formerly we had in our campus only, but now I find it very difficult to trace it. Once upon a time in my house also, I located one plant, disappeared gradually. Anyhow, this is one pteridophyte which is a xerophyte. Xerophytes are very rare. Xerophyte means a plant which grows in the dry places, extremely dry places. This is found on the rocks, and it is not found in the forest. It is found in the rocks of Chitradurga we have collected. And see beautiful fan-shaped leaves. And underside narrow sore are there. Actinopteris radiata it is called. Another interesting xerophytic. I think only 2 xerophytic pteridophytes we could collect, and one among them is this. So just to say that even xerophytic representatives are also there. Next. So with this I have tried to give you an overview, a glimpse of the pteridophyte biodiversity. However, the biodiversity studies gaining importance in the recent years due to rapid depletion of species all over the world. So what is happening to other groups of organisms is also happening to pteridophytes also. We are losing many species before they are identified and documented. Very important. Many species are vanishing from the face of this earth before they are located, identified and named. So that is the tragedy of the thing, because workers are less. That too in pteridophyte, to do work people are very, very less. So the problem is with the pteridophytes is that. People say that 2/3 of world population is said to be in danger, extinction of so many times we are listening this, deforestation, habitat loss, destructive developmental activities, agricultural expansion, global warming and climatic changes. So these are working on pteridophytes also. So that arises the problem of their preservation. Why at all pteridophytes become important? See, considerable number of species grown as ornamental plants. They have got a very good place in the gardens, but they are less exploited. So more and more people should go to grow these plants which are very attractive. However, already we are having certain plants growing in the, of course, they are almost repetition type, Nephrolepis, Selaginella, and same type of plants are being grown, but we must go for some other plants also to introduce, especially those which are in danger. They can be brought and grown in gardens so that their lineage is survived, and also we can enjoy their beauty. Anyhow, there are many plants which are grown as ornamentals, and number of them are used as food plants. Tribal people, especially tribal people. I don't find any plants sold in the market. I think in some countries they say some plants are being sold in the market, but I didn't see in India any pteridophyte plants sold in the market. Anyhow, they are extensively used by tribal people, and many serve as medicinal plants. Various ailments, lot of work is going on, and already, especially by tribal people, from simple headache to cancer. They have tried various pteridophyte plants for treatment, and nearly 60 plants of our area, Western Ghats, have been identified as having medicinal properties. And even their extraction work is also going on in some places, apart from simply documenting from the interview with the tribal people. A role in bioremediation of wastewater, Salvinia, Teres, these are the two best examples. They can extract the very poisonous elements, arsenic and such other things which are found in the forest. They are able to remove them and accumulate them. So that's called bioremediation. In that they have got their role. And biofertilizer, I already told you, nitrogen fixation by the cyanobacteria which survive in the leaves of azolla. Useful in soil building and soil conservation also, they have got their role. Next. So now the concern is such useful plants and which can be exploited further are in the process of declination, and goes nothing without saying it. We have to say that we have to preserve the tropical rainforests. So because their diversity is there, and rapid disappearance of this biome throughout the world with many species yet undiscovered is of great concern. Next. Why should we, question comes, when you say if they are weeds, why should you conserve them? There are some reasons. Biodiversity. Pteridophytes represent significant component of Earth's biodiversity. Conserving them ensures the preservation of genetic diversity, which can be vital for ecosystem resilience and adaptation to environmental changes. They play a crucial role maintaining biodiversity within ecosystems by providing habitat and food for various organisms, insects, birds, and mammals. They have got many ecosystem functioning like nutrient cycling, soil stabilization, and water retention also, and filtering the pollutants by absorbing them. They got medicinal and economic uses. I already told you they are used in various treating various ailments, respiratory problems, skin diseases. I told you even up to cancer they are suggesting, but authenticity must be open to for discussion. Additionally, ferns hold cultural significance of folklore, art, traditional practices in many societies. Species also have economic value as ornamental plants, which I have just seen. Another important thing is they work as indicator species. If that fern is, a particular fern is there, we can say whether the climate is in a good condition or not. So because they are so sensitive to climatic changes, so they also work as next indicators. Then of course in the research and education also they are important. In the scientific research of various fields including ecology, evolution, genetics, understanding their biology and ecology can provide insights into broader ecological processes to help them conservation strategies. Additionally, they are valuable education resources teaching students about plant diversity, evolution, ecosystems. Aesthetic value and recreational value to be grown in the gardens, maintaining fern gardens, ferneries, and appealing to our eyes. Habitat restoration. Ferns often grow in areas where high moisture levels such as rainforests, wet lands and riparian zones. Conserving ferns means protecting the habitats which are critical for the survival of numerous other plant and animal species. So to build up a ecological system, to restore the ecological system in wetlands, their role is always there. So restoring fern population can also aid the rehabilitation of degraded ecosystems. Next. Climatic change resilience. Ferns have adapted to diverse environmental conditions over millions of years, making them resilient to certain environmental stresses. Studying ferns and their response to climate change can provide valuable insights into developing strategies for mitigating its effects of ecosystem. So a lot of work is in front of us so that the upcoming generation can take these topics and to preserve the genetic diversities. Above all, pteridophytes represent ancient lineage of plants. The old lineage surviving today in another appearance, and they can be valuable indicators of environmental health. Next. I need not tell about this, already we have discussed. Why the same thing, whatever is harming angiosperms or animals is also harming pteridophytes. So it's a long list of disturbances. Again and again, global warming, climate change, pollution, habitat loss, increasing urbanization, industrialization, encroachment of forest land, expansion of agriculture, all intertwined and interlinked things, ultimately harming the growth of pteridophytes as it is for other plants also. Mining activities, unplanned developmental activities, fragmentation by roads construction, railway constructions, unplanned felling of trees, overexploitation of resources. Next. Same thing I have elaborated here. Habitat loss, that is the main and important reason. Climate change by global warming, invasive species. So certain species, when they enter, they just destroy the previous natural one and they invade over that. Actually, Bracken fern is like that. It is actually invasive. So once the forests are cleared in any part of the forest, they just spread like anything. So they are also harming the growth of natural vegetation. Pollution, overexploitation, commercial collection. See, some tree ferns which are in the verge of extinction, they are collecting for, their trunks are collected for growing orchids. So actually these trees are in the verge of extinction, but invariably people are destroying them. Next. Now, if you compare the pteridophyte vegetation and the larger angiospermic vegetation, the pteridophytes have got special qualities which make them more sensitive to environmental change than the angiosperms. I have found out, listed here 4 things. One is specialized habitat requirement of pteridophytes. So pteridophytes are found in a very specialized conditions, like they require humid condition, they require shade, and they require shade of tree. If a tree is cut, underneath they grow. Tree is cut, they are exposed. So naturally they lose their habitat. Or if a tree is cut, along with the tree, so many epiphytes which are growing there, they will disappear. So like that, they have got a specialized requirement in their growth and in their nature. Delicate leaves, they are very sensitive. Delicate leaves are more vulnerable. Their leaves are more delicate than the angiosperms, so they are more vulnerable to climatic conditions. Then most pteridophytes are dependent upon microclimatic conditions. The reason for the successful survival of pteridophytes, any kind of disturbance in these microclimatic conditions can hinder the growth and evolutionary process occurring naturally in these plants, thereby leading to decline in their populations. So if there is a entire forest will be having one climate, if you enter into the forest, here and there you will be having certain microclimatic conditions, the slope and where exactly they are attached, and such small things which are considered as microclimatic conditions, they are very sensitive to that. One tree, many epiphytes. If one tree is cut, so many epiphytes hanging on them will disappear naturally, and their number is decreased, and certain which are specifically found only in that tree, they will be lost forever. So one tree, many epiphytes will be lost in the long run. Then another thing is, as I already just commented earlier, their dual generation life cycle is another important difference. they have to balance the two conditions having different requirements. So from that point of view also their conservation is a little tricky. So these are the 4 things which are special to pteridophytes. Next, how to conserve them. Conservation, as already you know, is in two ways, in situ, ex situ. Conserving where they are found and conserving outside they are found. So if you preserve the place where they are growing, it is in situ. If they preserve them outside their natural habitat, that is called ex situ. So of all the strategies, in situ is the best method to conserve the forest and less disturbance. As already we are saying, sustainable. We have to exploit the forest to the minimum and leave the place for their survival. So that is the best way of all the types. That is the best way. So some places can be declared as wherever you find extremely rare ferns, the bioreserve, fern bioreserve, something like that one can declare. Especially for pteridophytes, the most important problem is that laxity or paucity or dearth of taxonomies. As a whole, taxonomies have become very rare species. Generally we say like that. That too, taxonomy of pteridophytes is still lacking. So my appeal is to the younger generation, the students, to opt for basic science, that too for botany, that to take up research, that to become taxonomist. So that we can implement some of the thoughts to do this need of to attend to these problems of conservation. So more people should come up and take basic science. Botany is optional. Study the basics, morphology, taxonomy of pteridophytes, master the technique of identifying ferns and fernalize. So in the beginning it is somewhat difficult, but gradually you catch it up. To do the survey work of pteridophytes in their natural habitats, assess endemic species, find out rare and endangered species. So these two are very important because priority should be given to these two, to preserve the endemic species and to preserve the endangered species. Looks very simple, but very difficult to decide which are endemic, which are endangered. If you go to literature, we will see that lot of papers modifying this list. Some people telling it's endemic, next paper telling it's not endemic. Some people identifying some which are endangered, other people say it is not endangered. So if you go to so it's a very heavy task. You have to go to the field, find out their number, identify them, find out the distribution, go to literature, go to the different herbaria, find out how many are there in the herbaria, how many have already worked. So a lot of work in the field as well as in the lab. So these two are the great problems. So I have just highlighted some slides. We can go fast. Next. Endemic pteridophytes. Here are some papers. From time to time they are published, and 1984, 41 species in the list of endemic in India with reference to India. 1984, another paper on 214 species of pteridophytes as identified as endemic. Then Manikam and Irudurraj, 1992, telling 16 endemics to South India, 17 endemic to South India, like that. Next. Chandra and others, 234 endemic plants pteridophyte in India. But lastly, 2008, Fraser Jenkins, he has removed many of the earlier endemic species of pteridophytes as pseudo-endemics, which arise mainly due to mistaken synonyms of the lack of understanding of the range of species. He has listed only 47 species of Indian pteridophytes as endemics. So just to tell you that the problem is great to identify and to preserve them. Next. I have listed here the last paper according to that 47 species which are endemic to pteridophytes to India. 47 are final, latest work. Next. Next, same problem, same applies to threatened pteridophytes. The IUCN, International Union of Conservation of Nature and Natural Resources, that has listing plants into 7 categories. Again, appears to be simple, but very difficult task to make a plant, whether it is, of course, extinct we can say. Extinct in the wild also easy, but other critically endangered, vulnerable, near threatened, least concern, data deficient, that also we can see. So the middle 3, 4, 5 is very difficult to tell. So this is the standard list of making plants under different categories to give importance for their conservation. Next. World Conservation Monitoring has identified 1,650 threatened species of pteridophytes, 20 may be already extinct, 67 endangered, 91 vulnerable, 354 rare, and 1,318 likely to be in the categories, and when more information will be available. Next. So I told you that is very difficult to do in that way. So Indian condition, only advanced countries can do, but Indian condition, Chandra and et al. And others, they have made it a little simple, and they make 4 categories: at risk, critical endangered, near threatened, rare. So in India, 219 at risk, critical endangered 160 species, near threatened 82, rare 113. Next. So that is the list of endangered pteridophytes from Western Ghats, 17 here. So many are, I have shown in this list, Amorphis proliferis in Western Ghats, Helminthostachys we are having, Huberzia hamiltoni we are having. Next. Back. This also near Subramania we have collected this. That's also in endangered. Next. Here also we have got many which are available in our place. Trichoman, sorry. Yeah. North Indian ones. Next. List of endangered taxa in East Himalayas. Next. So 38 plants are listed here in the north back. East Himalayas, these are the plants which are endangered. Next. Now conservation strategies. I did not tell much about this. Already we have discussed in the various previous presentations. Establishing updated data of first work. You have to update the funds of Karnataka if you are focusing on conservation of Karnataka. And conducting survey of areas under threat to assess how many species are in the process of extinction. Organizing, supporting seminars, awareness programs like this. This is one of that. We are undergoing that third, this one now. Multiplying rare species by conventional method, tissue culture method, micropropagation, this is one of the methods. Medicinal aspects of ferns to be further explored. Next. So, of all the methods, this is the best method, in situ conservation. Next is restoration of degraded habitats. Next. This is growing them in gardens, ferneries, fernhouses. So, in this line, our work is not sufficient. We are doing only same plants being grown in the gardens. So, we have to give place to rare plants to collect from the gardens, from the forest, and introduce them into the gardens, so that at least here they will propagate, and later we can also reintroduce them, restore them in the forest. So, botanical gardens, arboretia, nurseries, they have to give importance to this. Next. Micropropagation of pteridophytes. Now recently many institutions, India here and there, they are working on the micropropagation of pteridophytes also with the successful rate. Next. Apart from that, education and awareness of the general public, policy makers, land managers, educate people about the ecological role of ferns and allies, and collaboration and partnership, legal protection, approaching government and making legal declarations, and genetic conservation. Next. Some institutions which are doing some work, but they have to take more initiation. So, Indian Council of Agriculture Research, they can do tissue culturing. Botanical Survey of India, BSI, I think already they are working on some. They have organized a workshop for interested people to work on the micropropagation of ferns. Indian Council of Forestry Research and Education. Next, Indian Institute of Science, Universities of Agriculture institutions, private research and biotechnology campuses. Aloysius College, they are doing. Smita Hegde, she is one among our nearest institution in our place, working on micropropagation of pteridophytes. Next, institutions that take up establishment of ferneries, Indian Fern Society, Botanical Survey of India, Indian Council of Forestry Research and Education. Next, botanical gardens, universities and research institutions. So this can take up establishment of ferneries. Next. So for ex situ conservation, special efforts are not given much to the ecologists of ferns when compared to the flowering plants. So they have not given importance to ferns as they give to angiosperms. Majority of gardens, there are very few ferns which are grown mostly as ornamental ferns and not as rare and endangered ferns. In India, there are very few ferneries to conserve the pteridophytes. Very few ferneries are there if you find out in our country. Next, some photographs. This is one from Germany. When I visited Germany, I had been to this botanic garden. Next, entrance of that. So here you can see a long glass house, say nearly a kilometer long. They have created this is a temperate country, tropical rainforest. They have created a tropical rainforest inside that, full of high trees, tall trees, having so many pteridophytes hanging and so many pteridophytes in the floor, and even they have grown tree ferns inside that fern house, glass house. Next, so this is the tree growing inside that, one of the tree fern growing inside that. Next, it's an open fernery, maybe a photograph from internet, maybe in England or so, I don't know exactly. All advanced countries are having such open ferneries also. Next, a closer view of a fernery. I think this from Kew Garden, I think. Next, it's also a fernery full of ferns growing epiphytes and terrestrials. Next, this fernery recently opened 2001, I think, in India, India's biggest open air fernery in Ranniket, Uttarakhand, recently opened fernery. Next, inner view of that open air fernery. Another view of the same place. See how thickly they are growing in that. Most of them are ferns. Next, this fernery in Malabar Botanic Garden, Kozhikode, where Dr. Madhusudanan was the director of that. He was the South Indian pteridologist. He was managing that garden. Lastly, in the midst of our modern world's rush of development, present-day pteridophytes stand as silent sentinels, reminding us of our ancient connections to Earth's history. Their resilience and adaptability speak volumes about the intricate balance of our ecosystems. Let us cherish these botanical marvels, for they not only adorn our landscapes, but also serve as living relics, bridging the past and present, and inspiring us to nurture the delicate web of life that sustains us all. Let us safeguarding biodiversity, and it is everyone's responsibility. Thank you.
Any questions? Thank you, sir, for being here with us today and for your wonderful talk on the conservation of pteridophyte. Now, I would like to request Principal Lakshmi Narayan Karan, sir, to present the token of gratitude to Dr. P.K. Rajgopal, sir. Thank you, sir, and thank you all for your presence. Thank you.
chairpersons to occupy their seat on the dais.
Presentation session 2. Now today we have our first presenter of today's session, Dr. Ashoka CH, Associate Professor, Department of Zoology, Dr. P. Dayanand Pai, P. Satesh Pai, Government First Grade College, Mangalore. He is going to present the paper on the title, Environmental Impact Assessment, the Need of the Hour. Each presentation carries 6 plus 1 minute, 7 minutes, and 3 more minutes for the discussion round.
ಹಲೋ ಎಲ್ರಿಗೂ ನಮಸ್ಕಾರ. Next. The earth has become very sick planet and urgently needs a cure. A disaster is looming around. Unless otherwise checked, the whole planet would become uninhabitable. These are the words of K.V. Narayan. So, I have taken an analogy of these wordings here because suffering with some diseases, like you think about human beings, cancer. Now the treatment is all under the trial, so we have to go for the modality where at least some way that can be cured. So such being the case of the disease what the earth suffers, what can be the medicinal modality? At least we could alleviate the disastrous situations what we ourselves have created, anthropogenic sources which have created a lot of havoc on this earth. So there is some hope of applying environmental impact assessment, a way through a policy or legitimate aspect of regulating the burden on earth's surface. Next. EIA is a planning tool to integrate environmental concerns into the developmental processes from the initial stage of planning. So how it has developed, glancing through the history, so 1970 you can see, 1980, 1990. So in India we have the legislation started in the year of 1994. Then it went on getting amended in 2002, 2003, like that. Later we got it an amendment in the year of 2006, and now one draft is given to us in the year of 2020. Next. Next. There are so many objectives of EIA. One of that I would like to mention, because we are facing a lot of these disasters. So once the disasters get manifested, then we have to recreate the situations. Then developmental processes are mandatory. Then how do we can manage it? So such being the case, that point is just taken here. This may be used for developmental planning. And why is the decision making at various stages of disaster risk management? Next. We do have a lot of constraints or obligations to apply it. One being, as I am addressing to the student community, we do not have the awareness. So the most significant of which are related to the lack of public awareness of EIAs. Next. Next. So this is about that draft EIA notification 2020, which is under the process. If it comes, it is thought that it will be favoring the industry. So there are so many things which are removed. It may not help us to protect our environment. So some of the things what I have observed from that draft is reduced time for public hearings. 1 year it is reduced to 6 months, like that. Exemption of projects, post clearance compliance, annual submission of reports, report preparation solely by project proponents. So whatever they prepare and give, submit, that we are doing all the remedial measures to the environment, taking care of the impacts, minimized or mitigated, they will write and submit. No public reporting for non-compliance, post facto clearance. They will start maybe after 2 years. If it is more profitable industry, then due to some obligations that industry may be asked to run continuously, and penalties of firms. So that like these things are there in that draft. Next. And if you see, they will give the clearance, draft will be prepared, there are agencies with whom they will be doing it. So different type of projects will be under the observation. Next. Next. So in a sorry, previous one. So this is about in 1 year. So January to December 2021, environmental clearances have been granted to 527. So each year that many number will be given anyhow. Next. Next. So you see 2017 to 2021, if you compare, almost it is in the parallel line. So there is no reduction at all, it is going to improve. Permission at last will be sanctioned. Environmental clearances will be given. This one incident I would like to highlight here. This is in Bangalore, Puttenhalli Lake, that has been referred as a reserve, bird reserve. There is near to that a power station, which you could see there in the picture. Next. So this was reported, and a group of people, I was one among them, and we through the legal aspect that was brought to the court, and that is that Puttenhalli Lake and Bird Conservation Trust, they objected for the continued work on that. Next. But see, 1 week back, there is a news that that power station is going to work. So whatever the team had started using the environmental impact assessment, finally that will be favored to the industries, and also there will be some way of lobbying. Then this, what we had to have as a tool to protect our environment or mitigate the effects of the more and more invested larger projects, how could we curtail the events from happening? Who could lead? Because legal aspect means people should spend a lot. It becomes a money matter. Who has more money towards their side, the result will come. Next. So I would like to conclude in the given opportunity. EIAs are crucial tools for providing decision makers with an objective assessment and for the development of mitigation strategies aimed at averting projects' negative environmental effects. The general population is now more aware of the potential dangerous consequences of advancements. In the decade of the 2030s, people around the world recognize that a full climate catastrophe is developing. at entrenched bureaucracies. For example, in business, media, education, religion and social services are still largely unprepared and ill-equipped to meet the challenges of worsening climate, deteriorating economies and a collapsing biosphere. Thank you.
Thank you, Dr. Ashok. Any questions from the audience? Any questions?
This is not my field of experience, just by going through the literature surveys I prepared.
I have a question. So, sir, I just wanted to know that we have EI, which is mandatory for the projects. But what happens if the EI, I think in this case, may not have been done adequately? So, what can be done? Like, what can the public do?
Yeah, public can participate by protesting, making jatas. That all we have done. And still, last week also they have done it. And when it comes to the lawyers whom they catch hold of, and some clauses will be there at last, because one clause, a certain area, one clause will give to the above all clauses will not be applicable. So it becomes they will search for such clause. And in this case, what had happened? The permission was granted before that area was announced as a bird reserve. So it happens, they will search for a clause. If it wants to be pro-environmentalist, then they will be giving that reason. If it towards the industries, they will search for that reason. If the more and more students, community become aware of such things, at least nowadays we have all the different ways of communicating. Big way of agitations can at least try to stop it for some time.
Yeah, but I think in many places, despite agitations, if the projects, they keep going on.
No, earlier there were so many sincere leaders who were only thinking about the conservation of the nature. Now, I would like to say that from the student community, such leaders should emerge out.
Also, one more question. So, EI is mandatory for some of the projects, especially
there are categories. They have A,
and
B, in that B1, B2, and some projects which will now it may not depend on the investment. Even the small project mining, it can also have a, depending on the list. There are lists about 27, 29.
Yes. So there are like the B2 category. I think they don't have to do an EI.
Yeah, B2 will not.
Actually, all the
there
projects,
are levels, central and state.
Yes, central and state. So there are some state level projects, for example, where EI is not mandatory. But those projects also create an
state
impact.
level also for the category B1, it is mandatory, but
Yeah,
it will be given by the state only.
Bit by the state, but B2 category projects also will create an impact.
Yeah, impact. That's
so what should be done then?
Then we should have this. I just worried about that discussion around EI.
Yes.
Because we know many things. We are seeing that though it is made that through all the types of vegetations and but bureaucracies again will make those industries to come up. And certain, like one photo I had shown, which they didn't oblige with the rules and regulations, they deviated, and now through the court it has been stopped.
Okay, I think the way is through the court.
Through the court.
Thank you, sir.
Yeah. Thank you, Dr. Ashok, presenting social awareness information. How to design research work on this information?
I'm very sorry that
you are not presented any research work here.
Yeah, no. It is just by going, because first of all, I was made aware of this.
Just, you convey to our audience about, EIA.
Yeah, there are now, you will get in the net all the things, internet, like the reports given by the Ministry of Environment and Forestry. Recent to get all the details, 2021-22 reports. Everything is available in the internet.
Yes.
So
Thank you.
better if you include such things in the syllabus somewhere. We all teach environmental studies for our students, but everything what they learn, that is again repetition of the 10th, 12th and PUCs. So if at all such things are added, probably we also will become thorough with the topic.
Thank you. Thank you, Ashok. Thank you.
The second presentation of today's session is by Mrs. Divya Shri, Assistant Professor, Department of Zoology, Vivekananda College of Arts, Commerce and Science, Puttur. She is going to present the paper on the title, Review on the Diversity and Conservation Status of Freshwater Crabs in Southern India.
Good afternoon all, one and all who are present here. I am Divyashri and I am here to present a topic on review on freshwater diversity as well as conservation status in the southern region of India. Next slide. So coming to this freshwater crabs, it belongs to the Brachyuran suborder, and coming to the distribution, it will be found in freshwater aquatic habitats such as marshlands, swamps, paddy field, tree holes, as well as leaf bases, tropical and subtropical regions. Next. So these freshwater crabs, most of them are nocturnal. They are omnivorous, scavengers, as well as opportunistic feeders, because it is observed that in Agumbe Ghats, coming under Western Ghat, they are feeding on the fishes. So if they have got a chance to feed on some other higher animals, they can opportunistic feeders. Then coming to the freshwater crabs, they are called as freshwater crabs when their life cycle is without larval stage and in the absence of marine water. Then only they are referred as true freshwater crab. But it is not necessary that they should be immersed in the freshwater habitat. So they can also live in the trees as well as the terrestrial habitats. So coming to this paper is mainly focused on review on the diversity as well as conservation of freshwater crabs in southern parts of India. So the papers were published between 2002 to 2022 with 21 papers. Next. So these are the 8 families which are observed under freshwater crabs in the whole world. Next. So in that 8 families, 3 families are observed in Asian region and 2 families, that is Zecarcinicidae and Potamidae, are observed in India, which include 125 species. So in the whole world we can see 6,900 crab species. In that brachyurans are observed 1,564. So in India, so 125 species, that is freshwater crab species, are present. In that 86%, that is 107 species of freshwater crabs are native to India. So that is called as endemic species which are present only in India. So this is one of the species called as Barythelphisa cunicularis, so which was first reported freshwater crabs in the southern India. So in most of our regions we can see this species, that is Barythelphisa cunicularis. Next. So I mainly focused on these 3 states, that is Tamil Nadu, Kerala and Karnataka. Next. So as I told, coming to the southern India, so it is observed that 73 species which belongs to 19 genera under Zecarcinicidae. So coming to one more family, that is Potamidae, which is observed in the northeast regions of India. So this Zecarcinicidae, except northeast region of India, you can see in the other parts. So these are the different genera, 19 genera, and these are about the number of species coming under that genus. Next. So coming to the Zecarcinicidae family, so 75% of the genera, that includes 60% of the species, are reported under Western Ghat regions of Karnataka, then Kerala and Tamil Nadu. So these are about the different number of freshwater crabs, and in the last 2 decades there is a much more discovery of this new species. Next. So these are about the 10 species of freshwater crabs which were commonly observed in Western Ghats, so which will be distributed in Karnataka, Tamil Nadu as well as Kerala, and these are the 10 species coming under 6 genera, so which was recently discovered and added to the freshwater crab diversity of Western Ghats. Next. So 67% or 82% of the species are endemic to Western Ghat regions of Karnataka, Kerala and Tamil Nadu. So when you see about Kerala, so these are the three species, Cylindrothelphisa granulata, longifalis and Kerala species are observed only in the Kerala, they are endemic to there. And coming to Tamil Nadu, these are the three species which are endemic to that particular habitats. Next. So coming to the threats. So when you speak about the threats, the major threat is of the behavior of the freshwater crab itself, because they are showing the high degree of endemism, especially when you speak about the mountain residing crabs, they will be having a limited distribution. So that limited distribution is mainly because of a restricted capacity to distribute. So they will depend upon a small range of environmental conditions as well as low fertility, then predilection of species specific habitats, and as I told, major threat for it is endemism. So along with that, you can see some anthropogenic activities, that is habitat modifications, pollution, forest loss, inappropriate farming methods, that is usage of pesticides or chemicals, chemical fertilizers, so which will become a threat. And one more thing is there is no systematic study that has been taken place in India, and main challenges are they are present in the hidden habitat. So you have to go and search about that species, and taxonomical difficulty to identify that freshwater crab, because while studying I have collected so many species, but along with the geography there is much difference in the color. So even though there is a difference in color, they belong to the same species. So that will be the one of the difficult. So IUCN has been categorized around 53 species of Western Ghat under different categories. So there is 22 species are categorized as data deficient, so there is no information about that one, and 31 species are not evaluated for conservation status, and 7 species for least concerned, and only 2 species, that is Oseothelphisa 2 species are classified as vulnerable species. Next. So coming to the conservation efforts. So these are some ideas. So declaration of the some more protected areas, because these freshwater crabs depends upon the microhabitats. Then preservation of sufficient amount of natural forest as their natural habitat. Then upkeeping the water bodies in a good condition without pollution or without contamination. Then minimum use of pesticides in the agriculture land, and water body conservation. Next. So these are the 2 species I want to mention here, so which was recently discovered in the year 2022 and 23, that is Gatiana Duyarna and Vela Bandhavya. So both are discovered in Western Ghat. So with this I want to conclude that the conservation of freshwater crabs is very important, because they will be playing an important role in as a scavenger as well as food chain. So for the conservation, we have to study them, we have to make a list of it, and as well as we have to conserve the natural habitat or microhabitat for the survival of freshwater crabs. These are the references. Thank you. Thank you for the opportunity.
Any questions?
Yes, sir. Yes, sir. There are some, as I told, the first species, Cunicularis, it will be abundantly available in the paddy fields as well as in agriculture lands, streams and as well. So they are captive and they will be edible.
Any other questions?
I have referred one paper about the effect of pesticides on this freshwater crabs, especially in agriculture land, about the effect of copper sulfate, sir.
Copper?
Yes, sir.
I'm talking about paddy field.
No.
Especially you find in wetland paddy field.
No, sir, I didn't
But copper sulfate is generally for an arecanut garden.
Yes.
But nevertheless, it can also reach paddy field through the rainwater and other things. But a direct effect due to fertilizer and pesticides in the paddy field.
No, not in paddy field.
Yeah, the Westry is is a review, it's not your
Yes,
work
sir.
actually.
No, sir.
No. Can you tell me the species name of your first slide, your presentation slide?
That is Gatiana Dvivarna, which was identified in discovered in 2022.
Where?
In Uttara Kannada district.
Thank you.
Yeah, sorry, 2022.
Okay, thank you. All the best.
Thank you, sir.
The third oral presentation is by Mr. Jean Sibyl Vegas, Research Scholar, Department of Applied Zoology, Mangalore University. He is going to present the paper on the title, Molluscan Diversity and Land Use, Land Cover Changes Around Mangroves of Udyavara Pangala Estuary.
Good evening. Good evening. I am Miss Jean Sibyl Vegas from the Department of Applied Zoology, and my topic is on the molluscan diversity of mangrove forest of Udyavara Pangala Estuary, as well as land use, land cover changes. Next, please. Okay, so mangroves are important tidal forests, and they have the presence of important fauna such as aquatic invertebrates such as crustaceans, gastropods and polychaetes. So the mollusks which are found are usually, they can be found on the ground, buried into the sediment, or even found on the trees. And also we have used the satellite remote sensing in order to study the changes of the mangrove cover which has taken place over the years. Okay. So the aim was set up by finding the lack of information from the literature related to the Udyavara Pangala estuary of Udupi district. Due to some limitations, one sampling site was selected for continuous sampling of the mollusk. So the mollusks were followed, the epifauna was collected from quadrates, the infauna was collected by the core samples, and the arboreal fauna was selected on 2 of the trees which are dominant in the mangrove forest. Identification was carried out using standard guides as well as by confirming with the experts from India and around the world. Yes. Land use, land cover changes was studied using remote sensing Landsat series. So for 2002, 2013 and 2022. And the different procedure was followed. You have the data processing and the image generation. We have used false color composite, NDVI index, which is the normalized difference vegetation index, unsupervised and supervised classification. Coming to the results, a few of the mangrove species also were recorded. 6 species were recorded from our site, and compared to the 2 other studies, so about 12 species are found in this estuary, of which we have recorded only 6. So these are some of the species, of which Rhizophora mucronata and Avicennia officinalis were more dominant, which was used for the record of mollusk, arboreal mollusk. Okay, these are my species which are found. Actually, there are 18 species. Okay, the 18th one is Melampus singaporensis. The 10 families are found, 18 species are found, majority of which were epifaunal. Epifaunal means they occur on the soil. 1 was infaunal and many of the others were arboreal. But 2 of these species were epifaunal as well as arboreal, as an amphibious nature. Okay. This is how they occur in the field. You have the 3 examples of epifaunal species and 1 example of the infaunal species. You can make out that the color is quite camouflaged, or there is a lot of mud which is found on the animals, which is making it like you have to find for it. Okay. And arboreal, you can see that they occur on top of the trees, which is on the root, stem and even the leaves. So out of the 2 different species we have selected, so the 4 species were common to both the trees. 1 was found only on R. mucronata, that is Littoraria carinifera, and 3 were found only on A. officinalis, which is shown in green. Okay. So these are my species which are found from the mangroves. We have the 18 species, showing the 3 bivalves as well as the 15 gastropod species. Coming to the density of the animals, so N. violaceum had the maximum density, and telescopium had the good amount of density in epifaunal, whereas for the arboreal species, N. violaceum and L. bengalensis had the higher density. This is the false color composite of the estuarine area, where the red color shows the presence of vegetation and the other heterogeneous patches, as well as the NDVI, which is going to help us differentiate mangrove as well as vegetation. This is the unsupervised classification, which is done directly by the software, and then we have done the supervised classification in order to get the different land cover classes, which can then be tabulated in order to see the changes what are found. So what we have seen is that mangrove area was 2.13 square kilometer in 2002, and it has reduced now to 1.43 square kilometers in 2022. So we have about 33% which has reduced. So this is the conclusion what is there. We have found a good amount of mollusks which are found in the mangroves of Udyavara Pangla estuary. And it can be said that the other sites also may have the presence of these species, but which has to be sampled in the future along with the ecological study and heavy metal pollution bioindicator activity of other species. And coming to the GIS mapping, we have seen that there is a decreasing trend of mangrove cover in the past few years. Hence, rehabilitation has to be carried out, as well as finding of suitable location for planting of new saplings, as well as regular monitoring can be done using even higher resolution satellite data. Yes, okay. Thank you very much. These are my acknowledgements.
Yeah, thank you. It was very good presentation.
Yeah, thank you.
Any questions from the audience?
Yes, sir. So the mangrove forest is very important because it provides protection from predators. It has high amount of organic matter which is found from the decaying leaves, and it provides a good substratum for the mollusks to come and attach in the estuary area what is there. Compared to marine, the diversity is less, actually, yes. But we have recorded some of the micro shells what are there, which is less studied. So that has given us a more higher value in the diversity number. But actually, since it is a very stressed environment, continuous daily inundation, as well as seasonal factors, it's not very suitable for all mollusks. Only those who are able to handle the stress what is there, they have the adaptation. Many of them have the pulmonate cavity also. So that helps them to avoid the tides. They can avoid and they go on the tree. Such things. Yes, resilient. Yes, and like Littorinidae family has been evolved along with mangrove forest itself. So that is how they are found more, and we will have the presence of other estuarine species also which will occur in the mangrove.
Okay.
Predatory crabs are there, sir. Crabs and birds. Aquatic birds I have not recorded. During my study time, because maybe we are there, I don't see. But mostly crabs are their main predators. They have the capacity even to sometimes drill hole into the shell and they can extract the animal. They may also. Migratory birds. I have come across, but during my study I have not recorded the predation behavior.
Okay, nice research work.
Thank you.
So have you done any analysis for the soil which is present in the mangrove areas? Because you have said, like as I said, it is rich in a nutrition condition.
Yes, organic matter is quite high in
Quite high.
Yeah.
So the mollusk diversity is more in that.
Most of my species are detritivores, so they feed on dead and decaying organic matter. So that is why we have got large number of detritivorous animals, as well as herbivores, grazing animals.
Yeah, which method you use for determining the species diversity indices?
Indices I have not done for this paper, but we can use past. We can do diversity, Simpson's index, Shannon's index, evenness, dominance.
Okay.
Yes, sir. Yes. In literature, it is said that mangroves have the capacity to actually escape the sea level rise. But since the human presence is already there on the shores, they are not able to go further into. So the prediction is that mangroves may disappear within next 100 years or so. So the species which are directly related to mangroves may have the problem of extinction.
Okay, thank you.
Yes,
All
thank
the
you,
best.
sir.
The fourth presentation of today's session is by Mr. Supreet Kadakol, Assistant Professor, Department of Zoology, Sri Venkatramana Swami College, Bantwal. He is going to present the paper on the title, An Annotated Checklist of Birds of Kalinjimale Reserve Forest, Mangalore.
Myself, Dhanraj, from SS College, Bantwal, and I am here to present an annotated checklist of birds of Kalinjimale Reserve Forest. It is done by combination of three my friends, which is Mr. Jnanesh and another my guide, which is Supreet Kadakol. Next slide. Biodiversity. What is meant by biodiversity? Biodiversity means the animals living on that place, which is called biodiversity. There is mainly two levels, which is one is high level. High level means look at on all the animals which is present on the earth, and another one is low level, which means that there is biodiversity within a calibrated ecosystem of an educational campus. Objectives of the studies: it is the study area which is very rich with the fauna, which is Kalinjimale Reserve Forest, which is well known for its biodiversity, and it supports more than 125 species. And the animals and birds of this campus are ample green cover, which means there is full of green trees, and these are open grassland, thick vegetation patches, and patches of scrub jungle. We used to document it by using binoculars and other cameras for capturing photographs. Your binocular we used, which is the Nikon, and the camera, which is the Nikon camera. Then next slide please. Methods: The methods which is we woke up at early at morning, nearly 7:30 to 9:30, and visit to the observation place, which is where the field work to be done, and we observe the birds which are there, and the area which is 2023 of reserve forest, which is 108 acres, which is spreaded out, and it is located in southwestern extremely Bantul Taluk called Kannada, and at an elevation of 236.26 meters from the sea level, with various structures including open grassland, thick forest patches, scrubbers, jungle patches, and water bodies. Next slide, please. This is where our observation site, and this is where we documented the birds in Bantul Taluku, and this is the map of Dakshina Kannada under the Bantul Taluku region, and it is Kalinjamale forest mainly located in Vitla town. Okay, next please. Result and discussion. The present study, which is revealed that there is more than 54 species which is belong to 16 families and 7 orders. From the study area, details such as common scientific name, frequency, and conservation status of the wetland birds are presented in. The order of cardriforms dominated the list, and the least one is at least by podicipediforms, and the syconiforms are syconiforms, pelacaniforms, anseriforms, and gruiforms are also present in that area. Next slide, please. Next. The family which is syconiforms, which is in silver color, which is mostly found, we found most syconiforms birds, which is example for syconiform birds are white egrets and white storks. These are, and the podicipediforms we found that like western grebes and horn grebes. These are we founded on this place. Next slide. These are the birds we observed in under our observation, and the orange manivit, red whiskered bulbul, and grey hornbill, ringed parakeet. These are the birds where we observed in our observation site. Next slide. These are the references of my, and thank you.
Any questions?
Hornbill? Yes, is there. Hornbill is there.
From which part, may I know? From which area?
Kalinjamale Reserve Forest, sir. Near Vitla town.
Okay, it's fine.
It is in Karnataka, sir.
Karnataka. White egret.
White egrets. Not Ardidae family?
I searched on internet, and Grimet book I referred only the birds watching.
What all the 9 species of egrets you have recorded? Herons and egrets? Egrets and herons.
Egrets and herons.
Can you tell about the species what you have recorded in your area?
I only searched internet, sir, and it is based on the student project.
Okay.
So I referred the mains or guidance, sir.
What was the reference you have referred?
References are here, sir.
Birds of International Saving. This is MSc project work.
No, yes, sir. It is a project work, and I done student project work. So
yeah, student project work means MSc
project work.
It is
degree project work.
Degree.
Yes.
BSc level. BSc level project work. Huh?
Yeah,
project
BSc.
Work. Yes, BSc.
Yeah. That's a degree college, sir.
Okay. Thank you.
Okay, thank you.
Good observation.
Thank you.
All the best.
The fifth oral presentation of today's session is by Ms. Manasa Bhatt, Assistant Professor, Department of Postgraduate Studies in Applied Zoology, Alvas College, Mudbidri. She is going to present the paper on the title, Impact of Habitat Loss on Biodiversity, a Case Study in Katpadi.
So greetings all. So my name is Manasa and I'm going to deliver the presentation on Impact of Habitat Laws on Biodiversity, a case study in Katpadi. So these are my table of contents. Okay, next. So what is biodiversity? A basic thing what we literally know about biodiversity is that it's a variety of flora as well as fauna. Okay, it might be the interaction and total number of species present in an area. If you go for the broader perspective, we can take about entire earth, okay, or if you go for the limited context, area like diversity in MGM college or anything. So mine, actually the project work which I have done, I have taken the limited context, that is in a particular area. My project or my presentation is mainly based upon anthropogenic activity. I am actually studying about Katpadi area, okay, I live in Katpadi and mainly this is my study area and I am mainly dealing about the cropland ecosystem. You know, everybody know about agricultural ecosystem or the cropland ecosystem, how beautifully and how diversity, okay, diversity of flora and fauna will be there in that particular area, isn't it? So I have studied for 5 years. So this is actually the study area, Katpadi near Udupi, and this is the entire cropland which I have studied. So here is the cropland over here, and this is actually the brinjal plantation from June. So from July to October we grow actually paddy, and this is actually the brown patches is for paddy, and here you are going to see this green color patch where from October say March we are going to grow brinjal, okay, and here is the plantation area where we have coconut as well as the arecanut plantation and mix of jackfruit trees and all those things. And here in the end we have the wild plants which is actually present in that particular area from 100 plus years. From my this one grandparents era, it's actually present in this wild patch, okay, all those trees and all those things. So here is the methodology. So from 2018 to 2023, I have conducted this work. So I have divided this work into 2 phases. Phase 1 as well as phase 2. The phase 1 is actually from 2018 to 2021, okay, where I literally studied about diversity of insects. Mainly I focused on insect because I love entomology. So I focused on insects. Later other flora as well as fauna. All those floras which are present, some are host plants and some are actually present in that particular area, and certain fauna which are actually visiting this particular cropland area. And the second phase where actually our agricultural land or the cropland which where we grow this one paddy as well as brinjal, we actually there is a land conversion, okay, decommission of the well, okay, deforestation take place in this particular cropland and led to the overall 60% loss of the insect species present in this particular area. Actually we didn't thought that, actually as an entomologist even I didn't thought that land conversion in this particular land area will give a drastic reduction in the insect species, which actually I loved. Okay, even I tried to retrieve the host plant in the end of the day, but it didn't work properly. So I want to give the presentation on that. So this is actually the result part. Okay. So in the initial phase, we actually, I actually recorded 182 different insect species belonging to 72 families and 11 orders, where I found Lepidopterans were actually in a larger numbers and Dermapteran species are actually very much in the least recorded. And I am not specialist in botany, but I have identified 60 plus plant species and 50 plus other faunal species actually visiting in this particular cropland area. So these are the types of insect based on food habit, nectar feeders. So I want to tell you that it's been like 10 to 15 years, okay. My father is actually a farmer, and 10 to 15 years, okay, you have seen the study area satellite wave where there is a wild plantations and all those things, right? So I have identified 182 plus insect species in this particular cropland area. So whenever we studied, okay, certain, there might be good consumers as well as the bad consumers in insects. Good consumers in the sense they are actually beneficial, okay, and bad consumers are actually the pests. The wild population of the plants, flora as well as host plants were there in this particular cropland, okay, all the pests or other insects. I have found lot of hemipterans, coleopterans, and even the nectar feeders, a lot of butterfly species, okay, lepidopteran species, along with that even this one coleopteran species, okay, even the hymenopteran bee species were in larger number, which actually helped in increasing the yield of the cropland. We used to get 25, say 35, okay, 35 was the highest quintal of cropland, this one paddy yield we used to get, and we used to win the prize in our regional area, okay, because of this particular insect diversity. I would say that we have a lot of insects in that particular area. Even the nearby cropland, there is actually continuous households, next to there is actually a basic paddy field, and we have a paddy field, and we have we have man-made plantation, that is coconut and arecanut, and here we have the wild plantation over there. So wild plantation, along with that we have different wild floras, along with that we have planted floras and all. Those things actually helped in increasing the insect population, and it helped in the increase in the crop production in that particular land. Soon after we actually converted this particular cropland, even we actually this one we have cutted the trees, wild trees especially, and other host plants which are actually associated with the wild trees. Here if there is a renje mara, we have other host plants of other like Tonycocerca, passiflora flowers and all, those were actually the host plants. Even those were actually we have removed, okay. So because of that, the nearby farmers, okay, there was no host plants, there was no tree, continuous house were there in that particular region. Even they get affected. There is no particular insects now which we seen in this particular this 2024 and 2023. So these are actually some of the insects which is recorded during 2018 to 2021. And some of the insect host plant interaction. Okay, you can see here the milkweed bugs and all those things. Even though there is a hemiptera, milkweed bugs and all, they are actually mainly feeding on the milkweed plants, or like your this one, Calotropis gigantea and all those things. So they won't be affecting the cropland. But the other things actually like your beetles, okay, and other butterflies as well, moths and all those things present in this particular cropland, they help to increase in the productivity of the cropland. So these are actually the common pests observed during this one. So most of them, okay, one thing actually I have noticed cropland is that paddy field as well as brinjal field. So whenever we used to actually add a lot of pesticides, okay, for the brinjal plantation in the end, like if it is from October, say March, in the end of the March and all, there was a lot of things. We even tried organic farming. Organic farming was actually good. We got a good result. But chemical things which we used, okay, we used to add every regularly whenever we used to find a lot of aphids and all those things in the end of March. But if you go for this one paddy field, okay, so we never actually got a determined decrease in the paddy field yield in 10 to 15 years, and we never used pesticides. Once this one after rainy season, once pesticides were not pesticides, insecticides were used in a little quantity, but nevertheless we never regularly added the insecticides and all those things to the paddy field. Even though there was hemipterans, there were butterflies, and there are phagophytic beetles were found, we never got any reduction. We used to get like a graph was actually same 25 to 30, maximum 35% quintal of yield we used to get. But if you go for the brinjal plantation, even in Katpadi and Mattu near the beach area, okay, there is a continuous cropland. There is actually continuous cropland area, and nowadays land conversion is actually common. They convert those cropland area and they will be converting into, they will sell, and the house will be actually built in that particular area. So because of that, even insect species, when I was a student in 2018, even we have even I have checked insect species in the Mattu, okay, that is near to the Katpadi. Even they had lot of insect species. Now because of the land conversion, even there is a reduction, okay, in the yield, because there is no area for flora, okay, which is actually reducing the insect species, and it is giving the reduction in the yield. The next one is actually plant species, some of the plant species which is actually identified. So even along with the insect species, I wanted to tell you, I wanted to connect the topic is in such a way that reduction, some of the faunal species which I have observed. So host plants which was recorded during the study area. This is the phase 1. Okay, this is the cropland, this is the well. Okay, now there is a land conversion of this cropland and decommission of the well. We cannot see any odonates. I have actually I'm an odonator watcher, and there is no new odonates which I find because of this decommission of the well. The only water supply which was there in that particular area, decommission of the well, and you can see how barren land it has become in the after the land conversion. Only certain things which I observed was which was all those insects which are associated with the this one coconut plantation and other things which are present in that particular area. So in the phase 2, 60% of decrement of the insect species were actually observed. So these are the insect species which I have recorded. Next. These are the plant species. So overall, I wanted to tell you that in phase 1, in phase 1 it was actually, we had continuous wild plantation. So in the wild plantation area, I used to find lot of birds. Lots of birds were having shelter in that particular things. First thing, there is lot of host plants. There are lot of flora, which is actually flourishing in this cropland also, wild plantation also, as well as here in the, coconut plantation and all those things. Soon after we reduce that host plant, we have destroyed this wild plantation as well as cropland. We will not see any insects. Reduction in the insects, there is no amphibians. There is a habitat loss for those things. There is no insects. There is no host plants for them. They cannot thrive. Reduction in the amphibians was seen. And even we used to get lot of snakes. We had lot of pythons, rat snakes. Even to our house, snakes used to come. Now there is no snakes. There is no reptiles. Even lot of havranis and all used to come. And even those were not there. Bamboo trees were actually in the wild species. Bamboo trees were also there, which was actually a basic area for the snakes. Lots of snakes were actually seen in that particular time, and later even those things are actually reduced. Even bird species are actually getting affected. Now you can just imagine insectivorous birds, and all those things. Now we can see how this particular alteration in this particular ecosystem, led to the loss, even the loss of plants and all those things, led to the migration of the bird species, because of loss of shelter and lack of cultivation. Now, even we have added other soil, now nutrition of that soil is actually lost. Now we cannot grow the paddy again, even if we want to, and even the reduction in number of pollinators, and there is that even it was acted as a breeding site. Lot of snakes, lot of mollusks were there. Even that is actually been destroyed. So overall conclusion I want to tell you, so soon after the land conversion was done, before the land conversion, I tried to retrieve some of the host plants, but I couldn't retrieve. So if it is possible, first we have to plan a little bit so that we can relocate those plants, or we have to reserve those plants, or we have to create an awareness. One thing I have, this main thing I wanted to tell you is that from this 3 acres ka land, where 2.5 acres were paddy plantation and 0.5 for brinjal plantation, and with wild plants near to this, they had lot of 182 plus insect species were there, led to the increasing yield of yields were there, and a lot of biodiversity was seen. Just in 3 acres of land, the destruction of those things led to the overall decrease in the biodiversity. Just imagine in a smaller area, this happens means if we destroy entire forest, or if we destroy entire thing continuously, so it is going to literally damage the entire biodiversity of this particular area. References?
Okay, thank you Manasa for your extemporary speech. Now questions from audience, I think, sir.
We are all aware that any development is at the cost of biodiversity or ecosystem. But what was the nature of your destruction here?
Nature was like, we never thought that biodiversity will be getting affected in that larger thing. We have first, deforestation was the first thing in that particular land area. Then the cropland was altered. So soon after that, wild plants were actually removed, where wild plants, along with that we had host plants also. Lots of insects which are continuously visiting, they were also lost, and even birds were also lost. Even the nearby farmers who used to get lot of yield.
My concern is, see, you wanted to go for cultivation, right?
No, we had land cultivation first.
Okay, that is over. Suppose somebody wants to go for cultivation of land with some other crop species. Then also you can as well do it. But certainly at the cost of certain species, but not that even yield increase, food production is also required.
Yes.
Then how do you compromise?
It's a thought-provoking question. You think in your own, okay? Any other questions? Yeah.
Interesting work you have carried out, but the thing is, it should have been in a systematic manner rather. Because when you say that biodiversity assessment has to be done, you have covered many aspects. The thing is, if it is a seasonal studies, covering all the areas, taking different habitat, taking different fauna, flora, so that would have been better, so the students would have learned something what are the species of plants and animal species are found. And I am really surprised that you have done 5 years of studies from 2018 to 2023. Within that, you have got a COVID period. My interesting question is, after COVID, because during that period, we believe that the biodiversity is at least regained, or probably no further loss was reported. Are there any such information you have gathered immediately after, because you have restricted only 3 acres of land?
Yes, 3 acres of
Are there
land.
any such information you can give based on your studies?
Sir, actually, during the time of Corona, a lot of species, that 182 species, whatever I have actually checklisted of insects, Corona time alli jaasti sikkittu. Land conversion soon after Corona was done. So because of that, the 3 acres of land, alli nange insect species kammi siklike start aitu.
You are a teacher in a degree college. Now, if at all there is a scope for you to give the students some project, what will you suggest based on your observation of 5 years of study? What kind of project you can suggest to the students?
Okay. So first thing is that I actually wanted I will do the division among the students to take few areas. In that few areas they will be main thing is that ಅವರು ಒಂದೊಂದು ಚಿಕ್ಕ ಚಿಕ್ಕ ಜಾಗನ ಅವ್ರು ಆಪ್ಟ್ ಮಾಡ್ತಾರೆ, ಅಲ್ಲಿ ಬಯೋಡೈವರ್ಸಿಟಿ ಫಸ್ಟ್ ಸ್ಟಡಿ ಮಾಡ್ತಾರೆ. ಇನ್ ಕೇಸ್ ಅಲ್ಲಿ ಏನಾದರೂ ಆಂಥ್ರೋಪೋಜೆನಿಕ್ ಆಕ್ಟಿವಿಟಿ ಆಗ್ತಾ ಇದೆ ಅಂತ ಹೇಳಿದರೆ ಅವರು ಫಸ್ಟ್ ಅವ್ರು ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ತಾರೆ. ಆ್ಯಂಡ್ ಲೇಟರ್ ಬೈ ಲುಕಿಂಗ್ ಇನ್ ಟು ಡಿಫ್ರೆಂಟ್ ಆಂಥ್ರೋಪೋಜೆನಿಕ್ ಆಕ್ಟಿವಿಟಿ ವಾಟ್ ಎವರ್ ಇಟ್ ಇಸ್ ಇನ್, ವಿಲ್ ಟ್ರೈ ಟು ಟ್ಯಾಕಲ್ ದೋಸ್ ಪ್ರಾಬ್ಲಮ್ ಇನ್ ದಟ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಸಣ್ಣ ಏರಿಯಾದಲ್ಲಿ ಫಸ್ಟ್ ಟ್ಯಾಕಲ್ ಮಾಡಲಿಕ್ಕೆ ನೋಡ್ಬೋದು. ಆಮೇಲೆ ಚಿಕ್ಕ ಚಿಕ್ಕ ಏರಿಯಾದಲ್ಲಿ ಹೀಗೆ ಆಗ್ತಾ ಇದ್ದರೆ ದೊಡ್ಡ ಮಟ್ಟದಲ್ಲಿ ನಾವು ಹೇಗೆ ಮಾಡ್ಬೋದು ಅಂತ ಲೇಟರ್ ನಾವು ಹಾಗೆ ಇದು ಕನ್ವರ್ಟ್.
You did MSc Bioscience or what?
Zoology, sir.
From where?
Alvas College.
Alvas. Good. Now, this study, can you say that it's an alpha diversity or beta? Which level of studies you can, in a broader sense, you can tell about your studies?
Alpha.
Whether it's an alpha study, beta diversity, gamma diversity.
Okay, it's alpha. Alpha level.
Anyway, please, because I'm sure that many messages can be taken from these studies, but still it can be fine-tuned. Thank you.
Better you should choose proper guide, number one, and also do literature survey, sampling methods, and what sir said, alpha diversity, beta diversity, what is diversity, you explained about biodiversity. So these are all the things you should look in, do review of literature, then you should understand how to conduct this work. Of course, you have done a lot of thing and you have collected lot of information, but systematically you have not properly planned and properly presented. And everything you should put into record. Inventarization is very important here. Okay, data collection, and properly you should present it. Okay, and analysis. Okay, so then you can come out with good outcome. Okay, all the best. Thank you.
Thank you, Manasa. Thank you.
The last presentation of today's session is by Ms. Neha Bhatt, PG student, Department of Postgraduate Studies and Research in Bioscience, Mangalore University. She is going to present the paper on the title Cissus quadrangularis, a traditional use and conservation, its pharmacological properties.
Good evening everyone. My name is Neha Bhatt. I am going to present the topic Cissus quadrangularis, a traditional use and its conservation. Next slide please. Next. Before going to the introduction, I have included the scientific classification of this plant. This plant belongs to the phylum Spermatophyta and subphylum Angiosperms and class Eudicots and family Vitaceae. The genus is Cissus and species is Cissus quadrangularis. Next slide. This plant is a perennial herb with medicinal properties distributed throughout the tropical world. It is believed that this plant is native to India, Sri Lanka and Malaysia, Java and West Africa. This plant is studied for its phytochemical constitutions, pharmacological activities, and toxicological evaluations. These yellow spots shows the distribution of the plant. And in Indian traditional system of treatment, namely Siddha and Ayurveda, these plants are extensively used for the management of asti, means bone related issues. Next slide. So traditional knowledge and uses. This plant is a vine that grows in Africa and parts of Asia, including in India. In Sanskrit, these plants are known as Asti Samharaka and Ashtangi. Asti Samharaka means it protects the bones from destruction, and Vajrangi means it makes the bone more, it strengthens the bone. These traditional literature on Indian system of medicine as well as the literature across the world suggests that these plants have proven efficiency in treating various ailments such as osteoporosis. Osteoporosis is the condition in which it is the bone disease in which the mineral density of the bone is decreased. These plants help in increasing the mineral density of the bone and hence treats the osteoporosis. And these are good wound treaters and also helps in bone and fracture treatment. Next slide. So objective of my study is to evaluate the properties of this plant and to screening for its phytochemicals using ethanol, methanol and aqueous solutions, quantifying the primary and secondary metabolites present in the plant. Next slide. So materials and methods. The selected plants where I have collected the plants from the Udupi district and Mangalore University campus in the month of October, and these plants were authenticated by the taxonomist of our department based on the organoleptic solubility and nutritional values of these plants. Next slide. So organoleptic solubility, functional properties were determined by following the standard procedures recommended in Indian pharmacopoeia. The quantification of the primary and secondary metabolites were done using these methods. Coming to the result, first table shows the organoleptic analysis. Organoleptic analysis is the property of medicinal plant which stimulates the sensory organs of the humans. It includes taste, smell, odor and color of the plant. The solubility is also tested using these following solvents. The table 2 shows the functional properties of the plant. Functional properties means the screening of amino acid profile is very important in the knowing the properties of the plant. It shows the high emulsion property and emulsion stability, which is important when it is used in food industry. It helps in binding of these plant products to other food products. Next. So table 3 shows the qualitative phytochemical screening. I have used 3 solvents, ethanol, methanol and aqueous, and these were the results found. Table 4 shows the quantitative analysis of the Cissus quadrangularis. The quantitative estimation of the primary and secondary metabolites may be useful in the analysis of the compounds. That would be essential to knowing the compounds which are responsible for the growth and development of the plant, as well as the therapeutical values and nutritional values of the plant. So this is the graph showing primary and secondary metabolites. It shows more alkaloid contents. Alkaloid contents will help the plants in protecting itself from the predators, and also it regulates the nutritional values and it regulates the development of the plant. Therapeutically, alkaloids are responsible for anesthetics, and anti-inflammatory agents. Next. So scope of my study in research and conservation of the plant. The plant has ethnomedical uses since ages, used by the tribes of Kerala. So the study of antioxidant, antibacterial, antifungal will help me to know more about the plant and help in the clinical study of the plant. These plants have to be conserved because lack of traditional knowledge, lack of knowledge in the youngsters. So these plants are conserved. The problem faced by these plants are, these are aggressive, fast growing plants. These will climb over other plants and trees, which will cover the whole plant, and it will block the sunlight, and thus eventually the plant will die. And also its weight, it is responsible for the death of the plant which supports it. According to Indian Biodiversity Portal 2018, in India, it is reported only in Assam, Kerala, Madhya Pradesh, Maharashtra, Tamil Nadu, Uttar Pradesh, and Andaman and Nicobar. My plan is to include Karnataka also into that list. So these plants have proven in the treatment of obesity, osteoporosis, diabetes, and heart diseases, and also for stimulate the muscle growth. So these plants will have more international demand in the upcoming days. So this was the preliminary work done by me. These plants are medicinally very important, and on the basis of the result obtained, the drug is useful in clinical conditions, and also this can also be used as potential ingredient to formulate nutraceutical products for medicinal and veterinary applications. Next. These are the references which I have used. Thank you.
Thank you, Sneha. Any questions?
Any earlier reports?
Earlier reports are there about this plant, but it is not more explored in Karnataka, especially in this region.
In terms of its potency, can you, because you have gone through the literature, I believe, can you say whether it is more potent, or there is some lot of variations between the reports which has been published before?
The nutritive values will change from region to region. So from this region, very less studies have been done. I have studied the No. I have due to the paper reviews. So my aim is to quantify the
just quantify
the metabolites present in the plant.
Compounds present in the plant.
Yeah.
Yeah, thank you, Sneha.
Thank you.
I think it's over. Anybody? Over. Okay, now I request on behalf of our judging committee, Dr. Lavina can give some opinion about on this presentation. You can speak here.
So, respected dignitaries of the dais and my dear friends. So, firstly, I like to thank the organizer of this BIOSEM 2K24 for inviting us as a judge. Mainly the principal sir, Ishwar sir, Pravina ma'am, and Manvita ma'am. Thank you. So we had a great opportunity to hear and interact with the resource persons also from yesterday. And firstly, we want to congratulate all the 14 selected oral presenters. Means yesterday we had 8 oral presenters and today we had a 6. So I congratulate everyone because you have been selected for that oral presentation firstly. So everyone has done very good presentation at their research or project knowledge. So when we come to the judgment point of view, I would like to say few points. So when you present anything, make sure your slide contains every content like introduction, it may be objectives, methods and materials, methodology, result and conclusion, reference, and reference. And one more thing, what we have noticed is in yesterday's slides, there was no alignment. So make sure next time there should be a proper alignment. Even the spelling mistakes also we will look for. Then font size. I think when we do a PPT, I think better to use a font size of 24, not more than that. And only so much I want to say, sir. More than, okay.
Yeah, thank you, Dr. Lavina, for your comments on their presentation. And also I thank organizers, particularly our Dr. Ishwar Bhatt sir, for giving me an opportunity to chair this session. And also I thank Dr. Lavina and Yogis, the co-chairs of this session. I thank all the presenters, and we are expecting their research, but unfortunately we didn't get their research. Only few members are presented their research. Others, they presented only the reviews. I don't know how to consider for this competition. We will think it over and we will announce the result at the valedictory section. Thank you. Thank you all for all. Thank you.
Thank you, judges. A warm welcome to everyone present here. We have a short session of 15 minutes now. I, Vaishnavi Pai, take this opportunity to introduce Dr. M.K. Naik, former Vice Chancellor, University of Agricultural and Horticultural Science, Shivamogga. Dr. M.K. Naik obtained his PhD from IARI, New Delhi, with Dr. S. Ramanujam Memorial Award of Genetics, Club IARI, New Delhi in 1990. I request Ishwar sir to escort and florally welcome Dr. M.K. Naik. Dr. M.K. Naik has had outstanding work experience and is a recipient of many awards. To name a few, Karnataka State Young Scientist Award 1994, University Best Teacher Award Raichur 2011, US India Fulbright Nehru Visiting Fellowship 2010. He is also a fellow of a range of societies such as National Academy of Agricultural Science, New Delhi, National Academy of Biological Science, Chennai. Being a travel enthusiast, Dr. M.K. Naik has explored various corners of the globe like America, England, China, Thailand, and many more. He has nearly 190 research papers in journals and books and has also attended several conferences. As Vice Chancellor KSN UAHS Shivamogga, he is responsible for the overall development of this new university as a unique integrated farm university accredited by ICAR ranked 21st and 32nd. Dr. M.K. Naik is an accomplished teacher, outstanding researcher, and an administrator of par excellence. He has a distinct vision for development of human resource and academic institution. We are deeply privileged to welcome someone as distinguished as yourself to our gathering. Welcome you, sir. I request Dr. M.K. Naik to take over. I also request Ishwar sir to florally welcome him.
I'll start here. Okay. Thank you very much. So maybe very good evening, isn't it? Yeah. Dignitaries sitting in this August gathering, ladies and gentlemen, particularly dear students. In fact, I am really sorry that this was a last minute presentation. So we have snatched away some time from the precious time that is allotted. So we will try to see that because these slides were prepared just now. So it might be a bit not in organized fashion, but I will try to see that it is summarized and presented as early as possible. Okay. So what I thought, being an agricultural scientist, I thought I should speak upon the agriculture and biodiversity. Morning, Dr. Vigneshwar, the dean and the ex-registrar of the university, spoke on traditional way of looking at the biodiversity. But what I have just planned today is to look at what exactly agriculture has happened right now and how it has affected the biodiversity, and in turn how it has degraded or what could have been done in order to regenerate the agriculture through biodiversity. This is what I have been planning. So most of our students will be, I am sure, benefited by this one. So can you just put off the light so that it will be visible? Yeah. After all, our story is a story of a hungry creature in search of food. All along the history, you see, the global population, it took almost millions of years to reach the first billion, 100 crore population. In 1800, we reached the first billion. In 1930, we reached the second billion. That means in 130 years. The next billion came in less than 30 years. And 5 billion, that is 500 crore, came in less than 27 years. And the next billion came in 12 years. And the next billion came in 11 years. So today we are 700 crores on the planet. And this is the only planet that is a living planet. Although we have been trying hard whether it is possible to go and live on moon. So Chandrayana 3rd, and we have already landed on Mangala to see the Angala of Mangala, but still not finding a place there. So that's why, so every time the gap between the billion is decreasing. So that only shows our ability of fecundity or reproducing ability of the human being to reach 700 crores. Today, or globally, Indian population is 140 crores, right? But we have also reached a paradoxical scenario. What is that? Food production is now 5 times more. That means it is 330 million tons, not 5, 6 times. Before independence, we have just having 50 million tons. Horticulture production is now 325 million tons, 6 times gone up. Milk production almost 6 times, now it's 200 million ton. We are first in the world. And egg production is 27 times. And the we are the only country in the world to enacted Food Security Act. And we have promised that we will be able to feed almost 80 crore people in the Indian continent to feed them under the public distribution system. So that is the kind of courage this agriculture has given through the farmers, that we will be able to feed 80 crore people under public distribution system. Exactly, the Honorable Prime Minister extended for the next 5 years also, that we will be feeding 80 crore people every day. So that is the biggest task, and that is the biggest courage that we have. But let us also remember that once upon a time we were begging bowl. In Kannada we call it as Bhiksha Patre. Today we are exporting the same food from where we imported. America was the one country which exported, from where we imported the food. But for that country only we are now exporting wheat. So that is the kind of change that has taken place. But at the same time, our Human Development Index is very low, not a healthy statistics. 130 out of 180 countries. Yesterday record news data has increased a little bit, but it is not good. And global hunger index, we are 102 out of 117 countries. That is not a healthy statistics. Similarly, almost 20% below poverty line, life expectancy of course has gone up, but we cannot build a strong India on the shoulders of stunted and wasted children. We cannot build up. So that is why it is very difficult that we have to have the children who are very robust, good strength, good brain development, that comes through protein. So all that I am talking about is biodiversity in terms of food production, horticulture production, animal, egg, all these are biodiversity related. Next one. I have just taken the photo of Dr. M.S. Swaminathan, whom we call it as father of Green Revolution, who was awarded Bharat Ratna just 15-20 days back by the government of India, looking at the contribution for starving, what he did, the kind of service in seeing that the starvation is vanished in the country. Otherwise, in 1950s, we were almost a begging bowl. Today, the same country is in a position to export. Not only export, we have got the courage to feed 80 crore people just on public distribution system. So that is why all that has happened is nothing but the gene revolution. The green revolution is happened because of the gene revolution, and that gene has come from the wonder genes. That wonder gene is nothing but Norrin gene 10. Norrin 10. That was the wheat gene. Similarly, DU genes, what we call it a teaching native rice variety. When we were children, the varieties were taller than us. In rice field, when you enter, the rice was taller than us. But today, rice is dwarf, that is a dwarf gene, wonder gene, that the biodiversity affected gene. And that diversity is because of the wonder gene that has brought in the green revolution. And that is where the importance of biodiversity. Suppose if those genes were not there, discovered by Norman Borlaug, that is the reason why he was given Nobel Peace Prize. Nobel Peace Prize was given because he could see that the starvation is stopped, hungry millions of people were stuffed, food were given. Where there is a food, there is a silence. Where there is a food, there is a peace. That is why Peace Prize was given to him. People do not ask for two things. There is a saying. If you ask a hungry person, he will neither ask for reason nor for religion. Does he ask? He will only ask for the reason for his filling the stomach. That is why he was given Nobel Peace Prize. Next one. Today there is a changed scenario. We were deficiency in food, now we are sufficiency. We are now having food security, we are now moving toward nutritional security and food safety. We are having food import, now we are having export. Earlier we talked about green revolution, now it is the gene revolution, BT cotton, and rice also has been dwarfed. Several genes have been used, and today there is a mega rice variety has come through the CRISPR technology. That mega rice variety is able to yield several tons more. Similarly, primary agriculture, we are moving towards secondary agriculture. We are talking about starch. Now we have to talk more about proteins, minerals and vitamins. And chemical farming, now we are moving towards microbial farming. And from few crops, now we are moving towards biodiversified crop. Can anybody tell me today what are the crops that you have eaten morning? Morning, what is your nasta? Yen tindidira? Rice. Afternoon? Chapati. So rice has come, wheat has come. Evening? Dal, tomato. So ultimately one day you will have 4 crops. But how many crops we can eat? We can actually eat 3,000 crops. 3,000 crops are eligible to be eaten. 300 are grown all over the world. 150 are cultivated. 12 are dominating in the world. Of the 12, 3 are fed by 70 to 80% of the people. Which are those 3 crops? Rice, wheat and maize. And another 9 crops, banana, sweet potato, okay, cassava, some other, together 12 crops dominating 60% of the belly. So if this happens, you don't require any atomic bomb to destroy human race. If 700 crores of people have to depend upon 3 crops, then no need of any atomic bomb. If anything happens to these crops, human race will extinct. So that is why biodiversity is important, because we have hardly 17% of the world's population in India, and 4% of the world's water is India, 2% of the world's area is in India, and we have 142 million hectare, and 14% of the contribution comes from agriculture. Today 40 billion worth of export is coming from agriculture. With all, agriculture has several challenges. Next one. So India today is different from what we expect India tomorrow in 2050. Our population will be 1.65 billion and our GDP will be 10 times more. Our annual income will be 9 times more. Our income distribution will be still widened, and food prices will be soaring up, and urbanization is now there are 31%, and by 2050 65 out of every 100 persons will be living in urban area, and you have to do agriculture in urban area. Can you do? So the future of agriculture depends upon less of land, less of water, less of resources, less of biodiversity and less of farmers. And if anybody wants to become farmer, there is no bride at all. Nobody to come forward for marriage. So that is the situation. Anybody here ready to marry the farmer? Girls? Nobody is ready. But all of us want our bellies to be filled. Where it comes from? So that is the biggest challenge. Next one. So see, these are the challenges. Rising rural distress, land problem, water problem, shrinking of farm size. Our farm size is less than 0.65 hectares. Once upon a time it was 2 hectares. Land is fragmented and fragmented. Ultimately in whole of coastal area, paddy land is in guntas. Sir will be knowing, I think you will not have a land for cultivation. Everything here looks like a business. So agriculture cannot be a business. Next one. So these are the sustainable development goals. There are 12. First is no poverty, zero hunger, and land. There is a 12th is sustainable consumption, climate action, life on the land. These are all directly related to agriculture, but we have to achieve it in less than 7 years, because 2030 sustainable development goals we have to achieve in 2030, but we are already in 2024, less than 6 years. I think yesterday there is a prediction by the World Bank that it will be achieved only in 2062. What you need to achieve in 2030 will be achieved only in 2062. That is too far. Okay, next one. So I am now talking about biodiversity. Today, this year is International Year of Millet. Why that year of millet has come? Because the green revolution happened in 3 crops. Which are those? Wheat, rice and maize. And to produce 1 kg of rice, you require 5,000 liters of water. To produce 1 kg of millet, you require 500 liters of water. To produce 1 kg of chicken, you require 10,000 liters of water. To produce 1 kg of pulses, bedekalu, you require 800 liters of water. So that is why, in order to see that less of water is required, less of low degraded land also can be used for cultivation, less of fertilizer, less of pesticides, less of every resource. That is why International Year of Millet is celebrated this year, and the Honorable Prime Minister made a plea in the United Nations to declare it as International Year of Millet. Today we have foxtail millet, finger millet, barnyard millet, brown top millet. These are the major crops which are healthy food, and they are they are designated as nutri-cereals. In Kannada we call it as siridhanya. Earlier we were calling them as trinodhanya. Trina means hullu, good for nothing. But now they are made siridhanya because they contain calcium, magnesium, vitamins. More than that, it is a very good for diabetic patient. For patients who are suffering from cholesterol, these are wonderful food. That is why they are called as superfood. That biodiversity to be maintained. Next one. I am just touching, being a plant pathologist, fungi. There are some botany students, I believe. Not a single day will pass off peacefully without encountering fungi or microorganisms, either for the benefit or for the loss. Next one. How? See, there are about 1.5 million species. After the insect, these are the biggest biodiversity species. Okay? And there is for every plant, there are 5 fungi. And they are dangerous for diseases also. They cause diseases. In 1940, we lost entire rice crop because of one small tiny fungus, brown spot disease. That was a helminthosporium. Okay? Next one. So there are, I don't want to go into the details. It is a eukaryote kingdom. 4,900 genera are there. Next one. But very important that these are required for every day. Their biodiversity is required for every day because they are important for biotransformation, yeast, single cell protein, bread making, alcohol industry, beverages, antibiotics, penicillin, I think I am sure, amoxicillin tablet, who has not taken? 90% of us will consume, that has come from microbes. Penicillin, okay, food yeast, mycoprotein, industrial enzymes, enzyme making, bread making, cheese making, all these useful metabolites, biocontrol agents, everything comes from microbes. Tiny organism worth $1 billion. Can anybody imagine what is the worth of a penicillin tablet or a worth of a streptozicin tablet? Very difficult to imagine. Next one. So medicinal value, penicillin, cyclosporin, which is used for, immunosuppressant. When the heart has to be transplanted, it is needed. Lovastatin, that is a cholesterol reducing. Next one. Similarly, mycorrhizal fungi. I think all our forest species are maintained by mycorrhizal fungi. Edible fungi, mushrooms, and then yeast fungus. Next one. Biocontrol agents. Today you are talking about BT cotton. BT is a biocontrol agent, Bacillus thuringiensis. Okay. Next one, trichoderma. And these are also used as climatic indicators. Atmospheric changes are indicated by fungi, lichens. Next one. Then you see human and animal mycosis. Last year we suffered from COVID. During COVID, you heard of yellow fungus, white fungus, dark fungus, and those who are affected were dead also. That was because of this fungal attack. Then allergy, then spoilage of food, all our mycotoxin fungi. Next one. And these are the ones which are very deadly, causing abortions in human beings. Animals, they are also very deadly because they cause liver cancer. These are all very deadly fungus. Next one. Similarly, these are useful mushroom as a fungus, button mushroom, oyster mushroom, paddy straw mushroom. Next one. And they are highly nutritional and zero cholesterol food, less sugar, anti-tumorous, that property. one of the author writes, if you don't consume mushroom in your life, you will be missing something in life. They have anti-tumoral properties. They are also having antiviral property, and they are contain very less fat also. Next one. So this is the milky button mushroom. Next one. This is the pleurotus mushroom. I think now mushroom is available everywhere. Highly proteinaceous. Next one. And these are the mushroom pickles, soups, chutney, chips. All that is prepared from our university at Shimoga. Next one. This is a paddy straw mushroom grown in large scale. Next one. This is a milky mushroom. This is a very fond. Our Honorable Prime Minister likes this mushroom, and he is fond of getting it from Himachal Pradesh. So that is the kind of varieties of mushrooms now available. Next one. This is a large scale production. This is Dr. Prakasham. He is a consultant for mushroom, NRC mushroom. Next one. Scier mushroom. These are all biodiversity, diversity of fungi. Okay, next one. This is used for preparation of dye making. This comes to our arecanut garden. You find this mushroom coming up. That is dye is extracted out of it. Next one. See, this is a white fungus, yellow fungus, and this is how it affects the pulmonary, the respiratory system during our COVID era. These are the fungi which infected and caused the death of the COVID infected patient. Next one. So another most important is, this is what our Honorable Prime Minister wanted, that bioeconomy. That means biological resources and the biodiversity of microorganisms is explored for economic purpose as a renewable energy. Today, you see, a single spoon of soil contains $1 billion worth of microbes. Bacillus thuringiensis, Trichoderma, Pseudomonas fluorescens. Those are all billion worth of microbes producing several secondary metabolites, biomolecules. Next one. You can look at here, they are growth promoters, antimicrobial compound. They are used for antagonism, induced systemic resistance. Next one. These are the list of fungi, Trichoderma, Glyocladium, diversity. See, bacterial diversity. Next one. These are the antibiotics. You see, one of the antibiotics discovered in Shimoga Agriculture from our own laboratory. These are producing 12 antibiotics. Some of them are cancer curing, which are used in agriculture, but can cure cancer and tumor. So such of those $1 million worth of molecules which are to be explored in industry, then it becomes entrepreneurship. So that is why you call it as bio-entrepreneurship. Next one. Next one. Yeah. Look at the examples. Antibiotics, organic solvents, butanol, beverages, beer and wine, food, cheese and pickles, flavoring agents, organic acids, amino acids, steroids. These are all derived from biological origin of microbes. Next one. Look at the spirulina. Today it is a supermarket business. Those of you who know it, 55 to 70% protein comes from this one. It's a very good immune system, improves the digestion, it controls the appetite, and many times doctors will prescribe this food wherever the patient is suffering from indigestion. Next one. See, these are biofuel today, biofuel industry. There is a genetically modified E. coli used for ethanol production. Our government of India wants blending with petrol to the extent of 20%. But right now, we have only less than 4% of blending. And if you want our environment to be safe, if you want global warming to be reduced, if you want sea level rising to be avoided, if you want the coastal of Karnataka 320 kilometers or anywhere, if the coastal line civilization has to be restored, then you need to go for biofuel. And that is there in our microbes. Next one. Bioremediation. Today I think radioactive waste, oil spillage, alkane, pesticide degradation, all have to come from microbial origin. Next one. Probiotic food, which I already talked, yogurt food, tempeh food, kombucha, pickles, buttermilk. Next one. Arecanut. Today I think lot of arecanut is grown in Udupi and Mangalore area. I have taken that example here. 1 hectare of arecanut will produce about 12 tons of waste husks. But what can be done? And if you leave that husk on the roads all along Arsikere and Tarikere, it go once there is a rain, it will go and block the drainage. If you burn it, it will be environmental pollution. That is why we have identified a consortia of microbes which can degrade in less than 5 months. It can degrade into product which is compost, having high nutrition compared to farmyard manure. It has got a high CN ratio, NPK, calcium, magnesium. If you really want to avoid arecanut leaf diseases, add this organic waste into your arecanut garden. Your arecanut garden will be very safe, and within less than 4 months you can get the compost, which is available now 10 rupees per kg. So you can convert waste into wealth through this biological process. Next one. Suppose if you we conducted series of demonstration in Sagar area, or Surya Narayan Rao, many, Dr. Vigneshwar, they all saw this. If you leave it to the nature, it takes 2 years to degrade the husk, but it takes 4 months to degrade by the consortia of microbes. Next one. I just want to conclude that I think you have MGM College, Mahe, coastal belt is full of intellectuals, ideas, wisdom, knowledge. But the knowledge has to be converted into action. Action has to go for wealth. This is one outstanding example. One of our students from agriculture graduate. After BSc Agri, he went for master's degree in plant pathology. Then he went to PhD. Then he switched over to vaccine study. And he is the now Covaxin, supplied the Covaxin 700 million doses. I tell you, his name is Krishnamurthy Yella. Incidentally, he is not only a scientist, he is an entrepreneur. And what he wanted, we invited him as it was an honor for us to invite him and award the honorary doctorate degree. Our first honorary doctorate degree was given to him by our new university because he converted the knowledge into action, into wealth, and he has developed a vaccine for dengue virus, vaccine for your malaria, vaccine for many other, at least half a dozen viruses, including Covaxin, which is supplied, and that another hepatitis B, which was available in the United States for $70, which he brought it here and delivered 10 rupees per dosage, 700 million dosages were given. That is how hepatitis B was given. So that is why he is becoming a great entrepreneur and a great service was given, and he is also billionaire now. So that is how the knowledge, actual knowledge converted into wealth through the microbes. Next one. Last slide, way forward. Dr. MS Swaminathan, who was awarded the Bharat Ratna by the government of India just few days back, of course, posthumously, would have been better had he been awarded when he was alive, but better late than never. But what I want to say, he says 20, 40, 30, 20, 20. This is the formula given. 40 means the productivity has to be enhanced by 40% because we have about 26 million acres of land, degraded because biodiversity is lost. If the biodiversity is lost, one of the scientists was presenting, a girl was presenting, if the biodiversity is lost, that land is waste. So to regenerate the agriculture, to bring back the agriculture there, you need to go for generating biodiversity. So 40% enhancement of productivity has to come through that one. Rural poverty has to be decreased by 30%, and hunger has to be decreased by 30%, and emission because the carbon dioxide emission that may come from agriculture, because wetland paddy field contributes 54% of the methane. Dung will contribute methane, industry will contribute, and everything contributes for global warming. And today, this year we are expecting the hottest year for the last 25 years. So that is why I think people are now shouting, it is warming up. If you don't want the Earth is the only planet, living planet, we have to preserve it and protect it, share it and care it. So Mahatma Gandhi back in 1920 said, this earth can meet the needs of the people but not the greeds of the people. ಈ ಭೂಮಿಗೆ ಸಕಲ ಸವಲತ್ತುಗಳನ್ನು ಕೊಡಬಲ್ಲಂಥ ಶಕ್ತಿ ಇದೆ, ಅದರ ದುರಾಶೆಯನ್ನಲ್ಲ ಅಂತ ಹೇಳಿದರು ಅವರು. ದಟ್ ಇಸ್ ವೈ ವಾಟ್ ಐ ಆಮ್ ಟೆಲಿಂಗ್ ಈಸ್ ಇನ್ 1869 his birth, and his birth is considered to be symbol is Charaka. 1869. 1969 is the birth of internet, when we landed on the moon also. 100 years we traveled from Charaka to internet. Today whole world is in your hand, isn't it? Right in the evening you will talk to your daughter or a grandson living in United States. That is how the world has come. But at the same time, remember, schools and colleges were closed 10, 20, 30, 40 days back in Delhi for the want of oxygen. So this earth cannot meet the needs of the people, it cannot meet the greeds of the people. If you want to purchase, now water we are purchasing, but if you want to purchase oxygen, then there is deadly. So we don't want to wait for that. So if you really want Swaminathan said one word, countries which possess the grains will survive, but not the guns. Countries which will possess the grains will survive, not the guns, because guns is available by purchase, but grains are not available by purchase. That's why biodiversity we have to support, biological resource we have to support. By that we have to explore the biological resources through the knowledge for wealth, and see that this earth remains green forever. That is the message for this biodiversity. Thank you very much for giving me an opportunity. I think I am sorry I did not, it was a unexpected schedule. So I only promise that sometimes later I will bring a 1 hour full story of agriculture and biodiversity and voluntarily come and present to you, to the students. Okay, thank you very much. Thank you.
Thank you, sir, for your insightful presentation. I request Mrs. Saraswati, HOD of Department of Mathematics, to present Dr. M.K. Nayak with a memento of gratitude.
Okay. ಕೂತ್ಕೋಬೇಕಾ? ಟೈಮ್ ಇಲ್ಲಲ್ಲ.
Thank you, ma'am. Audience, let's please proceed for the tea break, which is arranged outside. I request you all to be back by 5 minutes.
ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲೇ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತ ಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949 ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ ಇದ್ದರೂ ಕಾಲೇಜು ಶಿಕ್ಷಣಕ್ಕಾಗಿ ಆ ಸಂದರ್ಭದಲ್ಲಿ ದೂರದ ಮಂಗಳೂರಿಗೋ ಮೈಸೂರಿಗೋ ಅಥವಾ ಮದ್ರಾಸಿಗೋ ಹೋಗಬೇಕಾಗಿತ್ತು. ಇದನ್ನು ಮನಗಂಡ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಉಡುಪಿಗೊಂದು ಕಾಲೇಜನ್ನು ನಿರ್ಮಿಸುವಲ್ಲಿ ಕಾರ್ಯಪ್ರವೃತ್ತರಾದರು. ಶಿಕ್ಷಣ ಕ್ಷೇತ್ರ ಪೈ ಅವರ ಆಸಕ್ತಿಯ ಕ್ಷೇತ್ರವಾಗಿದ್ದುದರಿಂದ ಈ ಹಿಂದೆ 1942ರಲ್ಲಿ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ ಸಂಸ್ಥೆಯನ್ನು ಸ್ಥಾಪಿಸಿದ್ದರು. ಈ ಸಂಸ್ಥೆಯ ಮೂಲಕ ಸ್ವದೇಶಿ ಸ್ವಾವಲಂಬನೆಯ ಶಿಕ್ಷಣಕ್ಕೆ ಪ್ರೇರಣೆ ನೀಡುತ್ತಿದ್ದರು. ಮದ್ರಾಸ್ ವಿಶ್ವವಿದ್ಯಾನಿಲಯ ಉಡುಪಿಗೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಕಾಲೇಜನ್ನು ಮಂಜೂರು ಮಾಡಿತು. ಕಾಲೇಜಿಗೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇಲ್ಲದಿದ್ದುದರಿಂದ ಆರಂಭದಲ್ಲಿ ಉಡುಪಿ ನಗರಸಭೆಯ ಪ್ರಾಥಮಿಕ ಶಾಲೆಯಲ್ಲಿ ಸಮೀಪದ ಕ್ಲಬ್ ಕಟ್ಟಡ ಸೇರಿಕೊಂಡು ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಜೂನ್ 27, 1949ರಲ್ಲಿ ಆರಂಭಗೊಂಡಿತು. ಕಾಲೇಜಿನ ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾಗಿ ಪ್ರೊಫೆಸರ್ H. ಸುಂದರರಾಯರು ನೇಮಕಗೊಂಡರು. ಆರಂಭದ ವರ್ಷ 79 ಹುಡುಗರು, 10 ಹುಡುಗಿಯರು, 10 ಪ್ರಾಧ್ಯಾಪಕರು, 7 ಮಂದಿ ಸಿಬ್ಬಂದಿಯೊಂದಿಗೆ ಕಾಲೇಜು ಅಸ್ತಿತ್ವಕ್ಕೆ ಬಂತು. ಆರಂಭದಿಂದಲೇ ಸಹ ಶಿಕ್ಷಣವನ್ನು ಒದಗಿಸುವ ಮೂಲಕ ಹೆಣ್ಣು ಮಕ್ಕಳಿಗೂ ಉನ್ನತ ಶಿಕ್ಷಣದಲ್ಲಿ ಸಮಾನವಾದ ಅವಕಾಶವನ್ನು ನೀಡಲು ಪಣತೊಟ್ಟ ಮೊದಲ ಸಂಸ್ಥೆ ನಮ್ಮ MGM. ವಿದ್ಯಾರ್ಥಿನಿಯರು ಕೂಡ ಊರಲ್ಲಿಯೇ ಕಾಲೇಜು ಶಿಕ್ಷಣ ಪಡೆಯುವಂತಾಯಿತು. ಮೊದಲ ಬ್ಯಾಚ್ನಲ್ಲಿಯೇ ವಿದ್ಯಾರ್ಥಿನಿ ಟಿ ನಿರ್ಮಲಾ ಪೈ ಮೊದಲ ರ್ಯಾಂಕ್ನೊಂದಿಗೆ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದಿಂದ ಚಿನ್ನದ ಪದಕವನ್ನು ಪಡೆದದ್ದು ಇತಿಹಾಸ. ಅಂದಿನಿಂದ ಇಂದಿನವರೆಗೂ ಪ್ರತಿ ವರ್ಷ ರ್ಯಾಂಕ್ ಗಳಿಸುತ್ತಲೇ ಸರಪಳಿ ಮುಂದುವರಿಯುತ್ತಾ ಬಂದಿದೆ. ಉಡುಪಿಯಲ್ಲಿ ಪೈಗಳ ಪ್ರಯತ್ನದಿಂದ ಕಾಲೇಜು ಪ್ರಾರಂಭವಾಯಿತು. ಆದರೆ ಸ್ವಂತ ಕಟ್ಟಡ ಇರಲಿಲ್ಲ. ಸಗ್ರಿಗುಡ್ಡದಲ್ಲಿ ಡಾಕ್ಟರ್ ಪೈ ಅವರು ಕಾಲೇಜಿನ ಕಟ್ಟಡಕ್ಕೆ ಶಿಲಾನ್ಯಾಸಗೈದರು. ನಿರ್ಮಾಣ ಕಾರ್ಯದಲ್ಲಿ ಊರವರೊಂದಿಗೆ ಉಡುಪಿಯ ಅಷ್ಟಮಠಗಳ ಯತಿಗಳು ಕೈಜೋಡಿಸಿದರು. ಕಾಲೇಜು ಪ್ರಾರಂಭಗೊಂಡ ಮುಂದಿನ ವರ್ಷವೇ ಉಡುಪಿಯಿಂದ ಈಗಿನ ಸ್ಥಳಕ್ಕೆ ಸ್ಥಳಾಂತರಗೊಂಡಿತು. ಸಾವಿರದ 1952ರಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಭಾಗವನ್ನು ತೆರೆಯಲಾಯಿತು. 1958ರಿಂದ MGM ಕಾಲೇಜು ಮೈಸೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಕಕ್ಷೆಗೆ ಒಳಪಟ್ಟಿತು. ನಂತರ 1980ರಿಂದ ಮಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಅಧೀನದಲ್ಲಿದೆ. 1956ರಲ್ಲಿ BA, BCom, BSc ಪದವಿಗಳಿಗೆ ಒಪ್ಪಿಗೆ ದೊರೆಯಿತು. ಇದೇ ವರ್ಷ ಕಾಲೇಜಿನ ಆಡಳಿತ ನಡೆಸುವ ಜವಾಬ್ದಾರಿಯನ್ನು ಶ್ರೀ TA ಪೈ ಅವರು ವಹಿಸಿಕೊಂಡರು. ಮುಂದಿನ 12 ವರ್ಷಗಳವರೆಗೆ ಅವರು ಕಾಲೇಜಿನ ವ್ಯವಸ್ಥಾಪಕರಾಗಿದ್ದರು. UGC ನೆರವಿನಿಂದ 1964ರಲ್ಲಿ ಪ್ರತ್ಯೇಕ ಗ್ರಂಥಾಲಯ ನಿರ್ಮಾಣವಾಯಿತು. ರಾಷ್ಟ್ರಕವಿ ಮಂಜೇಶ್ವರ ಗೋವಿಂದ ಪೈ ಸಂಶೋಧನಾ ಕೇಂದ್ರವನ್ನು 1965 ಡಿಸೆಂಬರ್ 17ರಂದು GP ರಾಜರತ್ನಂ ಅವರು ಲೋಕಾರ್ಪಣೆ ಮಾಡಿದರು. ನಂತರ 1966ರಲ್ಲಿ ಮಾಧವ ಪೈ ವಿಜ್ಞಾನ ಮಂದಿರದ ಉದ್ಘಾಟನೆ ಆಯಿತು. ಇದರಿಂದ ಕಾಲೇಜಿನಲ್ಲಿ ವಿಜ್ಞಾನ ವಿಷಯಕ್ಕೆ ಹೆಚ್ಚಿನ ಪ್ರೋತ್ಸಾಹ ದೊರಕಿತು. 1972ರಲ್ಲಿ ವಿಕ್ರಮಶಿಲಾ ಕಟ್ಟಡದ ಉದ್ಘಾಟನೆಯೊಂದಿಗೆ ಕಾಲೇಜು ಕಟ್ಟಡಗಳು ವಿಸ್ತರಿಸಿದವು. ಹೀಗೆ MGM ಕಾಲೇಜು ಹಂತ ಹಂತವಾಗಿ ಯಶಸ್ಸಿನ ಪಥದಲ್ಲಿ ಮುಂದೆ ಸಾಗಿತು. ಉಡುಪಿಯಲ್ಲಿ MGM ಕಾಲೇಜನ್ನು ತೆರೆದ ಬಳಿಕ ಡಾಕ್ಟರ್ ಡಾಕ್ಟರ್ ಮಾಧವ ಪೈಗಳು ಜಿಲ್ಲೆಯ ಬೇರೆ ಬೇರೆ ಕಡೆಗಳಲ್ಲಿ ವಿವಿಧ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭಿಸಿದರು. ಇಂದು ವಿಶ್ವದ ಪ್ರಮುಖ ವಿದ್ಯಾ ಕೇಂದ್ರವಾಗಿರುವ ಮಣಿಪಾಲದ ಕಾಲೇಜುಗಳು ಹಾಗೂ ನೆರೆಯ ಜಿಲ್ಲೆಗಳಲ್ಲೂ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳನ್ನು ಆರಂಭ ಮಾಡಲು ಮೂಲ ಪ್ರೇರಣೆ MGM ಕಾಲೇಜಿನ ಯಶಸ್ಸು. ಈ ಎಲ್ಲ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆಗಳಿಗೆ ಮುಕ್ಕುಟಮಣಿಯಾಗಿ ಇಂದು MGM ಕಾಲೇಜು ರಾರಾಜಿಸುತ್ತಿದೆ. ಶೈಕ್ಷಣಿಕವಾಗಿ ಮಹತ್ತರವಾದ ಸಾಧನೆಯನ್ನು ಮಾಡಿರುವ MGM ಕಾಲೇಜು ಉಡುಪಿಯ ಸಾಂಸ್ಕೃತಿಕ ಚಟುವಟಿಕೆಗಳ ಕೇಂದ್ರವಾಗಿ ಗುರುತಿಸಿಕೊಂಡಿದೆ. ಕಾಲೇಜಿನ ದ್ವಿತೀಯ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ ಕೂಶಿ ಹರಿದಾಸ ಭಟ್ಟರು ಆ ಹುದ್ದೆಯ ಘನತೆ ಗೌರವಗಳನ್ನು ತಮ್ಮ ಪ್ರತಿಭೆ, ಪಾಂಡಿತ್ಯ ಮತ್ತು ಪರಿಶ್ರಮಗಳಿಂದ ಹೆಚ್ಚಿಸಿದರು. 2010ರಲ್ಲಿ ನಡೆದ MGM ಕಾಲೇಜಿನ ವಜ್ರ ಮಹೋತ್ಸವದ ಸವಿನೆನಪಿಗಾಗಿ ಆಡಳಿತ ಮಂಡಳಿಯು ಹಲವಾರು ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡಿತ್ತು. ಕಂಪ್ಯೂಟರ್ ವಿಜ್ಞಾನ ಹಾಗೂ ಸ್ನಾತಕೋತ್ತರ ತರಗತಿಗಳ ಅತ್ಯಾಧುನಿಕ ಸೌಲಭ್ಯಗಳನ್ನೊಳಗೊಂಡಿರುವ ವಜ್ರಸೌಧ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ವಾದಿರಾಜ ವಿದ್ಯಾರ್ಥಿ ನಿಲಯವನ್ನು ವಿಸ್ತರಿಸಿ ಸುಸಜ್ಜಿತವಾದ ತಮೇರಿ ಕಟ್ಟಡದ ನಿರ್ಮಾಣ, ಆಧುನಿಕ ಶೈಲಿಯ ಒಳಾಂಗಣ ಕ್ರೀಡಾಂಗಣ, ಹವಾನಿಯಂತ್ರಿತ ಆಡಿಯೋ ವಿಡಿಯೋ ಹಾಲ್ ಇತ್ಯಾದಿ ಅಭಿವೃದ್ಧಿ ಯೋಜನೆಗಳು ಪೂರ್ಣಗೊಂಡಿವೆ. ನ್ಯಾಕ್ ಮೌಲ್ಯಾಂಕನದ 4ನೇ ಆವೃತ್ತಿಯಲ್ಲಿ ಕಾಲೇಜು A+ ಶ್ರೇಣಿಯೊಂದಿಗೆ ವಿಶಿಷ್ಟ ಸ್ಥಾನಮಾನವನ್ನು ಪಡೆದಿದ್ದು, ಕಾಲೇಜಿನ ಸಮಗ್ರ ಗುಣಮಟ್ಟ ಹಾಗೂ ಉತ್ಕೃಷ್ಟ ಸೌಲಭ್ಯಗಳನ್ನು ಗುರುತಿಸಿ ಈ ಮಾನ್ಯತೆ ನೀಡಿದೆ. ಕಾಲೇಜಿನ ಸಂಸ್ಥಾಪಕರಾದ ಡಾಕ್ಟರ್ TMA ಪೈ ಅವರ ಕನಸನ್ನು ಅನಂತರದ ಆಡಳಿತಗಾರರಾದ ಶ್ರೀ PA ಪೈ ಅವರು, ಶ್ರೀ T ರಮೇಶ್ ಪೈ ಅವರು ಮತ್ತು ಶ್ರೀ KK ಪೈ ಅವರು ಕಾಲೇಜಿನ ಯಶಸ್ಸನ್ನು ಉತ್ಕೃಷ್ಟ ದರ್ಜೆಗೇರಿಸುವುದರ ಮೂಲಕ ನನಸುಗೊಳಿಸಿದ್ದಾರೆ. ಅಕಾಡೆಮಿಯ ರಿಜಿಸ್ಟ್ರಾರ್ ಆಗಿದ್ದ ಡಾಕ್ಟರ್ ರಾಮದಾಸ್ M ಪೈ ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ನಿಕಟಪೂರ್ವ ಅಧ್ಯಕ್ಷರಾಗಿದ್ದ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈಗಳ ಆಡಳಿತಾವಧಿಯಲ್ಲಿ ಕಾಲೇಜು ಬಹಳಷ್ಟು ಅಭಿವೃದ್ಧಿಯನ್ನು ಕಂಡಿದೆ. ಪ್ರಸ್ತುತ ಅಕಾಡೆಮಿ ಆಫ್ ಜನರಲ್ ಎಜುಕೇಷನ್ನ ನೂತನ ರಿಜಿಸ್ಟ್ರಾರ್ ಆದ ಡಾಕ್ಟರ್ ರಂಜನ್ R ಪೈಗಳು, ಕಾಲೇಜಿನ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಅಧ್ಯಕ್ಷರಾದ ಶ್ರೀ T ಸತೀಶ್ U ಪೈಗಳು ಹಾಗೂ ವಿಶ್ವಸ್ತ ಮಂಡಳಿಯ ಸದಸ್ಯರಾದ ಡಾಕ್ಟರ್ HS ಬಳ್ಳಾಲ್ ಮತ್ತು ಶ್ರೀ ವರದರಾಯ ಪೈಗಳ ಸೂಕ್ತ ಮಾರ್ಗದರ್ಶನದಲ್ಲಿ ಕಾಲೇಜು ಹೊಸ ಸಂವೇದನೆಯತ್ತ ಸಾಗುತ್ತಿದೆ. ಸ್ಥಾಪಕ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ H ಸುಂದರ್ ರಾಯರಿಂದ ಇಂದಿನವರೆಗೆ ಹಲವಾರು ದಕ್ಷ ಪ್ರಾಮಾಣಿಕ ಪ್ರಾಂಶುಪಾಲರಿಂದ ಸಾಮಾಜಿಕವಾಗಿ ಸ್ಪಂದಿಸುತ್ತಾ ಸಾಧನೆಯ ಪಥದತ್ತ ಸಾಗಿ ಬಂದಿದೆ. ಪ್ರಸ್ತುತ ಪ್ರಾಂಶುಪಾಲರಾದ ಪ್ರೊಫೆಸರ್ K ಲಕ್ಷ್ಮೀನಾರಾಯಣ ಕಾರಂತ ಅವರ ನೇತೃತ್ವದಲ್ಲಿ ಇಂದಿನ ಸಾಮಾಜಿಕ ಅಗತ್ಯಕ್ಕೆ ತಕ್ಕಂತೆ ಅನೇಕ ಅಭಿವೃದ್ಧಿ ಕಾರ್ಯಗಳನ್ನು ಹಮ್ಮಿಕೊಂಡು ಸಾಧನೆಯತ್ತ ಆಶಾಭಾವನೆಯನ್ನು ಹೊಂದಿ ನಿಂತಿದೆ. ಅಮೃತ ಮಹೋತ್ಸವದ ಸ್ಮರಣಾರ್ಥ ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಪ್ಲಾಟಿನಂ ಜುಬಿಲಿ ಕಟ್ಟಡ ನಿರ್ಮಾಣ, ಮುದ್ದಣ ಮಂಟಪವನ್ನು ಮೇಲ್ದರ್ಜೆಗೇರಿಸುವ ಮೂಲಕ ನವೀಕರಣ, ಶ್ರೀ T ಮೋಹನ್ ದಾಸ್ ಪೈ ಕೌಶಲ್ಯಾಭಿವೃದ್ಧಿ ಕೇಂದ್ರದ ಸ್ಥಾಪನೆ, 75 ವರ್ಷಗಳ ಸವಿನೆನಪಿಗಾಗಿ 75 ಅರ್ಥಪೂರ್ಣ ಕಾರ್ಯಕ್ರಮಗಳನ್ನು ಆಯೋಜಿಸಲು ಸಂಕಲ್ಪಿಸಲಾಗಿದೆ. 1949-50ರಿಂದ 2023ರ ತನಕ ಕಾಲೇಜಿನ ಇತಿಹಾಸ ಶೈಕ್ಷಣಿಕ ವಿಷಯದಲ್ಲಿ ಅತಿ ಮಹತ್ವದ್ದಾಗಿದೆ. ಒಬ್ಬ ಧೀಮಂತ ವ್ಯಕ್ತಿಯ ಕನಸು ಈಗ ಲಕ್ಷಾಂತರ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ವಿದ್ಯಾರ್ಜನೆ ಮಾಡುತ್ತಾ ಒಂದು ಸಣ್ಣ ಶಾಲೆಯಲ್ಲಿ 89 ವಿದ್ಯಾರ್ಥಿಗಳಿಂದ ಆರಂಭಗೊಂಡ ಸಂಸ್ಥೆ ಇದೀಗ ವರ್ಷಕ್ಕೆ 3,000ಕ್ಕೂ ಅಧಿಕ ವಿದ್ಯಾರ್ಥಿಗಳು MGMನ ಅಂಗಳದಲ್ಲಿ ಶಿಕ್ಷಣ ಪಡೆಯುತ್ತಿದ್ದಾರೆ. ಈ ಎಲ್ಲ ಸಾಧನೆಗೆ ಕಾರಣರಾದ ಪ್ರತಿಯೋರ್ವ ಆಡಳಿತ ಮಂಡಳಿಯವರು, ಪ್ರಾಧ್ಯಾಪಕರು ಹಾಗೂ ಪೋಷಕ ವೃಂದ ಮತ್ತು ಎಲ್ಲ 75 ವರ್ಷಗಳ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೂ ಧನ್ಯವಾದಗಳು. ಇಂದಿನಿಂದ MGMನಲ್ಲಿ 75ರ ಅಮೃತ ಮಹೋತ್ಸವದ ಹಬ್ಬ ಶುರುವಾಗ್ತಿದೆ. ಪ್ರತಿ ವರ್ಷ ದೇಶದ ನಾನಾ ಭಾಗಗಳಿಂದ MGM ಕಾಲೇಜಿನಲ್ಲಿ ಓದಬೇಕು ಎಂಬ ಆಸೆಯಿಂದ ಬರುವವರು ಎಷ್ಟೋ ಜನ. ದೇಶದ ಪ್ರತಿಷ್ಠಿತ ವಿದ್ಯಾಸಂಸ್ಥೆಯಾದ ನಮ್ಮ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಈ ಮಟ್ಟದಲ್ಲಿ ಬೆಳೆದು ನಿಲ್ಲಲು ಅನೇಕ ಐತಿಹಾಸಿಕ ಹಂತಗಳನ್ನು ದಾಟಿ ಬರಬೇಕಾಯಿತು. ಇಂದು ಬೃಹತ್ತಾಗಿ ಬೆಳೆದಿರುವ ಈ ಸಂಸ್ಥೆಯ ಮೊದಲ ಹೆಜ್ಜೆ ಆರಂಭವಾಗಿದ್ದು ಉಡುಪಿಯ ಮುನ್ಸಿಪಲ್ ಮೇನ್ ಶಾಲೆಯ ಚಿಕ್ಕ ಕೊಠಡಿಯಲ್ಲಿ. ಮಣಿಪಾಲದ ಮಹಾಶಿಲ್ಪಿ ಪದ್ಮಶ್ರೀ ದಿವಂಗತ ಡಾಕ್ಟರ್ ತೋನ್ಸೆ ಮಾಧವ ಅನಂತಪೈ ಅವರು ಉಡುಪಿಯಲ್ಲಿ ಜೂನ್ 27, 1949 ಆರಂಭಿಸಿದ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಮಹಾತ್ಮ ಗಾಂಧಿ ಮೆಮೋರಿಯಲ್ ಕಾಲೇಜು ಎಂಬ ಈ ಜ್ಞಾನ ದೇವಾಲಯಕ್ಕೆ ಇದೀಗ ಅಮೃತ ಮಹೋತ್ಸವದ ಕಾಲ. ಶ್ರೀಕೃಷ್ಣನ ನಾಡು ಉಡುಪಿಯ ಪುಣ್ಯಭೂಮಿಯಲ್ಲಿ ಉಡುಪಿಯ ಮೊಟ್ಟ ಮೊದಲ ಕಾಲೇಜು ಎಂಬ ಹಿರಿಮೆಗೆ ಪಾತ್ರವಾಗಿ ಈ ಭಾಗದ ಜನತೆಯ ಶಿಕ್ಷಣದ ಕನಸನ್ನು ನನಸಾಗಿಸಿದ ಈ ಶಿಕ್ಷಣ ಸಂಸ್ಥೆ ಇಂದು ಬಾನೆತ್ತರಕ್ಕೆ ಬೆಳೆದು 75ರ ಹೊಸ್ತಿಲಲ್ಲಿದೆ. MGM ಕಾಲೇಜಿನ ಸ್ಥಾಪನೆಯೇ ಒಂದು ರೋಚಕವಾದ ಯಶೋಗಾಥೆ. 1947ಕ್ಕೂ ಪೂರ್ವದಲ್ಲಿ ಕರಾವಳಿ ಕರ್ನಾಟಕದ ಜನತೆಗೆ ಉನ್ನತ ಶಿಕ್ಷಣ ಎನ್ನುವುದು ಗಗನ ಕುಸುಮವಾಗಿತ್ತು. ಹೈಸ್ಕೂಲ್ವರೆಗಿನ ಶಿಕ್ಷಣಕ್ಕೆ ಉಡುಪಿಯಲ್ಲಿ ಅವಕಾಶ.
These days filled with knowledge and enlightenment. And now it's time to wrap it up and put the finishing touches on this exquisite event. On this note, I would like to request our beloved principal, Professor Lakshminarayan Karanth, Mrs. Shruti Nayak, Head of the Department Botany, and Dr. Manita TK, Head of the Department Zoology, to escort the dignitaries onto the dais. Audience, please accompany their footsteps with a huge round of applause. Thank you, everyone. A warm welcome is the first sign of hospitality. I now request Mrs. Shruti Nayak, Head of the Department Botany, to deliver the welcome address.
Distinguished guests, esteemed faculty members, and dear students, a very warm welcome to each and every one of you to this auspicious occasion of our valedictory ceremony. Today, we are privileged to have a gathering of eminent personalities who have graciously accepted our invitations to be a part of this memorable event. First and foremost, I extend a heartfelt welcome to President of our valedictory, Professor Lakshminarayan Karanth, Principal, MGM College, whose vision and leadership have been guiding a light for our institution. Your uncovering dedication to academic excellence has inspired us all, and we are grateful for your constant support. Welcome you, sir. I request Dr. Ishwar Bhatt, sir, Department of Zoology, to florally welcome our principal. A special welcome to our chief guest, Sri B.P. Varadrayapai, Secretary, AG Manipal, whose presence add honor and prestige to this ceremony. Your encouragement have been instrumental in shaping the success of this program, and we are honored to have you, sir. I request Mr. Yogesh Kumar, Department of Botany, to florally welcome Varadrayapai, sir. We are also privileged to have with us our guest of honor, Dr. Sripat K.S., Professor of Zoology, Mangalore University. We are truly honored to have you on this special occasion. Welcome you, sir. I request Dr. Vijay Kumar, Department of Zoology, to florally welcome Dr. Sripat, sir. I would like to welcome Mrs. Pravina Kumari, a seminar coordinator, your dedication and hard work behind the scenes have made this event possible, and we are grateful for your effort. Welcome you, ma'am. I extend my heartfelt gratitude to Dr. Manita TK, Head of Zoology Department, for her invaluable contribution to this seminar. Dr. Manita, a silent worker, has been instrumental in making this event resounding success. Thank you, ma'am, for your support and commitment to this program. Your contributions have been invaluable, and we are immensely grateful for your presence and participation in this seminar. Welcome you, ma'am. Last but not the least, a heartfelt welcome to our enthusiastic audience and our dear students. Your presence here today is a testament on this special occasion. Let us cherish this moment together as we celebrate the achievement of our students and look forward to a bright and promising future ahead. Thank you.
Thank you, ma'am. I extend a warm and hearty welcome to you as well. Good feedback is the key to improvement. BIOSEM 2024 was the joint effort of 2 departments and countless people. We would like to know how well our efforts have resonated with you. I request any 2 participants to come onto the stage and to share with us their valuable feedback on this event. Yes, thank you. Please come onto the stage.
Respected dignitaries, conveners, delegates, students and friends, I am Ms. Manasa Bhatt, lecturer from Department of PG Studies in Applied Zoology, Alvas College, Moodbidri, and alumnus from MGM College. The 2 days of this seminar has been very fruitful and thought-provoking. We have learned a lot about several things about taxa ranging from algae to angiosperms, nematodes to vertebrates. The threats to the existent is very worrying and gives us a drive to help conservation in our own locality. I thank all the resource persons for sharing their experience with us. Regular conference like this brings a lot of good awareness among us. I would like to congratulate the organizers for carefully planning out the program and for clear communication with the delegates and for the food and safety provided. Actually, I'm really happy to present my first paper. I would like to thank Manita ma'am for her constant support, and our madam Dr. Rashmi ma'am from Alvas College. I would even like to thank her for her constant support. Even Jean ma'am and Divyashree ma'am for constantly supporting me. I was so nervous for presenting my research paper for the first time. They supported so much. Thank you so much. I'm really happy to be here.
Hello everyone, I'm Muskan Kumari from St. Joseph Vaz College. I thank every participant, speaker and the presenter who presented over here by taking so many efforts and initiating and giving us so many knowledge which we didn't knew. And also I would like to thank my professor, Father Bolmix Pereira, to give me a chance to come here and learn so many things and ask my queries to the speakers. And lastly, I would thank the college, MGM College, to organizing this event in a very big scale and calling all the colleges from surrounding. And living here was very good experience. In the hostel as well, we had really fun. Thank you.
Well friends, I am Father Bolmix Pereira from St. Joseph Vaz College, Goa, and Muskan is my student. And Kamal College and PES College from Goa is also participating in this seminar. And we would like to, on their behalf also, I would like to appreciate this seminar, especially the departments of Zoology and Botany with the IQAC for organizing this national seminar. Last 2 days have been a festival of knowledge for biodiversity and biodiversity conservation. And the organization has been very good. Last 2 days, there was no waste of time. The entire timetable is so compact and well organized. I would appreciate that for doing this minute preparation and getting the speakers to fill the gap without wasting time. The speakers have been very good, sharing the biodiversity conservation knowledge with us. And even before the seminar started, I don't know who we were conversing with on the email, but there was so efficient response from your side that personally I felt like, okay, we can go over here. The organization is good, the people are good, and we can travel far away from Goa to come to this seminar. So I would appreciate that also for being hospitable also. The hospitality that we have seen for last 2 days, we appreciate that. And all the members of the organizing committee, from yesterday till today, I have not seen a face that is without a smile. All faces are smiling faces and so welcoming and ready to answer any query regarding the stay over here and the program over here. So I would like to appreciate that and thank you. Once again, a big thank you for all the speakers who have been very resourceful and of course those who have come with their research work and made the presentations. All were really beautiful. Thank you so much.
Thank you, Father, for the very kind words, and thank you, ladies, for the kind feedback. To be inspired is great, but to be an inspiration is an honor. On that note, I request the chief guest of this valedictory ceremony, Shri B.P. Varadaraipai, Secretary, Academy of General Education, Manipal, to speak a few words.
Good evening to all of you. Respected dignitaries on the dais and off the dais, and delegates of this conference, BIOSEM conference. And yesterday we started with the inaugural session. The session was well begun. As today also I think session is coming to an end, and it's also a good job done by organizing team. And the feedback goes very well. That means we are able to cater to the any audience, not only 100, 200, and you are capable of handling 500 delegates also. That is the caliber of MGM College. And both the departments have worked very, very hard. And this is the first of such conference which is conducted on the occasion of Platinum Jubilee celebration of MGM College. And the entire the event has I think clock precision. So I congratulate entire team and the MGM College for this stupendous work. And this is the beginning. And you are allowed to conduct, I think, another series of 5 or 6 conferences are coming, seminars are coming, and wish you all the best. Thank you, one and all.
Thank you, sir. Thank you, sir, for the kind words. The fragrance of a flower spreads only in the direction of the wind, but the goodness of a person spreads in all directions. Ladies and gentlemen, we have with us today as the guest of honor, Dr. Shripad K.S., Senior Professor of Zoology, Mangalore University. I request Vaishnavi of 3rd BSc to give a brief introduction of the guest of honor.
Today we have with us Dr. Sripada K.S., Senior Professor, Department of Applied Zoology, Mangalore University, Mangal Gangotri, Karnataka, as the guest of honor for the valedictory ceremony. Dr. Sripada completed his BSc in Botany, Zoology and Chemistry from Mysore University in 1984. He then pursued his MSc in Zoology in the University of Mysore in 1986. Post MSc, he obtained a diploma in sericulture from University of Mysore. Dr. Sripada earned his PhD in contributions to cell biology of Indian Chiroptera. He has 30 years of teaching experience at Department of Applied Zoology, Mangalore University. He has received several awards and fellowships for his research. His research interests are ecology, biodiversity and conservation, vermitechnology in waste management, cytogenetics and molecular systematics. Dr. Shripad has completed 2 major collaborative research projects as principal investigator and co-investigator. He has around 47 national and international research publications and 7 book articles. Currently, he is the chairman of Board of Studies in Applied Zoology at Mangalore University. He is also the member of IQAC of Mangalore University. Dr. Shripad is also the external expert for recognition of research center, project proposals and thesis evaluation of various universities. He has organized several extension workshops and seminars. He is involved in preparation of action plan for development of 5 villages adopted by Mangalore University. It is indeed an honor to have you with us, sir. Welcome you, sir. Thank you.
Thank you, Vaishnavi. I now request our guest of honor, Dr. Shripad KS, to deliver his valedictory address.
Very good evening to all. Respected President of today's evening valedictory function, the other dignitaries on the dais, of the dais, I am really not sure whether I justify myself standing before you as a guest of honor, because in front there are really stalwarts who have really contributed in the field of biodiversity in general and various aspects of agriculture. So, my other friends and participants. Now, very rightly, the topic is selected for the conference, Biodiversity Conservation, Impact of Anthropogenic Activities and Climate Change. But many times we come across that many organizations conduct such programs, particularly in the recent past, only on biodiversity and conservations. But when you look into the implication of such program, we are not sure how really it is being taken by the participants and also the outcome of the program. I'm happy that a few participants, they came out and they mentioned about how really the program went. I'm sure that many aspects have been covered. I went through the program and I found that a very interesting topic has been selected by the organizer. When Dr. Ishwar called me on the other day that I will please be the chief guest of the program for the valedictory, I was really not sure that whether I can really make it. But I still, because I love biodiversity and conservation, even though my research area initially was cell biology and genetics, later molecular genetics. But in the recent time when I thought that the students are more interested towards biodiversity conservation aspect, I thought better I can stick on only to this area. But when you look into the way it is being taken, only the studies are being conducted here and there, taking at the regional level or probably at the global level. The statistics say, in the recent statistics say that as the population growth increases, I'm really happy that Professor Naik, the former Vice Chancellor, mentioned a very in-depth knowledge about how really the population is impacting and how really we have improved upon to save our own biodiversity. A lot of aspects have been covered in his, a brief talk. But similarly, I am sure that the other participants also have presented. But what I really understand from the, program, such program, what is the message we can take, either as a student or as a faculty members? Many times we just leave it. In the end, normally there will be a panel discussion. When you make a panel discussion, we come out with some kind of suggestions also, the comments, and also how best it can be taken up further. Now, I'm really happy that Manipal Academy, I know that there are 6 institutions am I right? 6 institutions, I believe, Bandakas College, the MGM College, Karkala Bhuvanendra College, and Mahavira College, Vijaya College, and yeah. Now, when I was a student in, I hails from Sagar, Shivamoga district, and after my graduation, I moved to Mysore. Then onwards, I really like this area, the coastal area particularly. Even though I had little opportunity to go out, Bangalore University I preferred for my place of teaching and also for research activities. Now, we give a lot of importance to coastal region, and at that time there were really stalwarts who have really contributed both not only in zoology, plant biology, and here is a man who has really contributed on botany side. So at that time, we study biodiversity at different level. I don't want to go in detail about all these subject aspect. Maybe at the species level or ecosystem level or at the genetic level. Any level you take, so we know only little, but we know that the species are really is being lost. Ultimately, we say the anthropogenic activity. Nowadays, we talk about climatic change also, or climate change. Now, how really we can stop or probably regain back the loss. Now, each one of us needs to contribute individually and also in group. What is required is coordination, cooperation, and understanding. Now, my earnest request is that whatever contribution earlier these institutes used to do. Now, when we visited recently, when I affiliated college, particularly some of these institutions, I found that the museum they were a good museum. Many of the colleges, even outside also, they are very good. Museum is a place like, biodiversity hotspots and biological park. Museum is a place where we can learn a lot. Nowadays, there are a lot of restrictions. We may not be able to go for, biodiversity species sampling. Even if you sample, probably there will be a lot of restriction, and we don't have a good taxonomist to identify the species. Unless you know the species, and unless it is properly named, we are unable to really, give the conservation status. Now, as you know that the status assessment as per IUCN, International Union for Conservation of Nature, every day we are losing the species. Maybe as the population increase, as before it was mentioned, that 10 times more than that, the species are lost. Even though the estimates say that there are 10 million species, only hardly less than 2%, 1.7%, some people say, that has been named and they have been grouped. Now, because of lot of ambiguity in the taxonomic identification, now people are finding it very difficult to place them in different IUCN threatened categories. Now, what is to be done? Now, in this direction, even though efforts are being made at the national or at the global level to save the species, a lot of legislations are there, but we are not following very strictly. Even if it is followed, but they are not being implemented properly at the regional level, maybe at the higher level also. Now, we can adopt only those techniques which is really not harming much on these diversity. We may not be able to save these species unless otherwise we protect the habitat. We concentrate on species. We say that they are endemic species or very critically endangered species, but we are really not sure what is their needs. What are their needs actually? Now, the studies are, even though conducted for many groups, taxon, mainly on vertebrate groups, very less on invertebrate or other microorganisms. But we are really lacking a lot of information on those areas. Now, as we say that, diversity part, only 2% of the land is found to be assessed. It is assessed nowadays. Now, biodiversity hotspots concept, even though it is there, now people say that hardly some areas are being studied in detail. Certain areas are still unable to be accessed. So, based on the data, certain critically endangered species have already gone. Now, there are ways by which they can be saved. Now, recent technology can really make further our conservation effort to be strengthened. Now, earlier we used to go to the different areas for assessing species diversity and the status assessment. Nowadays, satellite data can be obtained, or now the recent advancement is drones. Drones can really give a lot of information, particularly on the status of any kind of vegetation as well as a certain group of animals also. So, and in addition to that, somebody was talking about when I, briefly I was there in the presentation, the nutrition analysis. Now there are sensors are available. We can make use of in the field some of these sensors. We can see what is the quality of the nutrition or the soil quality, so that we can say which kind of habitat really can be managed and protected, which in turn save the species. Now my good advice for the youngsters and also the young researchers and also the faculty members. Now I am really worried because there are only few faculty members are left in these colleges. Really starved, only few permanent faculty members. Now there are a lot of problems. They are being transferred here and there. There is no proper way by which these teachers can concentrate on motivating the student. Very difficult. Now what is most important for us now? Whatever is the facility or whatever is the limitations what we have. In the short time, we can do something better to save our species. On one hand, there is huge loss. On the other hand, climate, also anthropogenic activity, human activities is further being increased. Now all these colleges, the faculty members and the students, they can, like this conference, my suggestion is they can organize a conference in such a way that all, maybe botany, zoology, putting together, not only from the same college, from other institution, in joint. They can organize a big program where students can be drawn and they can be really trained how best the diversity assessment can be done. Now, when I used to attend many of these CMP workshop, when I say CMP workshop, conservation assessment management plan. Now, regularly it is being conducted by, under the direction of Conservation International at different, institution at the national level for different taxon. Now, the difficulty is even though the papers are being published, the papers are not complete. The data are not complete. When when I attended few invertebrates and vertebrate taxon data assessment, we found that the data are very ambiguous and very difficult to give the status. Ultimately, it will go to the IUCN data book. No, we found it very difficult. Why I'm telling all this? Now, the students are the young researchers who are make any kind of studies. Please be very systematic, even if it's a small contribution you are making. Concentrate on only one particular area, see the diversity. Maybe if you are clear about what kind of taxon we can take, see that are there any good taxonomies. Now we have Botanical Survey of India, Zoological Survey of India, but still these institutions also have a lot of problem in identifying good taxonomies for a particular taxon. The students make a lot of assessment, but ultimately the species identification is not being properly being done. That is the situation right now. Anyway, so I'm very much thankful to the organizers and the participants in particular, because they have taken a lot of interest to come over here. Please be serious in your studies, not just for the sake of attending the conference or organizing the program. We have to do it. Ultimately, there must be some data which can be recorded, which can be taken up further. So, once again, I thank the organizer and other dignitaries on the dais and off the dais and all the participants for inviting me for the program. I wish a beautiful future. We see that all of us will join together to see that our nature is protected and we'll leave something to our next generation people. Thank you very much.
Thank you, sir, for the enlightening words. I now request Mrs. Praveena Kumari, Seminar Coordinator and Assistant Professor, Department of Computer Science, to speak a few words.
Good evening all. On account of Amrita Mahotsava, to commemorate 75 years of glorious existence of MGM College, series of seminars, conferences, workshops are being organized. In the series, first one organized by Department of Zoology and Department of Botany. It's Bioscience 2024, 2 days national seminar. All the sessions were fruitful. We are thankful to all the resource persons and the delegates for accepting our invitation and for their active participation in 2 days national seminar. We congratulate all the paper presenters. We say first impression is the best impression. In the series, first one is 2 days national seminar BIOSEM 2024, and it went on smoothly. We hope upcoming seminars also will be organized in a fruitful way. Thank you.
Thank you, ma'am. We have now arrived at the much-awaited moment of finding out the winners of the Best Paper Award. I request Dr. H.P. Gurushankar, Assistant Professor, Department of Zoology, Central University of Kerala, Kasaragod, to announce the winners. And I request our guest of honor, Dr. Shripad K.S., to kindly bestow the honors upon the winners. Over to you, Dr. Gurushankar.
Yeah, sir. Yeah, good evening to all. You know, it's a expected moment. Most of them are awaiting. Who will be the winner in this oral presentation? First of all, I thanks organizing team to assigning this tough work to me, particularly our Iswar Bhatt sir and other team is a Madam HOD, Zoology Department and Plant Science, and other our co-chairs, they are also helped me to decide the winner of this oral presentation. We have decided, we have selected the winners based on their content of their work, clarity, presentation skill or style, then their slide preparation. Already our co-chairs clearly mentioned about their font size and everything. Some of the slides are not visible. It should be minimum 24 size font size, or in a clarity it should be bullet point. Instead of bullet point, entire abstract they are keeping in one slide. It's not so visible. Okay, and their clarity and question and answer, how they have answered the questions raised by audience and our team. Based on that, we were selected 2 best presenters. Any guess from the audience? Any guess? No guess. Yeah, I think just we are not going to give first prize or second prize or first awardee or second awardee. Just we selected best 2 presenters. First one is D'Souza from Goa, Andrea D'Souza.
So I request Dr. Shripad sir to issue the best paper award prizes.
Yes. I think you go center. Middle. The next best presenters from Mangalore University, Alvas College. I hope Alvas College. Mangalore University. Okay. So Jean. Okay. Jean Cycle. I hope, our chairman can give, I think secretary can give this one instead of our Sripad sir. Yeah, you can. Congratulations to winners, best presenter winners, and also congratulate all the presenters. Thank you.
Thank you, sir, and hearty congratulations to the winners. Now, I would like to request our principal, Professor Lakshminarayan Karanth, to present a certificate and memento to the paper presenters who have participated in the oral presentation sessions conducted on both days. Participants, I request you to please come onto the stage as your names are being called and accept your certificates. Ms. Pratiksha Sail, Assistant Professor, Department of Zoology, Carmel College for Women, Goa. Please give a big round of applause. Mr. Keshav Murthy, Assistant Professor, Department of Zoology, G. Shankar College, Ajarkad. Ms. Karen Braganza, Assistant Professor, Department of Zoology, Carmel College for Women, Goa. Dr. Balmax Pereira, Assistant Professor, St. Joseph Vas College, Goa. Thank you, sir. I now request our chief guest, Shri B.P. Varadrai, to present the certificates for the rest of the paper presenters. Dr. Ashoka C.H., Associate Professor, Department of Zoology, Dr. P. Dayanand Pai, P. Satish Pai, Government First Grade College, Mangalore. Mrs. Divya Shri, Assistant Professor, Department of Zoology, Vivekananda College, Puttur. Ms. Manasa Bhatt, Assistant Professor, Department of Postgraduate Studies in Applied Zoology, Alvas College, Modibidri. Ms. Sneha Bhatt, PG student, Department of Postgraduate Studies and Research in Biosciences, Mangalore University. Ms. Sneha Bhatt. Okay, thank you. Thank you, sir. It is now time to honor the chairperson and co-chairpersons of the oral presentation sessions conducted on both days. We are immensely grateful to them for accepting our request and lending us their precious time on both days. I request our chief guest, Shri B.P. Varadaraj Pai, to kindly do the honors. First, I would like to invite the chairperson of the oral presentation session, Dr. H.P. Gurushankar, Assistant Professor, Department of Zoology, Central University, Kerala, Kasaragod, to please accept the honors. Sir, please. Please give Sir a big round of applause. I now invite co-chairperson Dr. Sukesh, HOD in the PG Department of Botany, Alvas College, Mudubidri. I now invite co-chairperson Dr. Lavina KB, Assistant Professor, Department of Biosciences, Mangalore University. Thank you, sirs. Thank you, ma'am. It is now time to acknowledge the gratitude and admiration we hold for our guest of honor, Dr. Shripad Case. I request our beloved principal, Professor Lakshminarayan Karanth, to present a memento to the guest of honor, Dr. Shripad Case. Thank you, sir. I now request our beloved principal, Professor Lakshminarayan Karanth, to kindly deliver his presidential remarks.
Respected dignitaries on the dais, invitees, my dear students, today's seminar went well. First of all, I thank our respect management for sponsoring this 2 days seminar. Not only that, from yesterday onwards up to this end of this semester, we have seminar from each department. And as I told in the yesterday, coming October, we celebrate our Amrita Mahotsava program. So on that behalf, we conducted seminars in various departments. So I'm very much grateful to our management, especially our trust president, Satish Pai sir, and our always helpful secretary of AGE, Professor Varadaraj Pai sir, for sponsoring this seminar in a very beautiful manner. I take this opportunity to thank you and Satish Pai sir. And I also thank Department of Zoology and Botany organizing this seminar in a very beautiful manner. This actually we started yesterday. This is actually the stepping stone for other seminars to take place. So they have almost taken 1 month preparation for this, both the HODs and all the members of the department, Ishwar Bhatt sir, Yogesh sir and others. Say they have done lot of work. Even I think this seminar is is not even less to the university seminar. Even the UG college, they have conducted in a very beautiful manner. So I thank both the department for organizing this program. And I mainly thank delegates from different places. Right, even now it is already 5:45. Look at how many participants are. So once again, from the our MGM college side, I again thankful to all of you. So lastly, my point is that from 2 days here, a lot of discussions. So 7 invited talks and 14 papers were presented in this seminar, mainly to conserve biodiversity and climatic change. My dear invitees and friends, with the government support only, with the proper resource planning in the country or any kind of the action of the government only, we can preserve our biodiversity. If it is not there, we cannot expect sustainable development in future. So seminar has discussed all these aspects in a very beautiful manner. Once again, I request my department is that whatever the 2 days programs are there, so they have to write it in a separate manner, that we publish in our Amrita Mahotsava, this bulletin or other books are concerned. So once again, I thank organizers for conducting the seminar in a very beautiful manner. Thank you, one and all.
Thank you, sir. Gratitude is the fairest blossom which springs from the soul. As we move towards the final part of our valedictory ceremony, I invite Dr. Manita TK, Head of the Department Zoology, to present the vote of thanks.
I also thank Professor M.K. Nayak, sir. So senior most person. See, I have forgotten to tell him. See, in our college we have the, butterfly park. That's actually his idea. So I think, have you seen or not that? So you can just go through and see that. So he is always with us. For our all kind of activity, he is always with us, sir. On behalf of the college, I thank you, sir.
Good evening, one and all present here. As we are concluding the session, I would like to thank you all for gracing this occasion. First of all, I would like to thank our principal, Professor Lakshmi Narayana Karan, for his unwavering support. His vision and leadership have guided us every step of this way. Thank you, sir. Now, I would like to express my sincere gratitude to today's chief guest, CABP Varadharaipai, for his esteemed presence. Your words of encouragement and wisdom have inspired us all to strive for excellence in our endeavors. Thank you, sir. Next, I would like to express my gratitude to Dr. Sripada, guest of honor, expert in zoology, for gracing this occasion. Thank you, sir. I would like to thank seminar coordinator Praveena Kumari, ma'am, for her support and dedication and contribution and involvement in organizing the seminar. Thank you, ma'am. Next, I would like to thank Mrs. Shruti Nayak, HOD of Botany. Without Botany, zoology is incomplete. It was a wonderful journey together in organizing this seminar. Her contributions, involvement, I thank Shruti ma'am for her contribution, dedication and involvement throughout the completion of this event. I express my deepest gratitude to Department of Journalism, MGM College, for their constant support in conducting this seminar. I thank Mr. Sujit Kotian, HOD of Journalism, Mr. Manoj Kumar, Faculty of Journalism Department, and their team for documentation and live streaming of this seminar. Next, I would like to thank Mr. Vineet Rao, Coordinator, Gandhian Study Center, for all the support from the beginning to the end. Thank you, sir. I would like to thank Mrs. Latavi Nayak, Assistant Professor of History Department and Warden of MGM Hostel, for providing accommodation facility to our delegates. Thank you, ma'am. I would like to express my gratitude to all the former faculties. Your presence and involvement in the seminar made it fruitful. Thank you all. I would like to thank our judges for sparing their time in MGM. It was a tough task for selecting the best papers, but you have done your job very well. Thank you, all the judges. Next, I would like to thank all the faculties of MGM College for the moral support and encouragement. Thank you all. I would like to thank all the resource persons, speakers, volunteers, participants, and all the members of organizing committee who have contributed their time, effort, and expertise to make this event a resounding success. Thank you all. Once again, I thank MGM College Trust for funding the seminar. Thank you. As we conclude this seminar, let us carry forward the knowledge and insights gained here today and continue our collective efforts towards the conservation of biodiversity for the betterment of our planet and future generations. Thank you once again, and may we all strive to be stewards of our environment, ensuring a sustainable and harmonious coexistence with nature. Thank you.
Thank you, ma'am. A very sincere thanks to you as well. Your organization of the seminar has been impeccable and instrumental for the success of this seminar. The end of a story is a new beginning for many others. On this note, let's conclude a remarkable and memorable valedictory session. This is Ramya from the Department of Botany, signing off and bidding adieu to you all. Now let's officially end this seminar by rising in unison for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंधु गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
I request the resource persons and the organizing committee to stay back for a group photo. Resource persons, I request you to kindly come onto the stage for a group photo.
Boys.